Hi everybody, welcome to the celebration of the 25th anniversary of the Department of Ecology and Evolutionary Biology at the University of Michigan. Yes, I love it. But we're here not just to celebrate 25 years of being a department, we're here to celebrate the long and rich history of ecology and evolution at the University of Michigan. As some of you might know, the very first professor hired by the University of Michigan was Asa Gray, who was a leading ecologist and evolutionary biologist, probably the strongest proponent of Darwin's theory of evolution in the United States, which really set the stage for this university being the place for ecology and evolution. And we're here to celebrate that with all of you. I am Nate Sanders. You have all received many emails from me over the past weeks and months and years. I start a sabbatical on July 1. You will not receive an email from me for an entire year. So feel free to not look for messages from me. For the alumni who are in the room, thank you all for coming. I know you all have busy lives. You're doing many things. Some of you are writing memoirs that you're not calling memoirs, you're calling other things. You're all incredibly busy. You're taking time out of your life to come here. I really appreciate it, as we all do. Before I get too far into this, I want to thank the people who really made this event happen. Jen Wolf is standing right there eating oatmeal. She's going to wave. Yeah. Jess Grunmeyer is right here. She is the events coordinator. She is really the person who made all of this happen. Yesterday, the entire EEB staff and the facilities was in the room making all of this happen. For those of you who know this room, it typically does not look like this. And so Rob and Dave made all of this happen with the EEB staff. One of the amazing things about this event is that, if you had grad ecology with me, you know that I really believe in the history of this department. I see Matt Hack nodding his head right now. He took the class and was a GSI. The grad ecology class is really, to introduce students to how folks go about doing ecology, but also how folks at the University of Michigan have done ecology. People in this room know more about this than I do, but if you just, list all of the major advances in ecology and evolution, 95% of them, I'm biased of course, came from the University of Michigan, and we really highlight that in our grad ecology classes. I look around the room and I see the pillars of ecology and evolution staring at me, and it makes, I'm getting a little bit sweaty under my arms thinking about it. But more importantly, I see the future of ecology and evolution. These grad students who are sitting, around the room, you can look around and see them. This is really the future of ecology and evolution. And so this event is so special because it blends where ecology and evolution came from, but also where it's going. And so we're here to celebrate that. And so thank you all for doing that. We all know that, much of the research in ecology and evolution that anyone does anywhere came from this place. But at the same time, this place is also dedicated to teaching undergrads, reaching out to them. And I see, some of the very best undergrads I've ever interacted with sitting in the room right now. They were excited to be here. They're going to present posters this afternoon at 5 p.m. that I hope you all will engage with. We care a lot about teaching in this department, and so one of the things I want to do before I hand it over to John Vandermeer and Deborah Goldberg is celebrate some of the excellent teachers we have in the room. Some of the best teachers we have are some of our grad students. And so we annually have a GSI award. It's a big deal. They're nominated by students, by faculty, and not only do you get a really fancy certificate that I'm going to hand out, but perhaps more importantly, you get $1,000 for being one of the best GSIs in the department. This year there are 3 GSI awards that we're giving, and these folks were nominated by multiple people. I see one of the people in the room right now Max, come on up. Max was nominated by several people for his work in ornithology. Everyone said he went well above and beyond. His passion for birds is evident. You all know that. So Max, congratulations. I haven't seen this person yet. Is Ivana in the room? No. So Ivana Barnes also is an outstanding GSI. And Adia Mosley McLeod, who I also, Adia, where? Don't see Adia. So if it's tradition in the department, if the person isn't in the room, the money goes to the chair. And so we also want to celebrate some of the work that many of our grad students do, folks in the room do. In this department, we celebrate justice, equity, diversity, and inclusion. This year, the JEDI Award goes to Sean Richards, who I know is not here, but you all know Sean. He's an incredible citizen of the community. He has done a huge amount of work in the Bahamas to get STEM activities infused in high schools. He's not here, but he wins the JEDI Award this year. So congratulations, Sean. And we also always celebrate the best paper by a grad student in this department. There are many papers published by our grad students, as you know, and all of them are really, really good, but this year 2 of them are great. And the first award goes to our friend Mateus, who I saw sitting right over here. Congratulations. Yep. You all will be seeing many papers from Mateus. You've seen many already, but look for more on the way. He's going to be a postdoc with us for a couple more years. And the second best paper goes to Natasha. Is Natasha here? Yes, Natasha, yes. So Natasha has not only been an incredible scientist, as you all know, but Natasha has been instrumental in, thank you, congratulations. Yeah, shake, there we go. Natasha has been instrumental in building this community and keeping grad students in touch with faculty and faculty in touch with grad students. And so Natasha, thank you very much for all that as well. Couple of quick announcements. So if you're new to the building, if you need to go to the restroom, go out the door and turn right. I'll be keeping time for the first set of talks, and there's a coffee break at 5 o'clock. There's a poster session, but if you're doing a poster, set them up beforehand. And I think that's all of the logistics. And so now I get to introduce 2 of my idols, John Vandermeer and Deborah Goldberg. I think everyone in this room knows who they are and what impact they've had on this department, but also on ecology and evolution. I think John Vandermeer, he will tell you this, so I will say it first. He is the oldest person in the room. And has been affiliated with the University of Michigan since I think The Origin of Species was published, if not sooner. And so John is going to take 15 minutes, and then Deborah's going to take 15 minutes. So John, welcome to the stage.
Thanks, Nate. This on. So can you all hear me, right? So I'm going on 86 this year. Am I the oldest? Anybody can top me? No. I know the 2 are closest to me, but so I have all that to say. I have only 15 minutes to say it, so I have to say it really fast, okay? I arrived here. I'm supposed to do the older stuff, so just begin with 1966, which is when I first arrived here. I was a graduate student in 1966 in the PhD program. And what I discovered here, I don't know exactly how to say this in a way that captures the truth of it, but I found an incredibly vibrant community. And since I see Doug Futuyma and Rob Caldwell right there, they're two of the people that I continually think of when I think of that vibrancy. We were constantly in conversation, constantly in debate. We had the fourth floor ecology group, where it wasn't just the three of us, but a group of other people. I wish that Doug Gill was with us, but he's not here with us right now. But anyway, it was an incredibly vibrant time at the University of Michigan. I'd specifically like to call out Doug Futuyma, my great friend from a long, long time ago, for two things, Doug. One time he said to me, he doesn't know what I'm going to say, by the way. He's a bit nervous. Anyway, first one, he took a course in OTS in Costa Rica. And when he got back from that, he was so incredibly enthusiastic. And I remember the very day he looked at me in the eye and he said, nobody can consider themselves an ecologist unless they've been to the tropics. That changed my life, Doug, because I have been to the tropics at least once a year ever since, every year except for the COVID year, okay? Secondly, with possibly that same conversation, no, it wasn't that same conversation, a little bit later, Dick Levins had just been here and he had given a talk, and Doug was really impressed with the talk, but Doug looked at me and he says, I really think you ought to go do a postdoc with Dick Levins. And so I did, and that changed my life, Doug. Okay, so Doug changed my life. Rob and I also had a lot of interaction together, a lot of intellectual discourse between the two of us, but we defended our thesis more or less at the same time. We had a joint thesis party, celebration party, at Marson Base's house. So those are really great times. I have been reminded by Deborah Goldberg, and she'll tell you more about that when she gets up here, but if I don't say this, she's going to get really pissed. That time was really intellectually vibrant, but it was also famous for something else. It was famous for something that was not very inclusionary. Yes, we were a bunch of white guys, and we liked to talk with one another. But at the time, I think there was one woman faculty. I believe Sally Allen, right? She was the only one. One woman faculty members, and really very few graduate students even who were women, and certainly no African Americans, despite the fact that we're very close to Detroit. She was right about that, okay? So on the one hand, there was this vibrancy that I recall with great joy. On the other hand, there was this problem that existed, and it's a problem that persisted for a long time. When I did my postdoc, I wanted to mention something about, I have to keep my time going here. I wanted to mention something about my postdoctoral years because they were tremendously formulative for me and sort of generated what I became when I came as a faculty member here at the University of Michigan. I did a postdoc in the Committee on Mathematical Biology at the University of Chicago with Richard Levins, but Richard Lewontin and Dan Jansen were sort of equal co-advisors with me, and that group was an incredible group. So my time there was incredibly influential. I don't know how many times I'm going to be able to say incredibly here, but well, then I arrived at the University of Michigan in a faculty position after spending a year at Stony Brook in a faculty position where I went, as Doug reminded me last night, with Larry Slobodkin. But for personal reasons, I could not stay there. The faculty at Stony Brook was really quite good, and I was happy there intellectually, but personally, there was an issue with, I don't know how you say it, Long Island, I think they told me yesterday. And so, for personal reasons, I had to come back to the University of Michigan. Came back to the University of Michigan, and when I came back to the University of Michigan, those were pretty high political times. We were in the middle of the Vietnam War, and for me, having just been in Costa Rica and seeing what was happening in Costa Rica, actually getting sort of out of the field stations and talking with some of the farmers, not talking with them, but seeing what was going on, and watching the massive expansion of the banana plantations taking over tropical rainforests, why I could immediately see the struggles going on in Central America were going to become important. And so that was brewing at the same time when I first came. Inside the department, inside the department, the first thing I tried to do, basically because of my political perspective, was to set up a diversity committee. When I tried to set up a diversity committee in 1972, everybody laughed at me, okay? It was not a thing that was even thought to be possible. What is it that you possibly mean by diversity? We're doing great, okay? And so that happened very, very quickly. And that extended, it was an incredible issue in the department. I think that those of us who were here at the time don't fully understand how important that was. Let me give you just one example. A young woman from a Latin American country came here to do a master's degree. She could read English perfectly fine, but her competence in English to understand what was being said to her was not all that great. So she was taking a course from a faculty member. Nobody here was that faculty member, okay, I guarantee it. From a faculty member in zoology, it was biology as it was at the time. And the way she describes it, without even looking up from his desk, he answered her questions. And her question basically was, I'm having a little bit of trouble, because English is not my first language, I'm having a little bit of trouble with the lectures. Could you give me some extra readings or something? And all he said was, well, if you can't muster the material in the course, you just better drop it. So that's what he said to her. She dropped the course. And, well, I was going to say that was it, but that's not it. Turns out that story continues, because she did find a community of people, mainly graduate students who were associated with the organization Science for the People, and an offshoot organization called the New World Agriculture and Ecology Group. And it was basically a study group, study and political action group that she got incorporated into, and they provided her with that sort of a background, that sort of a community, a community which was both intellectually, culturally, and politically satisfying to her, and that enabled her to become, as a matter of fact, a professor. And so she currently is a distinguished university professor at a R1 university, and has all sorts of honors, including a membership in the American Association for the Advancement of Science, the American Society, and the National Academy, and some other stuff too. Anyway, that was a success story ultimately, but not because of the culture of the department, because of the culture of something that was in the department. That's something that was in the department is something that I always wanted to incorporate somehow in a more formal way for the department as a whole. I was going to, at this point, I was going to have a call out to David Tillman, but I don't have time. I'm really sorry. Okay, time's running out. I'll tell you what it would have been later. Okay, it's good. It's okay. It's good. It's good. Anyway. The next experience, and now I got to go through this pretty quickly. I encountered a group of people. I was invited to participate in a committee, a university-wide committee, based on the grant that the university got from the National Science Foundation for Advance, which was to diversify the faculty, especially with regard to gender. When I first joined that committee, my idea was this was going to be a political action committee like others I had been in, and we were going to do, I don't know what we were going to do, because they were all senior faculty members, at the time. And what I discovered was it was a group of people. There was there were professors from, I wrote it down here, professors from chemistry, MCDB, mathematics, computer science, and aerospace engineering, and myself. And what it was, we got together for an entire semester, and we basically read the literature. It turns out there's a huge amount of literature in economics and sociology, political science, and other social sciences disciplines. And we spent that semester just reading the literature. It was like a graduate seminar, It was very, very cool. And so I was totally surprised. I thought it was just going to be a political action group like others I had been in. But then it sort of began morphing into a, quote, political action group. And the group as a whole, that group, the advanced group, we decided, well, first of all, we decided to name ourselves, calling, it was the STRIDE committee, which is, I think I can remember, it means strategies and tactics. Well, it's an acronym, okay, but it's a very good acronym. Okay? It became the STRIDE committee. And we put together a slideshow, and the whole idea was to go to different departments and tell the departments, give the departments information as to how they might proceed with diversifying their faculty. And luckily, Doug Futuyma was chair of the department at the time, and Deborah was sort of his lieutenant. I can't remember what you actually were, but the two of them, they not only welcomed our group to give this presentation to the department, but they almost insisted that we do it as soon as possible. So EEB was the very first department that got that rap from the STRIDE committee, and that turned out to be really important with the feedback we got from them, and then it went on and on and on. It's still going on, and now every search committee at the university is required, anybody on a search committee at the University of Michigan is required to take the STRIDE workshop before participating on the committee. We'll say that the results of all of that activity has been that the university is much better, still not where it should be, but it's much better in terms of gender issues. The balance of male and female faculty member has changed, as has the diversity of the graduate students. But the STRIDE committee, then in later years when I was involved in it, we took on the issue of race and ethnicity. At that time, there were approximately 6 to 7% African American students in the department, and I don't know how many, but fewer than that faculty members in the university. While I can say I think we made progress in terms of the gender issue, I don't think we've made any progress at all, I'm sad to say, in the race and ethnicity issue. I would like to say some negative things about the university. I have nothing really negative to say about the department of EEB, really, but about the university, there's some negative things to say. And Deborah said it. Deborah thought I shouldn't do this, but I'm going to do it anyway. I remember the University of Michigan. This didn't happen when I got here, but I do know the history of it, and that is who became a really pretty good friend of mine, Chandler Davis. He was in the math department. And he was fired by the University of Michigan under the presidency of Harlan Hatcher, whose name still occupies the graduate library here at the University of Michigan. He was fired because he refused to testify before the House Un-American Activities Committee in the 1950s. He did go on, he left the country, went to Canada. He had a perfectly fine academic career in mathematics, but he and two others, one of whom was in our department, were actually fired. Tenured professors that were fired because of their political affiliations. I don't know if you think that might be relevant to something today. And then my personal experiences throughout history of this. I've been involved in a lot of political projects at the University of Michigan, and at the level of the university, the response has not always been positive. As a matter of fact, frequently has been really very negative. But I will say that to be positive about this negative part, the students at the University of Michigan have been really magnificent. Let's not forget that the very first teach-in against the war in Vietnam happened here at the University of Michigan, which expanded in the rest of the country, by the way, and in my view really did contribute to the ending of the war in Vietnam. The Port Huron Statement was written by students from the University of Michigan. That became the platform for Students for a Democratic Society. So I have a lot of other things to say there, but time's running out, and I just have one quote here. Let me read you this thing, what I say here. Let's see. If I can, oh, yeah. Students good, administration bad. That's what that says. And I finally would like to say, as I have written here, ode to the University of Michigan. The University of Michigan has treated me very, very well. It has provided me with a home. It's a home where I can engage in very, very stimulating and I think productive activities at a research level, at a teaching level. It's provided me a place where I can engage with the things that I'm passionate about from across the entire spectrum. And I'm really grateful for that. And so grateful to the University of Michigan for that, and frequently to the Department of the EEB. Several chairs of the department have been very, very supportive of my extracurricular activities, okay? Maybe they don't want to be called out for that, but I thank them for all the work that they did on my behalf also. So thanks.
So, Deborah Goldberg, who I've already introduced, many of you know. Deborah was a long-standing and long-serving chair of the department among many other accolades. And so, Deborah, I trust that you can click on your presentation that you uploaded 7 minutes before the symposium started.
Deborah will never change. Where's the slideshow bit here?
Bottom right.
There we go, bottom right. There we go. Okay. Oh, yeah. There?
Yeah, turn it on.
Oh, turning on would help, yeah. I managed to turn the slide on. Yeah, Is this okay now? Yeah. Great, okay. So I have to say I'm a little bit overstimulated here because I'm seeing so many people that I haven't seen in anywhere from 6 to 30 years, and a lot of wonderful hugs, and I'm kind of losing track of what I was going to say here. So this might be a little discombobulated. Nate, I didn't give Nate a title, so he said reflections on the program. So I am going to reflect on some history and tell some personal stories, but history overall. Talk a lot about the beginning of EEB, where I was the interim chair at the beginning of EEB. We'll talk more. I want to talk about the relationship of EEB, Museum of Zoology, and the Herbarium, because that's incredibly important to what makes this department so wonderful. And then, if I have time, we'll talk like a brief list of some of the things that I'm personally proud of for having done while I was chair. So, and okay, so let's start with some history. Nate already talked about that first professor of botany, Asa Gray, so I don't have to do that. After that initial start, the illustrious start to biology at Michigan, things more or less followed the usual process for departments in biology across the country. There were first this horizontal separation into botany and zoology, separate departments. Eventually those merged into a department of biology. In our case, we had a formal merger in 1975 to a division with separate departments with various names that kept changing all the time. The only thing those departments in the division did really was the graduate program, which was incredibly important, but they didn't have to deal with the rest of that. That got formally recognized in 1986 by becoming a department of biology, and then those subdivisions became even more fluid, but they still basically did the graduate programs. And then in 2001, as we'll get back to, we did the vertical split into EEB and MCDB. Now, I'm going to do a little personal stuff here. I arrived about, 1983 as a brand new, very naive and very young and skinny assistant professor. And here's where I'm going to get back to something John was talking about. There were not many women in the department at that time. When I came, I think there might have been 4 of us, 4 or 5 out of 30, 40, whatever it was in biology. John's counting, I can see. And it wasn't so much that there were overt acts of sexism. There weren't that many of those, or very many, but there was certainly an atmosphere, and that was exemplified by something happened. One of the stories I want to tell, just even before I arrived here. I came in the summer before I started my faculty position to talk with the department chair who hired me, Mike Martin. And I just want to say up front before I tell the story, I thought Mike was a fantastic chair. He was very fair, he was balanced, he was incredibly efficient, and in many ways he was a model for how I wanted to be as a department chair when I became that much later on. But when I came to talk about my lab, he took me down to the basement to the office of the building manager, Jim Might, who some of you may remember. And on the way down, he said, I just have to warn you. And he said, Jim was in the Navy, and he was in a submarine, and he still has some of those habits. No idea what he's going to do. I go into that office, and there were pictures of naked women all around his office. And I'm just so shocked, I hardly know what to do. And what I want to say here is that Mike was great. I love Mike, I respect him enormously, but the best he could do at that time that he could think of to do was to warn me about it. Not make sure he could take him down, not fire Jim. He got fired a couple of years later for other things. So that was kind of the atmosphere. That was the way things just were. Okay. Oh, to be fair, one thing about, just go back, one more thing about that. A couple of years later, we had some construction in the building, and there was those toolboxes that contractors have running in the halls, these big, huge metal boxes. And there were, again, some pinup pictures on those. And then I had enough confidence to go complain. And they disappeared immediately and never showed up again. But, again, that stuff just happened. It took active complaining to get rid of that. As John says, things are a lot better. They're not where they should be, and they're going a little backwards right now, but things are a lot better. Okay, so John alluded to this as well, and I should say, John and I go back a long way. He was actually a full professor when I was hired. I've now been retired 7 years. John's still going strong. We've been doing this back and forth in teaching in seminars this entire time, and I think we kind of played it up. But the idea was we were going to amuse the students to do that, so we try. Anyway, Michigan had a reputation as a very scary place. That incredibly vibrant, intellectually vibrant, exciting place was absolutely true. But along with that, and perhaps as a consequence of it not being very inclusive, and also causing it, was the fact that it was very aggressive intellectually, and sometimes even nasty. And I learned the phrase when I came here, raving cladists. I didn't even know what a cladist was before I got here. I didn't have that background. I wasn't in a place with a lot of museum people. So when I came, but I actually felt, but I was lucky in that I felt incredibly protected. I was lucky enough to have some amazing mentors when I first arrived as an assistant professor. Beverly Rathke, who quickly became a really good friend and helped me who to talk to, who not to talk to, how to interact with the somewhat difficult people in the department. Brian Hazlett also helped me navigate a lot during those early years. Sadly, they're both gone now. Beverly, just within a year after she retired. Brian, just earlier this year, which I was really sad to hear. But I do need to talk a little bit about John, which, when I came, I was, as probably all assistant professors, but maybe even more the women, I didn't belong here. I wasn't good enough to be here. John took me seriously. He took my ideas and my science seriously. And we debated and argued, and we rarely agree on anything, but it made me a much better scientist, but even more important, it gave me confidence. So into becoming the sometimes sort of loudmouth scientist that I am these days. So John, I thank you for all of that time. So.
Smoking.
And I'm not going to get sentimental here. We even wrote a book together, which one of the students just asked me to sign it, which was, you could get John to sign it too. Okay, back to the timeline. Keep track of time here. So, the split. We had a divorce, and I'm going to just talk about a number of questions related to this, to the split in the beginning of EEB. One was who wanted it. This is a picture of the department in 2000, just before we split. And you can notice the lack of diversity there, partly, but it was, the ostensible reason was that the dean, we had an external review, the external review committee, the external review committee recommended that we split, so the department was too unwieldy, et cetera. But I think a lot of us felt that the subtext was, and some people here can correct me, that really MCDB was really pushing the dean to do this on the grounds that EEB was kind of holding them back. That the fighting over resources, i.e. faculty lines, meant that they couldn't expand in the ways they needed to because of all these other positions. So maybe that's unfair, but that was certainly the way they felt. EEB people were not as enthusiastic about splitting, even though, yes, there was a fair amount of acrimony at times and a lot of debate and arguing over resources. I can remember some really awful faculty meetings. The ideal of a unified biology was still there. But in the end, that was clearly going to happen. And in the end, we decided maybe that was a good thing. And so it went ahead. And you had a question of who's going to lead it. I was appointed interim chair, and that is known as the chair's chair. That was given to me as a present at the end of that first year as interim chair. It was actually painted by Anne-Marie, Prosanta's wife. And I still have it, and I'm still very proud of it. And I want to see a picture of it. So, but now I get to tell another brief story about when I was appointed interim chair, which was when I went to meet with the dean about how I'm going to be about being that interim. I sort of gathered all my courage. I wasn't really good at these negotiations, and I said to the dean, well, I understand that when you become a chair, take on an administrative role, you get a raise in salary. And she looked at me with a completely straight face and said, oh no, that happens afterwards. That's a reward. You don't get that in advance. And I just said, okay, whatever. I'm just, as I said, I was still pretty naive at that point. I'm preparing a year later to go to my annual meeting with the dean, and Nancy Smith, our administrative manager, looks up salaries, and she sees the interim chair of MCDB got a big raise at the beginning. And I go back to the dean and have my committee, and I bring this up. I said, you said this, but that happened. And she looked at me again with a perfectly straight face and said, he negotiated harder. And I just want to tell you, the dean had come from a place where she was chair of the Women's Studies Department. She knew damn well women are not good negotiators, but she still let it go by. Saving money for college, I guess. Anyway, I got the raise, but good stories. Oh God. All right, what to do about the children? Joint custody, that's what usually happens. Divorce with joint custody of the undergraduate program. What to call it? That was the biggest debate. And the big debate was, should we have organismal biology in the title or not? So we have ecology and evolution. That was clear. That was what we had often been called. I think everybody in the new department felt that organismal biology, the basic functioning of single organisms, was hugely important. So that wasn't the issue. We weren't arguing about this is an important thing to do, but we were arguing if we do that, we had almost nobody in that area. So that meant we were going to have to use those, again, precious faculty lines to hire in those areas and therefore lose some of our amazing strengths in ecology and evolution. And so we went back and forth, a lot of very serious, intense discussions, and ended up very reluctantly not having organismal. We felt if we kept it in there, it would be false advertising. So, but it's still something I worry about enormously. In that I think where departments have split into this, you basically lose that whole organism. Yes, we can often do it around the edges, physiological ecology, evolution of morphology, but that core, how do single organisms function, is really, again, Michigan, where is it? It's kinesiology for vertebrates, and that's kind of where it is. And so that's a real loss for training. And I felt that gap. I was in an ecology and evolutionary biology department for graduate training at Arizona, which, by the way, has just celebrated its 50th anniversary as an EEB department last month. Judy Bronstein organized that symposium. I felt that gap my entire career because I didn't have that core training, and I worry about it for students. So it's just something that I think we still need to think about, and how do we solve that problem? And I'm gonna just skip all this stuff because I didn't get to, because I talked too much. Okay, I do want to say that just one piece of history that people may not know. The Museum of Zoology and the Herbarium were not always in the department. They reported independently to the dean. In the first decade of the department, the dean kept pushing to merge them into the department, I think more to simplify his life than anything else. Took a lot of debates. Obviously, a lot of people were very unhappy about that. We spent a lot of time talking about it and making sure that the department as a whole fully understood the importance, that why we are so great is the existence of the museums. We can't do it without them. And so we developed a statement of principles. We figured out how to deal with curation. There was some curation versus teaching. We managed to deal with all those issues, and I'm sure nothing ever gets completely solved, but it's one of the things that I am really proud of is, I think, how we did that. And I think I'm gonna just, I'm just gonna go there. I just want to thank, first of all, Jen and Giota for helping me with data at the last minute, and all the fabulous colleagues and staff and students that have been here. It has been a huge honor and privilege to be associated with the department and to have been chair, and I'm really happy to be back. So.
Good morning, everybody. Okay, so my name is Léo Laborieux. I hope everybody can hear me, yeah? I'll say, okay, so my name is Léo Laborieux. I am a first-year PhD student in Archibald Swain's lab, and I do a variety of things that range from scorpion taxonomy to global biogeographic patterns of species interactions and distribution. And today, it is my great pleasure to introduce Dr. Futuyma. So Doug actually graduated twice from UMMZ, first in 1966 with a Master's of Science, and then in 1969, if I'm not mistaken, with a PhD. Nowadays, he is a distinguished professor at the Department of Ecology and Evolution at Stony Brook University in New York, and also a research associate at the AMNH, the American Museum of Natural History. Among many other accomplishments, he is a fellow of the American Association for the Advancement of Science, as well as the American Academy of Arts and Sciences, as well as a recipient of the Civil Rights Award at the American Society of Naturalists. But it's a long list. It's actually quite amazing. So please join me in welcoming Dr. Futuyma.
No, I guess this will do. Huh? Okay. Well, thank you very much for that kind introduction. Can you hear me? I have no idea. Can you hear me well? Okay. And before I get into my own remarks, which are going to be rather brief, I would just like to pick up where Deborah finished, which was mentioning in particular the herbarium and the Museum of Zoology. Which even though, as I will tell you, I sort of dissociated myself from the museum, nevertheless, I more and more appreciate the importance of those institutions. I think they are really critically important for maintaining the knowledge and research on the diversity of organisms. That is, fishes and insects and plants in and of themselves their diversity, their characteristics, their distribution are objects of, I think, very important study. And this is part of, I guess, my feeling that the old term natural history is a term that should be considered an honorific. The title, this is partly more or less based on suggestion of what to talk about. How the University of Michigan's Department of Zoology shaped my career. So I'm just going to tell you a little bit of my own biography. And it starts with growing up in the Bronx in New York City. And that turns out to be one of the best places to grow up if you are going to become a naturalist. And that sounds incongruous, growing up in New York City, but the fact is that the Bronx has 3 large parks with extensive natural areas. It's where I became a birder early in high school. And one of those 3 parks, Bronx Park, includes the Bronx Zoo, which I think for much of its history was the largest zoo in the United States, that is to say with the greatest diversity of animals. And I became essentially addicted to going to the Bronx Zoo. And so for much of high school, classes would finish at 3 o'clock and I'd get on the subway and go up to the Bronx and spend a couple of hours at the Bronx Zoo. And there, I basically, because I spent so much time there, I came to know some of the working personnel. And in particular, I was absolutely infatuated with amphibians and reptiles. And I spent countless hours visiting the reptile house. I could have told you the distribution and the scientific name of every frog and every lizard and every snake that they had there. And so here I came to know some of the personnel. And that included, the Bronx Zoo at the time had 3 curators: curator of birds, curator of mammals, and curator of reptiles. And the curator of reptiles, it should have been reptiles and amphibians, was James Oliver. And he who went on to become the director of the American Museum of Natural History. And he was a herpetologist. He became a mentor to me and a number of other boys. And I continued to see him occasionally when I would come back from Cornell and to visit family at home. And so I maintained some contact with him and with the museum as an undergraduate. And as an undergraduate at Cornell, where I should say I was greatly influenced by 2 people, Bill Brown, William J. Brown, who was like the world's, I think, authority on the taxonomy of ants, but was really interested in speciation, and he had his own somewhat unorthodox ideas about speciation. And Bruce Wallace, who was a student of Dobzhansky, and taught population genetics. And that's where I really began to crystallize my interest in evolutionary genetics and diversity. And so the result of that was that I became more and more interested in herpetology, and on Oliver's recommendation, I applied for and was admitted as a graduate student in the Department of Zoology here at the University of Michigan, with the intention of studying herpetology. And so I had a desk space in the museum, in the herpetology division. So I arrived here in September 1963. And as I said, I had a desk space in the museum. And the 2 curators of herpetology, the 2 curators of reptiles, were Charles Walker and Norman Hartwig. Hartwig worked on turtle taxonomy. He and some of the other museum curators were among those who were instrumental in creating the Organization for Tropical Studies, OTS. And that led me to take one of their very first courses in Costa Rica. And later I taught in several of those courses, and that's where I think I first met Judy Bronstein and Steve Arnold and Rob Caldwell, and basically, much of my early experience was in the OTS. Norman Hartwig died the first semester. No, it was the next year, leaving Charles Walker as the only herpetologist. And Charles Walker was, he had some absolutely outstanding students, like Tom Ozell and Craig Adwer. But Walker was one of the shyest people, I think, and most reclusive people that you could imagine meeting. And I had almost no interaction with him at all. He would basically sit in his office at his desk, basically all day long, except for the faculty coffee breaks. And so I had very little interaction with him. And so for this, so instead, I found my intellectual stimulation among fellow graduate students, some of whom are in this room, like John, and as well as the ecology faculty, and also Richard Alexander, I'll mention again in a moment. Some of the most influential aspects of life in the department, the zoology department at that time, were Wednesday afternoon seminars in which there would be typically a guest speaker. But before that, there was a seminar meeting of graduate students only, in which graduate students gave presentations about their research ideas and so forth to other graduate students, which was a wonderful institution because they could feel, basically not feel as if they were under examination by the faculty. So that was one of the important aspects of intellectual growth here. Let's see. And I also attended, and that becomes most important in my career, the weekly meeting of the ecologists, ecology faculty and students. And so the most prominent ecologists, of course, were the famous trio of Nelson Harston, Fred Smith, and Lawrence Slobodkin. And these lunch meetings were really among the most stimulating events that you could go to. And so here, I ended up having an interaction with Larry Slobodkin after he gave a talk in which I really thought the idea he was promulgating about tropical diversity, I just thought made no sense at all. And I more or less afterward came up and told him that I think, Dr. Slobodkin, I think I disagree with something you said. To which his answer was, okay, good, you give the next week's seminar. Which was not a great way to start a relationship with the man who became my PhD mentor. But he sort of forced a degree of independence, shall we say. And so that interaction really changed my career, because I then basically moved over toward the ecologists, focusing on evolutionary ecology. Thank you. And Larry arranged for me to spend a summer learning population genetics in Chicago with Dick Lewinton, which pointed me toward my dissertation research, in which more and more I became interested in putting together ecology and evolution. And so my dissertation ended up studying genetic aspects of competition between species of Drosophila. As you know, Larry went on to create the Department of Ecology and Evolution at this new campus on Long Island, which became Stony Brook University. And he lured me, he lured John Vandermeer for a short while, as well as luring the infamous Steve Ferris, whom some of you will know about. But of these, only I am left to tell the tale. Finally, I want to again mention and credit Dick Alexander for the key role that he played in my development. He and Tom Moore in the insect division worked on acoustic insects, cicadas and orthopterans, and that led Dick into an interest in speciation. And he led, as a result, he led a year-long reading of the very large book by Ernst Mayr, Animal Species and Evolution, in which we met weekly, and one or another member of the group would lead the discussion. And we went through, literally line by line, that entire large book, taking notes and making criticisms, all of which we sent to Mayr, who, as far as I know, never read it, but we sent him. Anyway, this was a group of, including people like Mary Jane West Eberhard, who went on to, just a really great career. And I would have to say then that this experience, this entire experience that I've described to you, really shaped my interest in speciation and the evolution of diversity, and that had a lasting effect on my career, and I am immensely grateful to this Department of Zoology, as it was then, at this university for my career.
Thank you very much. I think that's Aimée's mic there. It is my immense pleasure to introduce...
It's weird.
Yes, the director of the biological station, professor of ecology and evolution in the department, one of the world's leading scholars in global change and soil ecology.
He has to say that.
And the mother of two incredible boys, who I also know very well. And so while she's doing that, I will keep their... Yes, great. So, Aimée Classen, the love of my life, and a world-leading scholar. I was trying to make that as weird as possible.
Thanks. It's really hard to follow Doug. I have had the pleasure of being able to interact with him on an editorial board, and he is an absolutely stellar storyteller. So if you have the opportunity while he is here, please meet with him, because he has incredible stories and he's just really fun to interact with. It's a pleasure to be able to talk to you guys today. I am the director of the biological station. We have a booth outside. Incredible things are happening at the biological station. So please stop by and talk to us. But I wanted to talk a little bit about our science with you all today. I haven't been here very long, so I don't have a big, rich history to present. I've only been here for almost 6 years. And I guess if I were to reflect about my 6 years here, I would say that we have an incredible junior faculty here. I mean, they are inspirational, amazing. We have these incredible people. And so it's fun listening to your stories, because I can't wait to come back and listen to them tell their stories. And I don't know how old I'll be, but I'm really excited about that. Okay, so I'm going to tell you a little bit about our science. And this is a picture of me in 2011. I was really chuffed at this point in my life. I just put in my tenure package. Woo! I had gotten an NSF grant to host this big meeting in Iceland of a bunch of ecosystem ecologists and earth system modelers. And I just started doing work in Colorado and thinking about interactions along gradients. And so I was really, really obsessed with how models, these gigantic earth system models, should be including interactions. If we're missing the interactions, how can we understand a system? The modelers at the time were very patient with me. And they're like, Aimée, we don't even have phosphorus in the models. Do you think it's really more important than a phosphorus feedback? And I was like, well, And so they told me, if we were to include these interactions in models, we would need to spend 7 years of our lives figuring out the math. And so it's like, you go out, you do a bunch of experiments, and you prove to me that we need to put interactions in models. And I was like, okay, because I was young. I was like, okay. So I'm going to talk to you a little bit about that work and sort of what we've been thinking about. Before I start, I just want to say thank you to all of the students, postdocs, collaborators, undergraduates who have shaped our understanding and thinking about these ideas. I've really been lucky and blessed to work with a just incredible group of human beings. So I think it's probably no surprise to anybody in this room that ecosystems are reorganizing, right? You go out, you can see that things are looking a lot different. We know that constraints are shifting, such as water and season length and temperature, and they can alter how processes, which I'm an ecosystem ecologist, I love the processes, can change. We know that these mechanisms are starting to be kind of uncoupled organisms and substrates and timing, and these taught some of our prediction breakdowns. So we wrote a paper in 2015 highlighting that global change ecologists really needed to start thinking about interactions interactions among organisms as well as among organisms and abiotic constraints. So we've been working on this for quite a long while, and like I mentioned, we recently had started working in mountains. Mountains are incredible places to explore these types of questions because they're temperature gradient, right? You can go from warmer places at low elevation to cooler places at high elevation. They're also interaction gradients. So we know that organisms tend to compete at lower elevations and be more facilitative at high elevations. So it kind of is like a mix of all my best things, working around these elevations. They're great study systems. So when I was a young ecosystem ecologist, which is a while ago now, we always thought about processes and temperature, and that net ecosystem carbon balance should shift in very predictable ways. And that very predictable way was this way, right? That as temperatures heat up, processes get faster. So warmer is faster. We would expect things like increases in productivity, decomposition, and respiration. It should be really straightforward. We should be able to model this like that, right? Very easy. But what our research group has been doing and finding in a lot of different areas is that this prediction doesn't necessarily hold. So I'm going to show you some data from a global scale experiment where we went out and we measured gross primary productivity using these little boxes at places all around the world. So we had three mountains. In each of these different countries. We measured the gross primary productivity of trees and ground layers and the gross primary productivity of just ground layers along these elevational gradients. So again, we have temperature that would decline with elevation. So we'd expect to see very obvious patterns in carbon cycling. So here again, this is Argentina. I just want to highlight again that we've got GPP of ground layers with a line at the bottom and the GPP of trees and ground layers at the top. Not at all what we expected. There's absolutely no universal temperature-carbon relationship. And this is not the only experience we found this. Like sometimes our graphs look like spaghetti at the wall, right? Like all kinds of different outcomes from these different places. And so what do we do about that when we're thinking about carbon modeling? So why are these responses so variable across different places? Well, for one thing, as an ecosystem ecologist, I'm obsessed with process, right? Photosynthesis, respiration, decomposition. But what might be driving some of the outcomes, sorry about saying driving, Doug, anti-driving. What drives some of these outcomes are some of these interactions. And there's a big gap in how ecosystem ecologists think about process and these interactions and modeling. So ecosystems are not coordinated systems, they're interacting ones. So that's the point I'm going to try to make today. All right, so to do this, I'm going to talk about an experiment that I had the absolute luck to work on. This is a 30-year warming experiment started by John Hart in the Colorado Rockies in a high elevation montane meadow. And after 30 years, we were able to go and harvest these sites to look and see how they'd changed over 30 years to like a 1.5 to 2 degree warming. And we were curious about whether responses were coordinated around these different plant compartments, which really just means like the above ground plant parts versus the below ground plant parts and the interacting organisms. And so here we have our graphs. We're going to always have our ambient and warmed on the bottom. We'll have our above ground plant biomass, shoot to root ratio, below ground plant biomass, and microbial biomass. So not unexpectedly, we see that with warming, we get an increase, 64% increase in above ground biomass. But what was more interesting is that we didn't really see a response of below ground biomass, which we would have expected because we know it's getting hotter and drier. So we would have thought more roots would be below ground, but we didn't find that. We didn't find much of a change in microbial biomass, which we also thought we would see. So that was a bit surprising. We know that above ground processes are sensitive to warming in the system, but there doesn't seem to be any coordination between these sensitivities in above ground and below ground plant compartments. So we also know that the strategies that these organisms are using is changing. So here we have plant functional traits, and we see that there's a negative response of specific leaf area and a positive response of leaf dry matter content, which means these systems are really becoming more conservative. And you can see this highlighted in a picture. Right over here we see a beautiful, very flowery ecosystem during a very wet and cool year versus a hotter and drier year on the bottom. So you can really see how temperatures can impact these above ground communities. So what about the breakdown? Are we seeing a shift in these organisms? Are their traits changing, and are we seeing shifts in these plant organisms? And in fact, we do. We see that there's an increase in shrubs, right? So we're getting a shrubification of these systems. We see a decrease in forbs and a decrease in graminoids. But what's even more fascinating, is that we're starting to see a change in our below ground subsystem, which is these AM arbuscular mycorrhizal fungi and saprotrophic fungi. What's interesting about this is you would think as plants get more stressed under heat and dryness, they should have more associations with mycorrhizal fungi, right? Mycorrhizal fungi are supposed to help plants persist in these sorts of environments. But what we find is that that's not true. We see a large increase in saprotrophic fungi, but a decrease in mycorrhizal fungi. And so there's some sort of breakdown between what's happening above ground and below ground. Okay, and so you might be like, right, Aimée, but this is just in the soil. We don't know what's really happening in the plant roots, and we know that mycorrhizae associate with plant roots. Maybe this is just because we're getting a change in species composition. But in fact, that's not true. So this is fungal colonization. And what we see is that even in the plant roots of mycorrhizal plants, they're ditching their mycorrhizal fungi. Super shocking. The ecosystem is becoming more stressed, and even the mycorrhizal plants are losing their mycorrhizal fungi. So this was really interesting to us, and we were thinking like what does this mean, this coupling between above ground and below ground breaking down? It seems like these ecosystems are becoming more like deserts, but we were curious about other processes that also drive systems in response to warming. And one of those is snow. These sorts of ecosystems just really love snow. Snow is like a blanket on the system. Microbes are really active in the winter, and if you get an early snowmelt, it can really change those plant soil feedbacks. And so we're curious about if we look at warming or if we look at early snowmelt, do we see different responses? Okay, 4 minutes, I got it. I talk fast. And what we found was like, yeah, we do. In fact, earlier snowmelt, we see a large increase in below ground sensitivity for roots, nothing above ground, and like just a small change in mycorrhizal fungi. So these two different drivers of global change that should be coupled, warming and snowmelt, are actually moving these systems in different directions, right? So we're getting this differential response with two different drivers of global change. So, this is interesting, and I'm going to make a little pitch here. This is a review paper that we wrote as a group a number of years ago. We looked at all of the global change experiments around the world. This was a 1,119 manipulative experiments. And what we found was that there was no response of net primary productivity, above ground net primary productivity, below ground net primary productivity, gross ecosystem productivity, net ecosystem productivity, or ecosystem respiration. And like, wow, right? Like, wow, like ecosystems aren't responding to warming? Like that absolutely can't be true. And I will say, I am part of this paper, and one of the lines we wrote, nonlinear, including synergistic and antagonistic effects among global change drivers, were rare. So I was saying that maybe these interactions don't matter at all. Like, oh, like crazy, because we know these systems are responding to warming. Ecosystems are responding to warming. But what I would like to argue is that the constraints and mechanisms are creating these reorganizations that look like stability, but are changing. So what we see are these, like, are they responding? Yes, but maybe they're responding in one way to snowmelt and another way in temperature. Or maybe they have different decomposers that are changing their geographic boundaries and shifting how decomposition happens, which changes carbon feedbacks, but we don't predict them yet because we don't think about these really important ecosystem interactions. And so I'll sum up to say that here is me in 2011. I've spent way more than 7 years thinking about this process and how we include ecosystem interactions potentially in models. I'll say that, do they matter? Yes, they do. But what do we do about it? I don't know. And that's one of the things I would love to chat with all of you as we go forward today. So one of the things I think a lot about is that we could look deeper and move beyond very rate-based measurements in ecosystem ecology and think about how we can identify some of these dominant constraints in ecosystems and how interactions can be some of those dominant constraints. We could explore data from different perspectives and think more closely about nonlinear, non-additive, and interactive temporal experiments. And so with that, I will say that ecosystems are not coordinated systems, they are interacting ones. So thank you very much. You want my phone?
No.
This doesn't seem to be working. It is? You hear it? Okay. So folks are standing up in the back there. Don't forget there are coffee and snacks in the back. There are cool drinks coming in. During the coffee break, we'll bring some more chairs in and put them across the back row. I will make this introduction less awkward than the last one. It is, you want to?
Does this work?
I don't know yet.
Yeah. Is the tech person here by any chance? I was just wondering if I could advance without having to stand right here, but I'm not sure. Anyway, there's usually a little dongle that gets plugged in here.
So it's my pleasure to introduce Catherine Badgley. Catherine is a professor in the department, as you know, and is the director of the residential college. If you ever have had the opportunity to go into the field with Catherine, it is an amazing experience. She knows so much about landscape ecology and every fossil and every rock you can ever pick up. And the times I've been in the field with her have been amazing. And as you all know, she's an incredible citizen of the department and inspiration to me. So, Catherine, take it away.
Thank you so much. And actually, let me, thank you, Nate. That's very generous. Let me just mention something I heard on my drive into work today, because this is relevant to what I'm going to talk about just now. And in a way, I hope it's relevant to all of us. Today is the 100th birthday of David Attenborough, who is, one of the best publicists for biodiversity. So I'm going to talk about the fact that 2 months ago, I was in the Power Center giving a TED Talk. Turns out there's a tradition organized by a team of undergraduates at the University of Michigan of putting on a TED conference every year, and it's been going on for more than 20 years. So how did this happen? I'm not sure how I got invited, but we won't go into. I was invited to submit an idea back in October, along with a whole bunch of other folks, I guess. I was selected as a presenter the following month. I started working with a coaching team in December. This is a coaching team of undergraduates, none of whom are science majors. I had weekly meetings with these undergraduate coaches, starting in December, proceeding all the way into March. The first rehearsal was in early February. There was a revision of both the script and the rehearsal itself in late February, a dress rehearsal on stage on the 13th of March, and then the program itself was Saturday afternoon on March 15th, with 8 presentations in all. And I took very seriously all the advice I got from my coaches, and what I want to do now is just summarize a few, maybe about half of the highlights that I presented in the program, and then tell you at the end what I learned. My main coach was Aimée, who was a freshman in the residential college, and she was absolutely terrific, and I took very seriously all of her recommendations. So the theme of the conference, which was established by the undergrads, was radiance. So I used that word, and my title was Achieving a Radiant Food System. So I started out by saying, we are on the threshold of a mass extinction, and the major cause is our food system. And I want to tell you that we could have a radically transformed food system that would have a very different outcome from what's happening today. But then so this was the question that I posed, that I take up in my entire TED Talk. Can a radically better food system protect biodiversity, improve our health, and combat climate change? Then, with the advice of the coaching team, I said, let me tell you how I came to this topic. I grew up on the side of a mountain in Colorado. And from an early age, I came to love the big skies, the meadowlarks, the wildflowers, and the vast landscapes of this area. And I spent many hours walking over these meadows with my dog. And I knew early on that things were missing from this landscape. I should have seen pronghorn antelope, rattlesnakes, elk, wolves, bears, or at least signs of them. But they had been either hunted out, poisoned out, or displaced by expanding farms and ranches. So this became a motivation for me to become an advocate during part of my life for the plants, animals, and landscapes that we share the planet with. It was also my first awareness of the modern biodiversity crisis as a child. And that led me to study biology and earth sciences in college and graduate school, and then some exciting opportunities in graduate school to start working with an early paleontology expedition in Pakistan led me to study paleontology, the history of life. And there I came to learn that there are lessons from deep time that are very important for us today. And one of those lessons is that the present time is highly unusual. We are seeing extremely high rates of extinction. This is a chart showing 1,000 species in these different groups that have become extinct since the year 1500. And if you convert this into rates, this is about 1,000 times higher than the background rate in the fossil record. So this is and we now that it took me a while to realize that the major causes are food system, and this led me and my husband Jerry Smith, whom I know many of you in this room knew, or to move to a farm over 30 years ago, a farm west of here, what we call Sandhill Farm. And here you see me in my garden with sandhill cranes in the background and a draft horse. And here we learned both what it means to coexist with a native biodiversity, even when you're losing some of your animals to them, and realizing that those predators, foxes and hawks, they belong here too. They were here before we were. It also told us something about what it takes the effort it takes to grow your own food, especially growing your own food with agroecological methods. So here are a few more glimpses of the farm. A drone view, the Three Sisters garden that I plant every year, and here is the evidence of some of the neighbors that we share the farm with from the tracks on the ice. And so every day this is a place for me to look at the challenges and the opportunity in the food system. So back to the main topic, the current food system is leading us toward disaster. However, it doesn't have to be that way. We could have a radically transformed system that would do quite a bit better. And so what I presented in my talk then, I said I will tell you in a few moments about the radiant food system, but in the first I want to tell you about 5 dimensions of disaster. And what I learned from working on ArcGIS story maps in a class this year led me to diagram these things differently than I would in a traditional lecture, using simply a big number that highlights the main point. So here, 4% of wild terrestrial mammals of terrestrial mammal biomass is wild mammals now. Most of it is livestock and humans. 45%, almost half of the world's land, is used in agricultural activities, and much of that is for livestock, or food for livestock, even though livestock provide only 17% of global calories. Industrial agriculture dominates global food production, even though most of what's grown here is livestock feed or food or crops for biofuel. Very little is directly for human consumption. And 28% of all the assessed species in these groups are threatened with extinction now, both in and out of agricultural lands. And 1/3 of greenhouse gases emanate from the food system. So these were the 5 dimensions of disaster. And you can see that without going into a lot of text, one can try to get the point across. Well, it doesn't have to be this way. We can do much better. So then I went on, again at the recommendation of my coaches, to tell stories of some of the farms I visited in a class that Yvette Perfecto and I have taught a number of times called Food, Land, and Society. Here we're visiting a farm that's just north of Ann Arbor, 3 acres. And the farmer grows primarily microgreens and salad greens. And after we'd spent a day with a tour, we'd done some experiments baiting for ants, of course. And we were standing around talking with the farmer at the end of the day, and one of the people asked, "Have you ever calculated how much biomass you grow and harvest here?" He said, "Well, actually, I did have somebody weigh that up all last year." And it was 27 tons. 27 tons on 3 acres. By the end of the day, we were asking ourselves, if Rob can grow 27 tons of microgreens on 3 acres, can't organic agriculture feed the world? And a group of us got together after the semester was over, undergrads, grad students, and ourselves, to tackle that question, and we eventually wrote a paper about it, and the answer is absolutely yes. So that also gave us the conviction that a transformed food system could do very much better. So then in my talk, I presented a vision of what a transformed food system would be in the year 2040. And why a vision? Well, a vision enables you to see into the future what some of the important aspects of a transformation would be, and then you figure out how to get there. So I want to give you a few highlights of my vision. The year is 2040. The Secretary of Agriculture has an advanced degree in agroecology, and this is Stacy Philpott, who's a graduate of our department and currently a professor at UC Santa Cruz, and I think she'd make a great Secretary of Agriculture. her steering of the Farm Bill into much more agroecological directions has now enabled organic farming to occupy 25% of U.S. farmland compared to just the 3% it occupies today. And this is my neighbor's organic farm, Tantry Farm, for those of you who know it, where we have not anything like a monoculture with maybe 12 different kinds of crops being grown in the same field. And because there's now been all the subsidies that we now have for synthetic fertilizers are gone, we use nitrogen-fixing cover crops that have replaced synthetic fertilizers. And diverse organic farms like this beautiful farm in Chiapas that Yvette and I took our students to is now harboring all manner of native insects, birds, and amphibians and mammals, as these farms do if you actually go and monitor them. And changes in dietary habits have been such that the United States population, for better or worse, is now consuming quite a bit less in the way of animal products. And in addition, in this vision, farm workers all receive a living wage. And so the whole vision that I painted in the talk is of a food system that actually facilitates biodiversity conservation and draws greenhouse gases out of the atmosphere. Furthermore, the dietary changes have led to a decline in obesity rates, heart disease, and stroke, and so the healthcare costs across the country have also gone down. And of course, social justice and food justice in particular are critical to this system because they benefit not only consumers, but especially producers, and they ensure social stability and social enthusiasm in this kind of system, rather than all of the griping that we hear about our current food system. So how can we all become agents of change in this transition? This is what I turn to next in my talk. And I'm just highlighting a few of the things that I presented. Well, actually, we have climatic crises that are coming upon us, just like the ones that Aimée talked about. And in fact, I just heard recently that we are potentially in for a super El Nino in the coming year, which may lead to much more severe droughts than we're accustomed to. This is going to lead to acute crop failures in many parts of the world and possibly massive death of livestock from heat stress. These are tragedies, but these also give an opening. These show the vulnerabilities of the industrial food system. They will give an opening for the radiant food system to achieve more prominence. In addition, the changes in dietary habits means there's less livestock on the landscape, fewer conflicts between livestock and native carnivores, and so the animals like wolves and mountain lions, which have been demonized for decades in the United States, will actually will have better chances of being protected. Now, all of these changes that I paint in the talk are highly relevant to Michigan, which is already a very agricultural state and poised to become even more important as drought and heat stress make the areas that have been the traditional bread baskets of the United States much less effective in the coming decades. And finally, it's important for all of these changes to be supported by a fair food policy, so we should advocate for that whenever we can at the voting booth and for the politicians that we support. The beneficiaries of a transformed food system would be the general public, farm workers, the farm economy, and native biodiversity. The losers are going to be the large agribusiness corporations that provide seed, synthetic chemicals, as well as the major food retailers. And these are huge corporations that have dominated food policy for far too long, both nationally as well as globally. So in closing, in the TED Talk, I talked about the fact that achieving a radiant system has great momentum already with vast beneficiaries. And that by doing so, we would create a world that is more just, a world of stronger relationships between the natural world and the human world, between farmers and consumers, between rural and urban communities. I said the world is asking this of us now, and I firmly believe that. And finally, the rewards are personal, societal, and world-changing. So that was the end of my TED Talk. So now just let me finish this talk with what I learned. Well, I learned, as many of you already know, the value of conveying many of the fundamental tenets of ecology and evolutionary biology for things we care about, topics we care about, to a general audience. And this talk was very well received, and I got lots of feedback immediately. And then, since this is now on the internet, on the TEDx platform, I've had people write to me from other parts of the world It's also the value of relating my research interest to my own personal background and to my life choices. That's not something I usually get a chance to do. And I think, as many of you have already done today with your stories about the history of the department, I think that I was told by the coaching team, this humanizes us in a way that just standing up and talking about research doesn't. I also learned the value of having a freshman coach. I mean, this is the most flipped classroom experience you can imagine. And it was wonderful. I mean, it's wonderful because you realize, first of all, you realize we are always students, no matter how old we are. And that's great. That's one of the reasons we stay in academia. But it also means that we have a lot to learn from these earnest people who are just starting out, and that they are eager to learn what we know if we will present it to them in a form that they can understand. And then finally, I learned simply how to make the talk more interesting. So I would like to close by encouraging all of you, no matter where you are in your career, graduate students, undergraduates, retired people, everybody in between, find an opportunity to do something like this, or even create it within your own departments. It doesn't have to be a formal TEDx program, because, and have the people with the least knowledge be your coaches, because they want to learn and they will help you become better communicators. So thank you all very much. We're on to the coffee break.
So we have time for a 15-minute coffee break. We'll reconvene at 10:45. Also say at the end of the day, we'll have time to ask questions and communicate with our speakers as well. And so, Catherine, thank you very much for a great talk.
It's not, yeah.
I think it's standing under.
It's a little weird. All right, find a seat. About to start. There is, yeah. Catherine got it to work. Yeah.
You gonna go through mine?
Find a seat.
Oh, the pointer.
Oh yeah, don't use a pointer because there are 3 screens.
I can't hear you.
What's that? Don't.
So you're going to make it illegal?
Yeah,
to
that's
make
right.
it clear? Oh, we're going to make it unclear to
That's
everybody.
right. That's right.
He's gonna circle the whole screen. He's gonna do this the whole time. And everyone's like, this
Yeah. All right, find a seat. We're gonna get started.
Amy, I got a pointer. I'm serious.
I think. Yeah, we do. All right, we are going to get started in 20 seconds. Yep. All right. You can't hear it. Can you hear me? I will turn up the volume. All right, let's find a seat. Alright, if you're not in a seat, you don't get a seat. Alright. Take it away.
Thank you. Good morning, everyone. My name is Patricia Torres Pineda. I'm a PhD student in the Lopez Fernandez lab. I study the evolution and ecology of freshwater, tropical freshwater basilids or life-bearing fishes. And it is my pleasure to introduce Professor Prosanta Chakrabarty, who graduated with a PhD in ecology and evolutionary biology in 2006 from Michigan, and is now Director and Curator of Fishes at the Museum of Natural Science at Louisiana State University. He is also Professor in the Department of Biological Sciences in Louisiana State University, and the Edmund K. Hunter Chair for Communication in Science Research. Among many other awards, Dr. Chakrabarty is a Fulbright Distinguished Chair, an elected Fellow of the American Association of the Advancement of Science, and a TED Senior Fellow. Please welcome Professor Chakrabarty.
Thanks, Pat. You're standing right next to me, but I still have to. It's really an honor to give one of these keynotes for EEB, the department that I love and that made me. And I changed the subtitle a little bit from what's in the schedule because I realized I was flipping through this A Naturalist in the University Museum by Alexander Ruthven, this 1931 book written by Ruthven on the 25th anniversary that he was director of the museum, and he was also serving as president of the university. And so he was playing multiple roles, and when I was an EEB student, we were at the Ruthven building. That's where the UMMZ was at that time. And I started in 2001, and this was an interesting time for evolutionary biology and natural history. There's lots of parallels between what I was seeing in 2001 and what Ruthven was talking about in his 1931 book about data. He was even talking about the importance of data. And I have to, this is a time where many evolutionary biologists in 1931 were eugenicists. There's many reasons. I was looking through this book, I was like, he must say something that would get him canceled today, but it's actually an interesting book. There's nothing in there. I wasn't looking for it, but I was like, he's going to say something somewhere, but he doesn't. And there's actually this wonderful Venn diagram that he's hand-drawn. This is 1931 again. And I used ChatGPT to clear it up a little bit. I didn't take a shower today to make up for the water use for the... But this diagram is very much something I would use today. He talks about public engagement. This is kind of my version on the left here. And many of the things he's talking about are still things that evolutionary biologists working at museums think about still today. And so I'm going to couch this talk in 10 lessons I learned as an EEB student, in the first cohort. And the first one used to hang this quote, "A finished museum is a dead museum," used to hang in the Ruthven building and was attributed to him, even though he probably never said that and someone else had said it well before him. So I'm now myself a director of a museum and at LSU's Museum of Natural Science. And we're known for our birds and our genomics resources collection. And I wouldn't have gotten this job had I not been a grad student here. I worked on the genetic resources here. I worked on fishes. I learned how to be a curator and a scientist. And so my position is wholly due to this, and I see lots of parallels between our two museums. Like Doug, I grew up in New York City, but not the Bronx. I grew up in Queens. And I went to McGill University in Montreal, and then I applied to grad schools while I was working at the Bronx Zoo, and I got a call from Bill Fink telling me I got the job while I was working at the zoo. Maybe we've met then, I don't know, Doug. And so then I did my PhD starting, I was 22, which I can't believe is 25 years ago, or more than that. And then went on to use the Old Boys Club to do a postdoc at the American Museum of Natural History with another EEB graduate, John Sparks, and at the American Museum. And then I've been at LSU for 18 years, which is, I cannot believe this amount of time has passed, with a couple years at NSF in Ottawa. And I started writing a book. Well, I didn't know it was going to be a book when I was a grad student here. That was sort of advice to newer grad students. So I thought as a senior grad student, it would be important to pass down like what I had learned, not knowing what I would use it for. And this is not a tell-all book. I forgot who said it. Was it Deborah that, or no, Nate said we're all writing our memoirs, but not memoirs. This is not a memoir. But the book, and I don't recommend it because it's now 10 years old, but it made me realize how much advice I got from senior grad students myself and other folks in this department, because that helped me sort of articulate what I thought were helpful tips for being a student and then going through. Now, when we were the first EEB cohort, we were hanging out a lot. We had softball games, we did a lot of karaoke, and because my wife had a job, Anne Marie had a job, we had HBO, so people would come to our house and we'd watch Sopranos and we'd watch Curb Your Enthusiasm. And this is my favorite quote from the show: remember when is the lowest form of conversation. This entire talk is remember when. And so there'll be Sopranos references throughout this talk. Now, I'm a New Yorker. I was watching Sopranos. 2 weeks, this is not just the 25th anniversary of EEB. 2 weeks after I moved here from New York, September 11th happened. I had no family here. My wife hadn't joined me yet. I was despondent. I couldn't call my family. I couldn't call my friends in New York because, the cell service wasn't working. I couldn't believe life was going on while New York was being destroyed. And it was a terrible moment. And I remember walking down going towards State Street, and 3 women passed me by. This is September 12th, and they looked at me and they said, I know you bombed us. And it really was like, oh goodness, people think I look like the people that flew the planes into the towers. And I went home like a coward and I shaved my beard because I thought that would help. And then I went to the museum by myself and I worked in the basement of Ruthven. So this is 2 floors down into the bowels, like the real bowels of Ruthven. And I found solace in working in the collections. It really helped me sort of get my mind straight again. And also my cohort and other people here in the department taught me to sort of get better, and we all together grew up, right? We had a lot of growing up to do. America changed, the department changed, I changed. So that was my second lesson from being an EEB. This is the picture that I always think of when I think of my time here. This was taken by David Bay, who is our much beloved staff photographer. This is me with the blobfish. Just one of the things I would just like go through the stacks, go through the collections, and look for stuff and learn about stuff. And it was a wonderful experience that I hope everyone gets to do, to just look at collections and love them. And I'm not the only one who loves blobfish. These are my 3 kids. when I went to LSU, I was like, I got to get a blobfish for the collection. We did. When I started my job at LSU, and I looked at the collections for the first time, I was like, how come I know where everything goes? It was like deja vu. And it's because the previous curator, the only other previous curator, Mike Fitzsimmons, of fishes, had organized it with Hub's numbers the same way University of Michigan's numbers were organized. So, you know, we still call it the University of Fishigan, because everybody at different museums throughout the country had this sort of connection back to EEB and Michigan somehow. So I learned, you know, to love collections, to tell the stories of these animals, and to regard them not just as data, but real important organisms that can tell us a lot about Earth history. Some of these quotes are real, some are not. But I think of this because I was going through I was like, what am I going to talk about? And I was like, let me look through all my Michigan stuff. I'm like, oh my God, I have so much Michigan stuff. And for the grad students, your files are digital now, but I still have the reprints that I studied, the things that I was working on, I still refer to them. But I kept a lot more than that. I have like random class notebooks, the flyer from my defense, my business card. I printed out the emails, my Hotmail account, like where I applied to work with Bill Fink and Jerry Smith and John Sparks, who I'd later work with. I don't know why I have this stuff, but they were important to me. They were transformative, like, oh my God, these people wrote me back. I have the Graduate Studies Bulletin, and I'm going to show pictures of some of the faculty. This is before EEB formed. This is before people had a faculty website. They had a handbook that they give to incoming students, or even prospective students, which I was at the time. And I thought Bill Fink looked like this. I think this is a picture from Harvard. And he looks like he's 40. And I met him, and he was an old man. I saw him yesterday, so I told him I was going to say this. But he had gray hair, and he was wearing all white. He just looked like an angel. And I looked at my faculty ID card now, and I was like, oh, I also don't look like my picture. So it's not, you know, just him. But he was like the youngest member in the fish division. And, you know, Jerry had hired Bill, but there were the big three, as we call them, Bailey, Miller, and Gosline. These are the people that made this, you know, especially the fish division at Michigan, at the UMMZ, world famous. And they taught me a lot as well. Bob Miller, who passed away the first year I was there. Gosline passed away in my third year, and Bailey fell after seeing my defense and really never came back. And so I didn't do anything on purpose, but yeah, they passed. Bob Miller had left all these wonderful notes and had paid artists to illustrate a lot of the bones that we had in the collections, and these were used in my dissertation. If I were to do it again, he'd be a co-author on these things. But he played an important role in sort of getting me to think about bones in a different way, as did Jerry Smith. So Jerry passed away 4 years ago. Sadly, it broke my heart to not see him before that. But he taught me to not just look at living things, but to look at fossils together. And he had me borrow this oldest fossil from Haiti, which is a cichlid. And I was studying these Antillean cichlids, and they all have on the inside of their teeth a cusp, a lingual cusp. And I borrowed the Smithsonian fossil. It was 15 million years old. I was so nervous, and I was scraping away the matrix with this blowing tool that can scrape away the rock, and I saw a tooth, and I started scraping away at it, and it blew air, and it flew across the room, and it's 0.5 millimeters big, and I was like, I have to find this. I spent 2 hours through the dust and the muck on the floor of the lab looking for it. I found it, and it had a cusp. And I was so happy. It takes up a quarter of a page of a manuscript, even though it's but Jerry taught me a lot about, you know, working with both living and fossil forms. This is my committee. So Bill, Jerry, Jimmy DeFoyle and Dan Fisher. This is what Jim would look like in that handbook. He's still Irish, I think. And I couldn't wait to get my dissertation on the shelf with all these historic names of other people who would go on to be fish curators. I really wanted to be the first student who had only been in EEB, not in biology. And so, you know, I didn't rush, but I was like, oh, I'm going to be it. And then I got scooped by Mercedes Pascal student, who's a wunderkind, Katja Coel. She's still a wonderful scientist in person. But together, she and I drank the honey liqueur, which was, I think, is that still tradition here? What's it called? Krupnik. Oh my God, I thought I had forgotten about Krupnik. And we drank it together, and we were the first EEB students to graduate as only having been in that department. There were a lot of great people in that first cohort. I'm not even the only museum director. Heather Lerner is now at UMass Amherst as a director. There's Andre Pulaha, who's working with George at, I thought it was AstraZeneca, but he reminded me he's moved on. Ann Fowler, who still lives in the city and is an important member of the community, when I reached out to her about this talk, she's like, well, you know, she called herself a dropout, and she reminded us we had a lot of attrition in that first cohort, and I didn't like the idea of people calling themselves a dropout because she's had success. And so these are my, you know, the 7 students I've had that have finished or that were in my lab. Not all of them finished. Many of them have gone on to, you know, prestigious jobs at museums themselves. One, AJ Turner, he spent 3 years in my lab, but he didn't complete his PhD, but he got a job at Rapid Genomics and is happy. And, you know, he wouldn't have gotten that job without 3 years in grad school. And that's success too, right? So there's different ways to succeed. You learn so much, you know, about opportunities as a grad student. You know, finishing your degree isn't the only option. You should do it. It's great, but if you go on and you do something else, you don't have to think of yourself as a dropout like Ann said. She's a wonderful person, and I'm going to tell her I said that. I love that Hernan Lopez Fernandez, his student and my student, have collaborated together. This is Pat, who just introduced me, and my student Shayla. Pat's from DR. Shayla's from Cuba. You know, more advice, you know, if you want your H-index to go up, make students that cite you and do the same thing you did. But, you know, it's wonderful to see this sort of second generation of folks who are doing wonderful things and still have this Michigan connection. Bill Fink taught me all I need to know about fishes. I didn't really know anything about it, about them. I didn't know about anatomy. I have all these sketchbooks where he sent me down into the stacks to look at bones. I don't know how I would teach ichthyology or evolution if I had not TA'd coordinated anatomy and animal diversity. The classes that you TA grad students, or is it GSA, I don't remember what you're called anymore, you know, you will go on to teach courses that are similar. So when I teach ichthyology, it's basically chordate without the cat. Evolution, you know, I'm using all the things I learned as a graduate assistant to teach those courses now. When I was working on that fossil, I was also looking at living species from Haiti, and there's a species that was described, Nandopsis vombergi, that was supposed to exist somewhere in Haiti. There was one specimen from Hamburg, Germany, which I borrowed. I couldn't believe I can get a fish from Germany that was really from Haiti sent to me. And a couple of years later, or a couple of years ago, I actually visited that specimen again with my daughter, who illustrated it for me in different ways. And but I learned to love fishes and anatomy there, but also field work. And that same specimen this is before ChatGPT, David Bay, again, created this for a seminar I was going to give, my EEB lunch seminar. And David was wonderful. If we'd go out for lunch like we did many times, he'd come up with a new name on the spot for himself. He'd be like, just make up a name. And then when they asked me, you know, what my name is, I'd say David Bay. And I did that yesterday with Hernan when we got tacos. He was a wonderful person who passed away, sadly, a few years after I left. But I learned to do field work. You can see from this picture, I didn't know what I was doing. This is my first trip. I didn't know how to hold a seine. I was sent with no money and no help. But we figured out, and that's good when you want to prove something doesn't exist, which is what we were doing. But I'd go on to do more field work while I was a student here. I went to Belize and southern Mexico, to the Yucatan with some great colleagues, Juan Schmitter-Soto and Ron Roldfield, who was an EEB student. They're going into a cenote and they're diving. And I was like so jealous that they're going into the depths and the darkness of a cave. And I didn't know how to swim. I learned how to swim here when Michael Phelps was here. So that was a great motivation. He was already a decorated Olympian, but I was like, I just don't want to die. And but I was so jealous. I learned to dive, and I love now cave work. So I'm a pretty experienced caver. I still fit in these things, I think. But I learned to love field work from being an EEB student. So I've now done a bunch of it, and having field-based research museum folks and other EEB folks really inspired me to do this. Among the many things I kept seeing Deborah Goldberg's name. And it made me realize how much of an influence she had, not just as chair. She was chair most of the time I was here, but just as a mentor. She wasn't on my committee, but I realized she was teaching us to be, that you don't have to be a jerk to be an effective leader, which I thought, seemed like the practice at the time. She was caring, and she cared about us, and she made us feel welcome, and she was wonderful. So this is this EEB chair that she showed us. This is a picture of you in bio a little bit later than the first picture you showed. She nominated me for awards, and she said nice things. She said I was a born administrator. And I was like, is this good or bad? I should go back to doing more research so she sees me. My favorite thing that Deborah called us, so we were the first cohort, we were nicknamed the implied cohort. Do you remember why? So in our acceptance letter, it seemed, to reading it, it read like we were going to get summer support every year that we were grad students. You didn't write this letter, but I think when they read it, they saw we were only going to get summer support for 1 year. We read it and we thought, no, we're getting it every year. So she didn't have to make it right. She could have said it's a mistake, but she gave us summer support for the 5 years, and it was, so we were the implied cohort. But we served on lots of committees together. This is my favorite Sopranos episode, by the way. And it taught me, doing service and the research is important. You're not just one-dimensional, and you learn things from being on these committees. I've worked for my scientific societies and other groups to improve how we teach climate change or evolution around the country. I worked as an NSF program director, doing those things, and Deborah really did teach me, you can do both things, and you can do them well, and you'll benefit from it. My favorite was she put me on the diversity committee when we weren't a very diverse department, as she mentioned, and they got the advanced grant, and it transformed EEB. And I really got to see the meaning and impact of, people who cared about that stuff, how much that would change not just the dynamics and the social aspects, but just the science that was being done in EEB. She sent me on to a conference, an HBCU conference, historically black and colleges and universities conferences, and it was in Louisiana. And it was the first time I was there. And I remember going to the conference, and there was some breakout on a bayou, and this guy says, "Ooh, an alligator garfish has the head of an alligator and a body of a fish." I was like, this is where I'm going to study fishes and teach fishes from now on. But Deborah sent me to that first conference. She was talking about the raving cladus, and boy, was I one. I bought it all the way through, but Dave Mendel was here too, and he taught me about maximum likelihood. This is Arnold Kluge, who's like the most raving of raving cladus. But I learned, I'm still not a methods agnostic. I learned, and I still ask students, why are you using this program instead of this? Why are we doing this instead of this? We talk about epistemology, we talk about Aristotle, we talk about Plato, And I learned, it's not just the application of it to phylogenetics, but to other programs. And I love going through my notebooks and seeing how much I was learning. When Lacey Knowles and Brian Carsons came, I learned even more, and I sort of wiped the blood from my lip as a raging Cladus and opened my eyes to how much more there is. And I learned that here. And I still have my lab notebooks, and we're doing Sanger sequencing of 2 or 3 genes at a time. And we were learning about saturation and aligning things that are trivial now. But it gives me a context to understand, now that we have whole genomes, what to do with all this data and the questions we should be asking. And I always say now the jungle of the genome, there's a jungle in the genome, it's natural history too. There's fossil genes that have went extinct from an ancestor that we can study, like paleontology. We can look at form and function. And I want to just give a little interlude to how I applied those first 8 lessons to the research I'm doing now. Because for the grad students, you will always be doing your dissertation. It's just a continuation of it in different forms. And we're working now on this convergence between a living species, this cave species. We spent 4 hours in a cave in Thailand going after Cryptotara demiacola. They're blind, they're depigmented, they climb waterfalls in the dark, and so we had to climb waterfalls in the dark. And it has weird anatomy that you can see when you see them walk. So this is a salamander, not a fish, right? It is a fish, but it's walking like a salamander. My colleague, Brooke Fleming, we took this after we brought it outside of the cave. So like something's going on in the anatomy because fishes can't do this. And we were making x-rays when I was a grad student. Now we're doing CT scans, we're doing MRIs. And you can see that this fish has a hip. Fish don't have hips. This one does. It has a connection between the vertebral column and the pelvic girdle that allows it to do this back and forth salamander walk. And fishes haven't had that since the first fishes that came onto land. So the water to land transition in things like Tiktaalik, it also has this hip. And so, we can look at that. We were looking at that convergence using the tools, advanced cutting edge tools from what I learned at Michigan. I also learned geometric morphometrics I saw Miriam Zeldich here. There she is with Bill Fink. And we were turning 3-dimensional objects into 2-dimensional things. And now we can add time and space, which is incredible. So we were looking at the stride and gait of these things based on their anatomy. I mean, things I couldn't have dreamed about doing when I was a grad student. We're doing these advanced morphometrics, we're doing CT scanning, we're doing whole genomes, and there's still a jungle in there, right? So now we can look at what's missing and what's been added. We can see that some things don't make sense. These things have no eyes, but functional opsin genes. Some things do make sense. They don't have color genes because they're blind and live in a cave. They don't have any circadian rhythm. These are things that you can read from doing genomics, which I didn't think was something that we could even think possible when we were grad students sequencing, a handful of genes. And so, I don't know what the future holds for the grad students now, but it's going to move fast. Lessons 9 and 10 are really things I learned after I left here. So I wasn't a very healthy student when I was here. I didn't mind my health. And some of you know I've been going through a lot of health issues these last 2 years, and I'm telling people to get their mammograms and colonoscopies, and I think that stuff matters as much as the science, right? I've been dealing with the cancer diagnosis that you just don't know when that stuff will happen, or when you'll lose some of your colleagues like Jerry or David Bay, people that I didn't get a chance to say goodbye to. But keep your old colleagues close too. You just don't know when they may leave you. This is me and basically Matt Friedman's whole lab in New Zealand. I've saw friends from our department in Singapore. I've come back here many times. This is the last photo I took of my updated committee members. I don't know if you still have a holiday party, the Macedon Santa hat. I did that. At least the first version. I don't know if it's the same one that you use, but I was like, the only thing I've ever sewn is a hat for the Macedon. But I'll conclude by just saying, I learned many lessons as being part of that first EEB cohort, and we had a lot of growing up to do, right? From September 2001 to now. The growth that I've seen in this department, I mean, just even imagining this building or these buildings wasn't something I thought was possible, and it's amazing to see. There are many versions of success, as I was talking about, for the grad students. You can have many versions of you, but stick to your roots. Remember the memories that you have and the mementos too. So in 2051, when we do the 50th anniversary of this department, and I'm long dead, you'll have your stuff to talk about as well. And I'll just leave by being sort of cryptic, like The Sopranos ended. So I'll leave there. But thank you for your time. Yeah.
Can you guys hear me? Cool. Hi all, thank you all so much for being here. My name is Matt Hack. I'm a PhD student in Ben Winger's lab. I study the biogeography and distributional ecology of seasonal migration in birds. It's my absolute pleasure to introduce Richard Prum. Rick earned his PhD from the University of Michigan in 1989 and is now the William Robertson Co-Professor of Ecology and Evolutionary Biology at Yale University. He's also the head curator of vertebrate zoology at the Peabody Museum of Natural History. I've been instructed to keep it quick, so I'll keep it to the highlight reel of Rick's many accomplishments, which include a Fulbright Scholarship, a Guggenheim Fellowship, a MacArthur Genius Grant Fellowship, the Lewis Thomas Prize. In addition, his book, The Evolution of Beauty, was a New York Times bestseller and earned him a Pulitzer Prize nomination. And on a personal note, Rick was my undergraduate advisor, and he has opened so many doors for me, given me so many opportunities, and opened my eyes to the many fascinating wonders of the worlds of evolution and birds. And I know that you'll all be as inspired by his talk today as I've been inspired by Rick for many years. So please join me in welcoming Richard Prum.
Just for the record, Matt showed up that way. He's responsible for all his great stuff. A pleasure to be here and to be with you all. And what a fun exercise to talk for 15 minutes on, this place, my history here, etc. Many difficult things. First, just finding slides, pictures. Nobody had a camera. I have this weird history of my time here. A lot of pictures of birds, handheld, and then really dark pictures of tropical forests, and not a lot else. But I did manage to go through the archives and find some stuff. This is where I start. This is, yes, a very embarrassing picture. In some ways, I showed up this way too. The key feature here are the glasses. I grew up mostly in southern Vermont. I got my first pair of glasses at the age of 10. I started bird watching in mere months. I started a life list, which as a 10-year-old means you know what you're going to be doing for the rest of your life. And it turned out to be true, right? And so luckily there's places for people like me at institutions like this. This was already at this age a Michigan story. One of my mentors, first birding mentor, was Tom Will, who was at that point in the late 70s a hippie Yaley hanging out in southern Vermont writing pretentious natural history essays for the local newspaper. I wrote to him, and this was the first adult I ever knew that was a bird watcher, and we actually shared some time here in the graduate program. I became really fascinated. Well, I knew I was into birds when I was an undergrad at Harvard. I became interested in phylogenetics. The only faculty members that were doing that at that time were Bill Fink, before he came here, and Peter Stevens. But the grad students were amazing. There was a systematic and biogeography discussion group with Brent Mishler and Michael Donahue and Wayne Madison were all grad students. And so I got very interested in that and inspired by Bill Fink. I did my undergraduate honors project on phylogeny and vicarious biogeography in toucans, and actually toucan systematics in general. So I knew I was into phylogenetics. I got my first keys to a museum but I also wanted to do field work. So I became fascinated by mannequins, a sexually dimorphic group of neotropic frugivores, lots of sexual selection, lots of male display, females doing all the parental care, et cetera. And I became interested in their courtship display. So after graduating from Harvard in '82, some reason I felt I could do this. I just went off to Suriname and started watching mannequins. And it was natural history, binoculars, notebooks, no fancy cameras, no nets, no anything. But I was starting to study the courtship displays of mannequins. I found one in the first week, white-fronted mannequin, Lepidopteryx serena, which had an unknown display. So I was watching him and for about 2 weeks and I knew I had a paper under my belt. It was a very exciting time. But my real quarry was this guy, white-throated mannequin. And so on one day, after studying Lepidopteryx for 2 weeks, I set it up down the Irene Eval Trail, which was a well-known locality for this bird, the crimson topaz. And as I passed that log in the trail, I heard a whoosh, something flew right by my head. And it turned out to be this guy. And in moments, he flew down to that log and started displaying. And in 5 minutes, I was the world expert on Cohort guturalis, which was all it took. But in fact, I spent all fall there, all months, and it was a fantastic experience. So I came back from Suriname with 2 manuscripts in hand. And ultimately came out as in this paper, which I published here at Michigan. Ultimately, the bird flew in, made a mechanical pop, dropped to the log, jumped up from the log, flipped around in midair, and landed back where he was coming from in bill-pointing posture with that white throat and everything. It's like nirvana. It was a pretty amazing experience. After that, I spent a summer at Coachacashu as a field assistant for Nina Pierpont. But in the fall of '84, I ended up here at the University of Michigan working in that building and that office right there. I think I think left or right. Anyway, it was right there. And there are no, as I said, nobody had pictures. This is the only proof I have that I've ever been in Ann Arbor. That actually says Ann Arbor, well, not Ann Arbor Hardware. I think the reason I was there is because the Photoshop was there, and the last thing in the camera had a couple shots, so you just stop to put before you gave the photos in to be done anyway. So that was a fantastic intellectual environment. We've heard it described a lot here. One of the fantastic elements in the museum at that point, the 2 curators in birds, 2 in mammals, 2 in herps. There was the bird mammal herp lunch every week where you gave seminars. And you were expected to sort of show up and give a seminar. And it was really informal. These are my slides. I actually found these slides and took a shot of them this week. So here you see CCC Pop-tickee-yeah, jumping, right, et cetera, et cetera. So that was a part of the environment. The other critical part was the Systematics and Biogeography discussion group. It was a fantastic weekly discussion of the philosophy and application of phylogenetics. Again, I don't have a lot of pictures in Ann Arbor, so here we go, pictures in the field. One of the marvelous things was that there was no summer salary, and I say that was marvelous because you had to figure out how to deal with that. Well, the way I dealt with it is I got funding to go to South America for the summer, and then sublet my apartment, and there you go. You got an affordable, great way to define. Did we complain? No, we thought it was great. So I went and did field work in Ecuador in the summer of '85 and '86, combining specimen collecting and ethology. I dreamed of doing phylogenetic ethology, bringing phylogenetics to the bird watching that was my tradition. My first quarry was golden-winged mannequin. Why? Well, it lived in the Andes, and I was dying to birdwatch the Andes. Now that's a great motivation for a dissertation, but you certainly don't tell your committee that. But I did get funding, and it took a while. I was working in Andean Ecuador with my wife, partner at that time, Ann Johnson, now wife, partner for over 40 years, and actually she's now an Emmy-winning natural history documentary. He's produced 15 Hours for PBS and has won 2 Emmys, and each time she went up and beat David Attenborough and BBC, right? He lost a lot more times, but there you go. We end up finding the bird in the tiny town of El Placer, literally a railroad stop That pig ate all of our food on the second day. And here we are. Actually, shout out to Bivouac, who gave me 50% off all camping goods that I bought for this field trip with money from the American Ornithologists Union. It's a gorgeous bird. To my stunning surprise, it turned out to display on mossy fallen logs. And you can't explain stuff like this, but it turned out to actually share deep or very specific behavioral homologs. It flew down to the log, jumped up, and landed back on the log, but instead of having its bill up, in the upper right, it had its tail up. So literally in a moment, I realized my dreams of phylogenetic ethology would work. My dissertation was possible, right? And it was quite an experience. Published it here in '87 in the Wilson Bulletin with Ann and I. And I published this phylogeny, which was based entirely on shared derived behavioral hypotheses of homology, right? This is in the Wilson Bulletin, now even less well-known ornithology journal. This is the first phylogeny ever in the history of the Wilson Bulletin, and it's entirely based on behavior. And in fact, this topology, with the addition of Antilophia as the sister group to Chirosifia, is still accurate. It's been supported by every molecular phylogenetic technique, et cetera. So I want to introduce now this other aspect, which is that my interest in phylogenetics was not universally shared. There was this time at which the majority of people in evolutionary biology thought that phylogenetics was, as I was told as an undergrad by certain leaders of evolutionary biology, not going to work and not interesting. What would you actually learn if you had the tree? So in the fall of '84, I was taking Peter Grant's behavioral ecology class. And the term paper was either one from a menu of 3 or 4 proposals, or you go to office hours and ask. And so I went to his office hours, and I said, I want to write a paper about phylogeny and adaptation. And he said, no. And I said, why? And he said, there's nothing to write about. My jaw hung open, and I left, and I did a one-draft wonder on ecology of parthenogenesis and got a high A. But that was 5 years before Baum and Larson, 7 years before Harvey and Pagel, or Brooks and McLennan, right? So there was really a challenge, which is that the architects of evolutionary biology did not think that phylogenetics belonged in the discipline. So when Felsenstein published his fantastic paper in '85, we clipped out this quote and hung it on our door in Ruthven, right? It was like, I won't read the whole thing, but it's like, nevertheless, to cope with the effects of phylogeny, phylogenies are fundamental compared to, comparative biologists may understandably feel frustrated upon being told that they need to know their phylogenies are grouped into detail, when this is not something they had much interest in knowing. There was also another intellectual challenge, which is that I was getting, because of my interest in courtship display, I was getting very interested in sexual selection. Yeah, and in this period, but I was a Fisher fan, and as we can see for this quote, the pale eccentric umbilically or pale eccentric mathematical types became Fisherians. Field biologists, good naturalists, bearded, besweatered, and booted, gradually found themselves to be good geners, right? This is from Matt Ridley. Well, there I am. The boots are not in the photograph, but I am certainly bearded and besweatered as I record bird songs, right? So there were all kinds of intellectual challenges. I continue my interest in phylogenetics and in biogeography, vicarious biogeography here. But ultimately, I needed a phylogeny, and so I became interested in syringeal anatomy, especially through the work with Bud Lane at the American Museum. So I used the phylogenetic tree and continued to study behaviors, and in my dissertation, I did lots of different tests of hypotheses of homology of behavior. So behavioral phylogeny exclusively, a combined tree, and a syringeal syringeal phylogeny with the behavior superimposed, and discuss that, right? So this was really about fundamental ideas in tree thinking and ultimately in sexual selection. This is my PhD dissertation defense committee, and I just found this slide 2 days ago. So that's Bill Fink, Mary McKittrick, me, Bob Payne, and Herb Wagner. Notice that Mary is holding a cigar. As a pair of cigars, I was her first student to graduate, and on it said, "It's a boy," you know. So, and the way in which this place changes your life, you know, I got that tie at Van Boven's. I still have that tie. Is Van Boven still around? Yes, indeed. Fantastic, life-changing place. Del Rio, sorry, not here. So concluding now, most biologists did not embrace phylogenetics or tree thinking in the 80s or earlier. As a museum university, the U of M and UMMZ played a vital role in support and development of phylogenetic methods and tree thinking during this time and later. And then subsequently, phylogenetics and tree thinking had a decades-rich intellectual history prior to the development of genetic data. And I think these are all sorts of important things in the history of this department and in the museum as well. Well, thank you very much. Oh yeah, lots of people to thank. My committee, Dr. Shore, also Steve Goodman and Michael Nachman, my good friends in grad school, kept each other sane and insane over a number of years. And then thank you to the department for the invitation.
All right, thanks, Rick. Next up is Thais Vasconcelos, and I was on a search committee right when I got here. There you are. And I remember the moment of seeing Thais's application come in and thinking what a rock star this person is and what a career they're going to have. We have to figure out how to get this person here, and we did. And so, Thais, it is amazing to have you as a colleague and as a director of the herbarium. So take it away.
Thank you for the nice words. How do I put this in full screen? Because
Yours is a PDF?
Yeah, mine is a PDF. Sorry.
Is there a way to... Yes. Does anyone know how to make this into a giant slide?
It usually shows full screen view if you can find it.
I don't see it.
I can also just...
Go left. Do that?
Yeah, okay.
Great.
I can present like this, I think.
Okay.
Okay, let me see. Okay, cool. Well, thank you for the nice words, Nate. And it's great to be here in the middle of this lineup of very intimidating speakers. Okay, so I'm gonna talk a little bit about our amazing herbarium in this talk entitled "The Stories That 1.75 million specimens can tell." So for those who are not familiar with our herbarium, it looks like this in the outside. It's a big room full of cabinets. But once you open each of these cabinets, what you find is hundreds of thousands of specimens of dried plants and fungi. There are about 3,000 herbaria worldwide like this one. But ours is on the large size, is ranked 40th globally with 1.75 million specimens. So I can say, not so biased, that this is one of our gems of the department. And, you know, sometimes we make a comparison with herbaria and museums. We compare them to libraries of biodiversity. But this comparison, it's not super fair because unlike libraries, each of these specimens is unique, so we cannot replace them if we lose them. Each of them are collected in the field by botanists and mycologists. They are dried and processed, like you see here. Shout out to our plant mounter, Nicole. And so they are not static. They are always growing through new collections. We grow through new collections that we make in the field or through donations. And because of the way that we process them, they are built to last forever. So we dry them, we keep them in climatically controlled conditions. So we have these amazing resources that we can use forever to answer all sorts of questions. So what do we do with 1.75 million specimens like this? I often say that each of these specimens tell a story, and that's because each of these specimens come with a surprising amount of information and data. They are not just the dried specimens that we see, the biological material, but they also come with a lot of metadata in their labels. So the labels often include the species, well, they include the species name, often the family, but not only that, but also where they were collected, details about the habitat where they were collected, when they were collected and who collected them. And combining all this information in 1.75 million of these data points, we have this amazing resource that we can use to answer all sorts of questions in EEB. And in a herbarium like ours, we have then almost 200 years of data from all continents and almost all countries in the world. So 200 years, so our herbarium, so a little bit of history of our herbarium. Our herbarium really started to grow when Michigan became a state, almost 200 years ago in 1837. And that's because the first governor, Stephen T. Mason, among other things, he advocated not only for making Ann Arbor as the University of Michigan's permanent site, but also for the first geological survey of the state of Michigan, which was an initiative to get to know what type of biodiversity we have here. And then by law, all of the specimens collected during those first geological surveys had to be vouchered in an institution of higher education in the state of Michigan. So that's when we started to grow as a collection, because before that, we had some collections made in the state, obviously, but they were often sent to institutions in the East Coast. And then after that, we started to grow our collections. The herbarium became official administrative unit within the university a bit later than that. But we can trace some of our oldest collections to 1837. And these first specimens, they are really a window to the past, which is, you see that that's a common theme when you work with collections. So that's because if we can look at them, we have a bunch of these 1837 collections from the first geological survey. And if we compare the information that we have from them with the information that we know today, we can understand how, for example, landscape changes. So based just on information that we have from the labels of those specimens, we know, for example, that oak savannas and tallgrass prairies were much more widespread than they are today. We can also understand extirpation. So there are a lot of these species that were collected then in the state of Michigan that don't occur in the state anymore. At least 13 of those collected in that first geological survey. And we can also understand shifts in community composition. So one that I find really interesting is that back then in 1837, only 5% of the flora was of introduced species, and today we know that the flora of Michigan is composed of 39% of introduced species. So that's pretty cool. If you want to know more about that, our curator emeritus Tony Reznicek just published a really cool book about these first surveys and how the flora of Michigan looked like in 1840. And if you want to know how to purchase one of these, talk to me, to any of the herbarium folks here today. So documenting the local and global biodiversity, like in the fashion of the first geological surveys, has been truly one of our bread and butters in the herbarium for most of our existence. It's been one of our strengths. And there are several famous examples. I think I could, putting this talk together, I thought like, well, I could talk like a whole day about the important collectors and systematists and taxonomists that contributed to collecting our collections and, you know, made a big chunk of the percentage of our collections before, like in this last 200 years. So I'm just going to mention some famous examples linked to our herbarium. Elzada Clover is someone who became famous, especially after the publication of this book a few years ago, Brave the Wild River by Melissa Sevigny. And she was the first woman to document the flora of the Grand Canyon by rafting down the Colorado River. Also the first woman to become a full professor in our botany department at a time when only 15% of the PhDs were awarded to women. And that was 100 years after the first geological surveys in 1937. And we still have a lot of the specimens collected by them in that trip in our herbarium. Other famous examples include Alexander Smith, who was a very prolific collector of fungi, collected over 100,000 fungi collections, also prepared these gorgeous photographs of mushrooms in the field that were really unique and novel at the time. Published also several, like dozens of monographs of different genera. And Ed Voss, many of you know Michigan Flora. He was the person who led the first volumes of the Michigan Flora, which is still one of our assets, and it's widely used to inform conservation land management in the state to this day. It's becoming an app soon. And yeah, just to give a few examples of famous people, important people that linked to our collections that worked on documenting biodiversity. And the task of documenting biodiversity continues to this day. And that's because after 200 years, we are still finding out what we have in our collections. You would imagine that at this point, we know everything that we have, but as we keep curating the collections, new things are still showing up. So we're still describing new species to science to this day, even based on collections that were made like 100 years ago, 50 years ago, and that were collected for a completely different purpose. These are two examples from our EEB PhD students, Paulo in my lab and Alden Durkis, who was in the Tim James lab, who contributed with describing new species of plants and fungi based on our specimens, just to give a few examples. Okay, so I talked a little bit about the history of these 200 years, but now moving towards the present, I think one of our strengths right now in this past 25 years is that the herbarium became a important source of genetic data. And that's a kind of a more recent development, because now we can actually sequence DNA from these dried materials, sometimes that were collected 100 years ago. And that has become one of the our main contributions to the EEB community. And that's because there are still many gaps to be filled in the plant and fungal tree of life, so I have to speed up a little bit. And we get requests from all over the world. In this past 25 years, we also entered the digital era. So that's when we started to database everything, image everything, and make everything publicly available online. It began as a process of preservation, but ended up transforming our herbarium and other herbarium from cabinets of specimens into a global digital infrastructure for ecology and evolutionary biology research. And thanks to that, we can now combine data from different herbaria to expand analysis of global change. For example, we can see how the species expand their ranges through time by combining data from different herbaria. This is an example of something that we are working in my lab with quantifying range expansion of invasive species in the Great Lakes region. Thanks also to digitization, I think people started to be more aware of the collections that we have, and then that has helped us to diversify the ways that we contribute to EEB research. So we have several cool examples here in the department today of folks using our collections, our physical collections, to understand plant arthropod interactions and plant defenses in the Weber lab. These folks are here today, so you can talk to them more about that later. And finally, I think in the last 5 years, because of our massive digitization, we have entered the era of using AI and computer vision to automate label extraction and trait data from these images. A person that has been doing a lot of that here in the department is Will Weaver, who's a PhD student in the Smith lab. He might be here today too. And that has dramatically increased the quantity and quality of trait data sets and occurrence data that we can get from these digitized specimens. With some recent examples, for example, in this paper that we collaborated last year, where you can see that it dramatically increases how we can collect data, especially from tropical regions where we have collections from, but it's hard to extract this data manually. Okay, so just to finish, I think the theme of working with collections is really a collaboration through time. Today we are using collections that were collected with a completely different purpose, for a completely different purpose in the first geological surveys to answer questions of global change, evolution, ecology. And that also means that the collections that we make today are going to be used for a completely different purpose in the future as technology changes. So I think that's like the message that I wanted to leave you guys with. And with that, just a special thanks to everyone that contributed to make the University of Michigan Herbarium the amazing asset that it is today. Everyone listed here worked in the collections in the last 25 years. As a staff or curator, and yeah, and then thank you all for listening.
So while Alison is if you are one of the speakers, your lunch is in room 1010, which is just out this door and to the right. If you're not one of the speakers, you have lunch on your own. If you walk out of this room, there's a pamphlet that lists all of the great restaurants within walking distance of the building. I can highly recommend Joe's Pizza if you like a slice of pizza. But now I'll introduce Alison Davis Rabosky of the University Museum of Zoology, professor and curator. He is an award-winning teacher and an amazing scientist. In about 5 seconds. Ready, go.
Thanks so much, Nate. Yeah, it's been a phenomenal first session of talks, and it's very hard to follow all of them. But I am here to talk about the Museum of Zoology, a little bit of its past, and hopefully where we think it's headed in the future. I wanted to start, though, with my title slide, and the title that I chose very intentionally for this talk, Preserve for Good. This is an amazing snake that we caught as part of our UMMZ herpetology expeditions that went throughout Latin America from 2016 to 2019. But do you see all the little Fruit Loops? Yeah, each of those is a specimen. So there's about 500 preserved snakes that are in this photo. And I was wondering if I can get a volunteer to tell me how many hours do you think it takes us to find one snake in the tropical forest? I wish they were like mannequins. One day, if I can just walk into the forest 3 steps and it displays on a log in front of me, I will feel very excited. Does anyone want to give a guess about how many hours for one? We got 40. 40 is pretty close. We had a 5 over here. Yeah, it's about 30. So for 500 snakes, that's about 15,000 hours of field work, of person hours, in order to make a handful of specimens, right, for a collection like ours. So it makes us think very intentionally about what can we do to make these specimens maximally useful to the greatest number of people for the longest period of time. How can we best preserve them for good? But luckily, you know, the UMMZ has been around for a long time. We have approximately the same history as the herbarium. So our oldest specimens are back from the early 1800s. And how we describe ourselves as a community, I think, has similarly evolved through time. And so we've gone through many iterations of trying to say what we are. So some of you were involved in like the earlier versions of this that already existed when I arrived in 2012. But we're increasingly a global community. It's not just about the people that are in this room, but about connections that we have to collections all over the world. But we're sort of united by a set of values, right? We want to understand the diversity of life on Earth, and we want to use those collections to advance societal well-being. And we're also here to help everybody else who has those same values as we do. And I think that sums up what our community is. Now, we sort of stand on 4 pillars, right? We want our collections to anchor the most innovative research that can truly advance the field of ecology, evolution, and biodiversity science. We want the UMMZ to be the most compelling place to come and work and learn, and to be on the very forefront of the biggest ideas in biodiversity science, which I think it sounds like from the talks today, the UMMZ has been doing for a very, very long time. We want to share those collections to the people outside of academia. We would like to help people in the public, policymakers, and other global communities appreciate the specimens the way that we do, and we want to preserve them in perpetuity, just like Thais said, right? We need to care for them with integrity, and they have to be continually enriched to serve humanity for generations to come. But when people ask me why is the UMMZ so amazing, I immediately go to our 2 greatest strengths in this order. The first is our people. We would not be the community that we are without the people who are the engine to drive all of those amazing things. I actually really love that Prosanta showed what we used to look like. I too have been in the second sub-basement of Ruthven working in the collections, but we're no longer in Ruthven. We're in a giant new facility, about 35,000 square feet of collection space, and these are where all these people work every day in something much greater, in the service of something much greater than themselves. And unfortunately, it was really hard to pick pictures because it's impossible to get the entire community on a page because we're enormous. So about 2/3 of our community is students. We are powered by undergraduates and graduates, grad students, who make up more than 200 people, just who have like card access to the actual collection space, right? This is not just visiting scientists. These are people who are actually there long-term doing work in the collections. And this is in addition to hosting hundreds of short-term visiting researchers and thousands of visiting students, including the fact that the biggest change I've seen since getting here is we bring every single introductory biology student into the collections every semester, about more than 1,000. To get them through, it takes a whole week, and it's an incredibly huge effort, but it's one of the pipelines that gets a lot of people into the collections early, right? These are first-year students finding out about the collections very early in their career. But our second major strength is the collections themselves. So we have more than 15 million specimens, about 5 million wet prep specimens, about 10 million dry prep specimens, which is a huge amount. We are one of the largest university-affiliated zoological collections in the world. And I like to point out that our insect collection alone is bigger than our Museum of Paleontology and Museum of Anthropological Archaeology combined. One division in the Museum of Zoology is that huge. And of course, we're still growing. In the last 3 years, we have accessioned about 200,000 new specimens. And the sort of stats I like the best is that those came from more than 40 countries and were the result of collection efforts by more than 1,000 different people to help you understand just how big our global community is. But of course, we all recognize that those collections were here long before us. They will be here long after us. We stand on the shoulders of everybody else in our work today. This is one of my very favorite pictures from the history of the UMMZ, where you have a bunch of herpetologists and entomologists who are out there collecting mammals in Schoolcraft County, Michigan, in the early 19-teens. And, you know, really standing here with all of the rest of you really sort of brings home to me how much you are all part of our tangled bank. Like, we wouldn't be here today without all of the rest of you and all the contributions that you've given to our community. And I think that's one of the best things about working in a museum. Some of my very closest collaborators are people who died before I was born. And I think that explains how our work can touch many people. And so I'm really going to tell very quickly 3 little vignettes about ways that I think the museum collections change our field. I'm going to talk a little bit how we innovate with historical collections, even ones that were collected 100 or 200 years ago. How we illuminate the dark data that are still there in our museums as we, you know, tackle the absolutely huge problem of trying to understand the biodiversity on this earth today, and how the lessons we've learned sort of change the way we approach collecting for future generations of scientists. So what does innovation look like? Similarly to Thais, I get asked all the time, what do you do with millions of specimens? Especially those intro bio students love to ask, aren't you done collecting? And my answer is absolutely not. I'm not done collecting. I think there are 2 answers to the questions. First, of course, like Thais said, we do things that the original collectors never imagined could be possible with their specimens 100 or 200 years ago. And I think that's true about the specimens that I collect today. I really hope they're used in innovative ways I cannot imagine. But secondly, we do things that you could never do if they had just vouchered a photograph. Photographs do not tell you what any of these frogs eat. They don't sequence their genomes, and they don't tell you what parasites are inside all their little bodies, but you can see them if you look inside a specimen. And so, you know, I like to use the time capsule metaphor. Most people have buried or unburied a time capsule at some point in their lives, but this is a time capsule of a single point in space and time that cannot be duplicated once that time has passed. And they preserve entire communities. I also like to think of them as sort of like the only objective and freely available and externally validatable reference there's going to be for what something was like in a space and time before now. They can't be deleted off a server. We all have had an outage of our educational online system today, and that can't ever happen to a specimen. I also like them because they're really your time series, and in both ecology and evolutionary biology, time series is literally one of the fundamental units that allows us to answer the questions that we can answer. So if you care about how species distributions, species interactions, all the way through morphology and genomes might be changing across time and space, collections are your way to answer that question. But there's a big question about who can actually access this information. Certainly, as you have a tray of frogs in one place in one time, not everybody can actually make it to those frogs in order to study them. So what can we do to liberate them from their jars and to get them all over the world as quickly and easily as possible? This is one example of technology that allows us to do this. So this is what a normal specimen looks like inside of a jar. But if you use CT scanning technology, which Prosanta showed us a little bit of, you can immediately, after 15 minutes in a machine, be able to digitally dissect what this frog looks like on the inside. Now, of course, this is a 3D model, so it can be used for all sorts of things. You can rotate it in any way that you want. You can zoom in. Isolate different parts of the skeleton. You can have just the skull if that's what you need. You can use it to measure things that you could never measure with just a scalpel, at least not easily. You can look at what the density of the bone is in different parts of the skull. Those are the ears that show up nice and bright there. You can then sort of zoom it back out, and it's like it was never even touched. You can look at the thickness of the bone in a different part. You can take out just the inner ear if that's what you wanted, which is incredibly difficult to get out of a skull other than that. And it turns out that if you soak this specimen in iodine before you put it in the CT scanner, then you can also visualize all the soft tissue morphology as well. So with a touch of a button, you can say, ooh, that skin is really annoying, I will turn it off, and you can look at any part of the internal anatomy, muscles, bones, nervous system, circulatory system, organs, anything that you want, and anything your student wants to see, or to beam it to anywhere in the world as long as people have an internet connection and and a computer. And of course, when you're done, you destain the specimen if you put some iodine in there, and it goes right back in the jar to be used for anything you might want to do in the future. So, with a very quick 15-minute regular CT scan, you could have all these kinds of downstream uses, including 3D printing, of which we'll have some at the poster session tonight if you want to see some cool 3D prints. And by soaking it in iodine first, you can get all of this other kind of information, including a lot of natural history bycatch. Was it gravid? Did it have any parasites? All of those are immediately identifiable. And of course, we have done this not just once, but for thousands upon thousands of specimens. We were part of the OVERT grant that was trying to teleport these specimens to every place around the world. We scanned 13,000 specimens as a consortium, looking at one genus by every species. We did not scan everything. Part of the challenge is that there's lots of birds that are not prepped as wet prep specimens. Their skeletons are disarticulated, hard to get wet prep specimens. But those CT scans have been viewed more than 1 million times and downloaded across many, many countries. And for one NSF grant, getting 200 publications is pretty good stats. What about some dark data? This one comes to us from the UMMZ Insect Division, where there was an old uncatalogued collection from a bunch of daily trap lines that were run in Malaysia from 1979 to 1980 by a couple of Peace Corps volunteers. This is what one jar looked like, and we sorted them and photographed them to come up with amazing images of every single thing that was in one of these jars, and then, of course, get them all databased. These are just one group of beetles. They're now just sorted to sort of like rough morphospecies because you need a lot of expertise to be able to know if that's a species that has been described or not. But what's super cool about this is this was entirely the work of an undergrad. So Won Kim was one of our EEB undergrads who graduated just this spring. He's also coming in as one of our PhD students this fall. And he's an expert not on beetles, but on true bugs. And what he says at the end, we need more people. These are ones he can tell are something new, but for all those other things that are in the jars, we need more expertise, more people to become museum scientists. So how does that help us change the way we're collecting for the future? One of the things that happened right after the pandemic was that the UMMZ launched a zoonotic surveillance initiative with the Center for Infectious Disease Threats. Hi,
everyone. My name is Emma Dawson-Glass. I'm a PhD candidate co-advised by Dr. Nate Sanders and Dr. Marjorie Weber. I study all things plant-arthropod interactions and the contexts that mediate those interactions. And today it's my honor to introduce Dr. David Tillman. Dr. Tillman received his bachelor's in zoology in '71 and PhD in ecology in '76 from Michigan, and is now a Regents Professor and McKnight Presidential Chair in Ecology at the University of Minnesota, as well as a Distinguished Professor at UC Santa Barbara's Bren School for Environmental Science and Management. A world-renowned ecologist, a pioneer in the field of biodiversity research. Among many, many, many awards, Dr. Tillman is a member of the National Academy of Sciences and recently received the National Medal of Science. Please join me in welcoming Dr. Tillman.
Thank you, Emma. It's a pleasure to be here. And all of you people, it's very reminiscent of my early life here. Let me see here. I think that might do it. So a couple days before I started grad school, I made the best decision of my life, which was to marry Kathy. And we've been together now, almost 55 years or something like that. So that's what I looked like when I started grad school. And the other picture is what I looked like when I got my PhD. I was trying to do a John Vandermeer look, but I just never could achieve it. So I had to show those pictures. I hadn't seen them in a long time. I dug these up. That was the only picture at our wedding. We had a very simple wedding. We decided we were in love or to get married. And so it was in Kathy's backyard. Her mother took one picture, and that was it. Very different from today, I guess. So that's sort of, if you will, this really is the beginning of this time, as I say, long ago in an intellectual galaxy now far, far away from where we are. It really was a different time, but in some ways probably not that different from now. And for me, really the stars did align when I came to Michigan and decided to do my PhD. I joined this program which had this incredibly intellectually diverse faculty. And these were strong-willed, smart people, each of whom knew that their way of looking at the world was insightful and maybe even the best way to look at the world. And it didn't matter whether they're evolution, like Dick Alexander, ecology, John and Steve Hubble and others, or theory, those same people, physiology, genetics, all kinds of classes. I had to take all the kind of things around the zoology exam to be able to make your way to stay in the department. We're there to challenge you, to make you think broadly. And it really made me think when I do ecology. I never understood why people considered ecology different from evolution. Darwin sure didn't. To me, you can't do evolution, you can't do ecology without knowing evolution and vice versa. The two are really one thing. And we only divide them into two because we have little brains. Anyway, and then there's all the high standards I saw all around me. And frankly, science is just amazing fun. So that's sort of how I felt we started. And I really felt like when I came to Michigan, I just felt this incredible energy. I mean, this explosion of ideas. The hallway conversations that John mentioned in his talk, where you bump into somebody in the hallway and you say, here's what I'm thinking of, here's what I think is really important, and nobody's doing this. And then somebody else say, no, that's got to be wrong. This is the important thing. Just great, great discussions as we really were going from being more natural history oriented. And I love natural history, don't misunderstand me. But going beyond natural history to adding in things like experiments and theory in ways which I think have been very important, but sometimes to the detriment of actually having people who know real organisms and nature, which is a major detriment. So I've been influenced by many people. I'll start with Steve Hubble. Steve came in, and halfway through his first year, we had met each other, and he had hired me to be his research assistant, which meant I opened his mail, I answered his phone. When publishers were calling him asking where the review was, I had to tell him that it's on his desk and he'll do it soon. He never did it. He never would do those things soon. I started playing with algae and looking how they compete in his lab. He did lots of things for me. The one thing that probably nobody knows is how I applied to become a grad student at Michigan. It was Vietnam War time. I was sure I would be drafted and have to leave the country, so I didn't apply to any grad schools. And so when I found out I wasn't going to be drafted, I bumped into Steve walking across the diag. I said, Steve, I'm not going to be drafted. I don't know what I'm going to do next year. He said, I'm chair of the admissions committee. I just admitted you. Now go fill out the damn paperwork. So that's how well I plan my life. And that is the truth. I do things I love, and I don't really have major long-term plans in anything. Well, Hubble had an event going on shortly after we met. He was going to take a group of people down to Costa Rica to study stingless bees. I knew about his ideas because in his lab, he wrote his first NSF proposal. And Steve was a magician at writing really good proposals. And he would hand me his first draft proposal and say, read this, Dave, and tell me what you think. And then again, read this, tell me what you think, over and over again, draft after draft. I got to see how a master actually wrote a grant proposal. I learned so much from him doing that. I don't think I gave him any help at all, but I learned an immense amount seeing what he did and how he did it. So, and there we are on our way to Costa Rica, 12 days in a car, a Suburban with no air conditioning. There's my future wife over there, Kathy. Anyways, let's see here. Then this other guy, right down the hall from the lab I was in, somebody I would talk with all the time, someone who taught a class along with Steve on ecological theory that changed my life, John. And John, I'd done a lot of, in my first couple years in college, I was in all math and physics and chemistry, no biology. I took a biology class that had ending lectures on ecology, and I was hooked. I never wanted to do anything other than ecology after that, so I took John and Steve's class in theoretical ecology. And I'd wondered when I jumped into ecology, what are the forces of nature that are like gravity and magnetism in physics? Well, John taught me what they are. They are looking at whatever it is that controls the per capita growth rate of a species. So basically, ecology is differential equations. And I still, when I walk in the field, I see differential equations. I tell my students, did you see that differential equation over there? And it's all because of John, so thank you. Julian Adams, another one. Julian was on my doctoral committee. He hired me to work in his lab. I don't know why. I knew nothing about what he was doing, but he was very kind and let me do whatever needed to be done in the lab. He had the first computer I ever saw. It was an ADP something 10 or something. Yeah, and to get this computer going, all of you people who are younger than 67, I have to tell you how it happened. Or 77, that's what I am. First, there were toggle keys in it where you had to toggle in a 0 or a 1. And you had to do that like 8 or 9 times. When you'd done that, you put a little tiny program into the memory of this machine, which had no memory, that could then let it read a paper tape. The paper tape then got it going so it could start reading other things and could finally run all of your computerized instruments, which I'd never seen before. Instruments gathered, hooked up to a computer to gather all the data, something I've copied throughout my life. But the best thing was my oral exam, the preliminary oral before you were allowed to be a candidate. Besides walking to your office where it was held and dropping the 6-pack of beer John Vandermeer said I had to bring, and being utterly nervous, I proposed my ideas on how to look at competition. And I had these really complex dynamic models I thought would be the way to do it, where you give, the way you sort of understand a complex physical system, where you give pulses, a Dirac delta pulse, a sine wave, a square wave, and from that you understand the internal dynamics. I knew all the math. I was really delighted by this. And Julian said, well, Dave, what's the simplest possible model you could imagine that might answer these questions? He introduced me to Occam's razor, and something that changed my life. So that's another event. And I would say I've had lots of grad students, and I have some grad students who in my mind I call, and maybe not to their face, I call unguided missiles. Incredible power, incredible intellects, but they don't know where they're going. I was a massively unguided missile, and I was the guidance systems that were put on me by all these people at Michigan were incredibly important to me. Beyond that, Nelson Harston, walk over, Steve Hubble dragged me every week over, even as an undergrad, to go to Harston's discussion of scientific papers. And I saw how Harston could find every single flaw in a paper and also find the good in the paper. And I tell my students, first you have to find the flaws, then you have to ask yourself, what is good, what's novel, what's important? You have to do both of those things really to be a scientist. And then others, Dick Alexander, I learned evolution from him. The ideas that he taught me changed my life. And Dan Jansen was another one who came in near the end of my career who had a big influence, who kept asking me these really tough, critical, big questions to which I didn't know the answers. And I spent a lot of my life trying to pursue answers to those kind of questions. Then finally, Steve Hubble left when his new wife wasn't going to be hired by Michigan, because back then Michigan women weren't really allowed on the faculty very often. So Peter and Susan Killam were hired. The committee was chaired by Margaret Davis, who made sure that Susan had a job when she came here. It was actually canceled when she arrived. You don't want to know how bad it was. But they were amazing mentors. They took somebody who had lots of ideas but didn't know really how to do good lab work and taught me how to do cutting-edge research on algae and competition and so on. So that was sort of the beginning story. I have another 45 minutes, right? So I've done a lot of work on lots of issues, and biodiversity has been a big one. I'll just list the only slide on biodiversity. But basically, it surprised us. We didn't know when we started setting this experiment up in 1993 whether changes in diversity mattered very much or not. Most people thought diversity wouldn't matter. We didn't know, but we thought there were reasons to believe it might, including a paper you wrote on over-yielding and Lockyer-Volterra models. So we now know from all these experiments, greater diversity leads to greater productivity, greater stability of that, resistance to invasion. There are the competitive interactions among our major kinds of species, Poaceae, Fabaceae, and Astrids, are highly phylogenetically constrained, which actually, because of their biochemical differences, turns out you can get a really good phylogeny of these species by looking at their tissue concentrations of nitrogen, calcium, potassium, and boron, and know nothing else. It's just like one-to-one phylogeny for our 60 or so species. It's really amazing. And also that because of these chemical differences, as these species grow together and compete, they actually increase all of the elements plants have to have, because each one is a specialist on extracting some of these from soils in ways that nothing else can extract it. But I want to now this is a homage to someone whose PhD thesis was on paramecium and competition. Because here I have all these plants competing, And I really sort of felt like I'd gone past Lactobacterra to other modes of action. But Lactobacterra has a power, because it is so phenomenally general, to let you see insights that might be harder to see otherwise. So I decided, given 30 years of data on the abundance of all these species in these plots, and how when one goes up and goes down, they seem to be impacting each other, I could estimate alphas, Lactobacterra competition coefficients. And here's sort of what we get. That's the whole spread of the competition coefficients. The only point I want to make right now is that the average value is about 0.2. That means 100 new grams of a new species coming in, on average squeezes out 20 grams, 22 grams of what's there. If that's really what happens, you can see why when you add more and more species, ecosystems become more productive. Now, a good question is why are things competing so weakly? And basically, I don't have time to tell you anything other than there are at least 7 major axes of differentiation that the plants I study are differentiated along. And in 7-dimensional space, it's really hard to get a lot of overlap. Okay, so this is some simple math that maybe John's already done in some paper I don't know about. But it asks if you have many species competing with just classic Lactobacterra equations, how does the total sum of all those competing species change, and how does it depend upon their carrying capacities, their average competition coefficient, and on n, the number of species? And so that's analytically derivable. And it turns out because even if there isn't a single value, if you just look at the mean K and the mean alpha does a really good job estimating mathematically the actual solution to the equations and the total productivity of all those species, even ignoring the variance that might occur among them. And so you can apply this to this case. We had a value of alpha, remember, of 0.22. In place of alpha, you put in 0.22. There were 10 species for which I could get alpha values. The other ones were too rare to have anything other than non-significant effects. And you plug that in that, and it says that the total biomass, when these species are all competing with each other, should be 3.36 times the average carrying capacity of each species. We know that because we grew them all in monoculture as well as in mixtures. And so this predicts a 236% increase in productivity. By the way, this is unpublished. If any of you people here want to publish this, please do. I don't need another paper. But actually what happened, so 200-some% increase predicted, 211% increase actually observed. So this simple approximation, going back to the very, very fundamentals of Lactobacterra, seems to explain a lot. Oh, wow, wow, this is fabulous. But if I talk you longer, I have to charge you tuition. I am a professor after all. So that was what happens in grasslands I know. Here's a system I know nothing about, just having visited once. So the mammal communities, the herbivorous communities of African plains. And there's a neat paper done by Rob Pringle's group, Kratzinel, I don't know this person, used DNA metabarcoding on fecal samples from the 7 or so major species of herbivores for which they could collect these fresh fecal samples, found out what plants each of these species was eating, and from that could calculate dietary overlap. And the lack of overlap is what causes competition. Sorry, the overlap causes competition. The lack of that is the alpha value here. So here is the mean, oh, I'm sorry, I didn't say that quite right. This shows the dissimilarity of diets. The similarity is a measure of competition. And so the similarity is basically 1 minus this dissimilarity that they calculated in this paper, giving an average alpha value of 0.35. So this means that by having 7 herbivores living in the plains of Africa, instead of having the average productivity that each one might have in monoculture, the 7 together should be about double, 126% greater, a little bit more than double the average abundance. So diversity there, if you will, should be doubling the productivity of that system. I have no idea if that's true, but I bet it is. Okay, now diversity. There are realistic concerns, as we all know, about what current and future human actions might do to biological diversity around the world, to the species that are increasingly threatened with extinction, and what might the long-term outcome of those threats be. So I'm going to just sort of jump into work that has occupied most of my more recent time. The top graph shows you just large mammal diversity. You can make maps like this for anything you want. The data are fabulously available, probably because of museums. And then the number of percent of species that are threatened. What you can see is that sub-Saharan Africa has incredible large mammal diversity, and very few of them are actually threatened in a serious way. So if you want to save diversity, you want to keep something like this so it doesn't change. So let's, well, let me see. I guess the next thing I want to point is this, is just that when you look at the IUCN data, the single biggest threat that is causing increased risk of extinction excuse me, is land clearing. Most land clearing is for agriculture. Some is for logging that ends up becoming agriculture, and so on. And so the issue then is, where are we going? And excuse me, you can look at the, is there water? That'd be nice. Thank you. So we've looked at the UN projections of future population. Thank you. We've looked at OEDC and World Bank projections of future economic growth. And the two main variables that determine crop demand are how many people are eating the crop and how much food, how much crop they demand to be grown for the food they're going to eat. And when you combine those variables in a way that we published a paper on this in PNAS in 2011, you can, from that, you can guess how much crop demand might increase in various countries around the world. And this is the ratio of future demand 50 years from now divided by current demand. And you can see that there's an incredible increase in crop demand from both population increase, but especially from income increase. The fastest growing economies of the world right now are in sub-Saharan Africa on a percentage basis. So 3, 4 times, up to 5 times more food demanded in 50 years than demanded now. That has serious implications. First of all, this region is a region which currently has inadequate diets for many of its citizens already. And so the tendency looks like it's going toward an increased number of people with inadequate diets. So food provisioning is a very critical issue that needs to be met. And the question is, how might that be met? Well, if it's met by looking at what has been happening over the last 60 or so years, and at the rates of increase in yields in each of those countries, for every country around the world actually we do it, what you see is that the amount of land that would be cleared would greatly increase extinction risk. So the bar on the bottom are the current average extinction risks, where basically 3 is on the verge of extinction, and 4 is really extinct here. In 3 of the tropical regions for both mammals and birds. And the bar on top in the yellow-orange box is what we project would happen 50 years in the future because of increased food demand, assuming that yields keep increasing the ways they have been in the past. And so basically sub-Saharan Africa would become worse than what we see right now in tropical Asia, where an incredible number of large mammals are threatened with extinction. So there was a meeting that was just held a few weeks ago at the Royal Society in London to address these concerns. And it had an amazing group of scholars there from all around the world doing agricultural research, economic research, development research, environmental research, and so on. That's just a fabulous meeting, probably one of the best ones I've ever been at. And I'm hoping we have some positive impacts because of this meeting. Halfway through the meeting, we met with 20-some people from major embassies, and they're mainly coming from sub-Saharan and other low-income countries about some of the ideas on sustainably increasing yields. Sustainably in the sense that it would decrease environmental impacts at the same time it increased yields. There are many possible ways this could be done. I'm not here proposing any one of them to show you, but I want to say this. The ideas have been tried, and this shows you for the world of the yield that is attainable, the yield that agronomists growing crops there on real farmer fields say, here are the actual yields for each of these crops that we know we can achieve on this soil and this climate in this region. What proportion of that is actually being achieved? For parts of the world, the proportion is 75, 80%, up to 90 in some parts of the world. But if you look at, let me see, what is that? Western Asia and across many parts of sub-Saharan Africa, the yields are only 15 to 30% of what they could be. And so finding ways to increase these yields, I think, is of great importance. First of all, it would allow these countries to meet the current and future food demands of their citizens in ways which I think access to a healthy, secure diet is a fundamental human right that we have to recognize and do everything we can to achieve. I also would say if it can be done, and our work suggests it really can be done, at the same time, we could be preventing lots of what would be needless land clearing and preventing both greenhouse gas emissions from that and preventing future extinction risks. So this is that's what the world looks like as far as where things are under yielding. I showed you a map of where the food demand was. And this is how much new cropland would be needed if all the countries achieve what the agronomists say is their potential yield, their achievable yield for all of the major food crops. And what you can see is that the two blue colors are needing less land than we have right now. And that actually applies through most of sub-Saharan Africa. It's only a few places that need much more land, that would need much more land than they have now, even though they will have increases in income that change their crop demand and increases in population. So I have 7 minutes left. Yes. I talked too quickly, I guess. Oh, thank you. I would love to hear your thoughts. Since I have some time, I was told if I get done early, I can have questions.
Any questions or comments? Or especially criticisms?
I'm just fascinated with the idea that your theoretical project in Minnesota ends up having a worldwide impact on how we can look at world food needs.
Well, it could have a real impact. I hope it does. That's the hard part is getting 8 billion people and all these governments to do what is really best for them, as we know. It's easy to propose solutions. It's very hard to achieve them, which is one reason why I was so excited by the Royal Society event that occurred.
I noticed that Australia needs to increase its agricultural output. How are they going to do that with a desert and very unreliable water sources?
Yeah. Well, that's a good question. There are some countries where lack of water means agricultural production is not really feasible in a meaningful way there. And so some countries will have to import food from other countries. And so if you looked at that, countries like the United States and China would only need about half of their current cropland. So those countries and other countries which can produce more than they need could be exporting to countries where they were growing food is a very difficult process. And in fact, because countries that tend to export food tend to have some of the highest yields, it means that exporting those to countries which have low yields actually greatly decreases how much land might be needed to be cleared there. The dilemma with export-dependent economies for food is that the exporting countries can use fear of loss of food as a political weapon against them. And I, again, would assert to me that's about one of the more immoral things you can do is to try to control another government by controlling the food its people have to live. But I think that is one of the big issues is the stability of food supplies. Why China, which does not have enough good land to feed its population, is trying to come up with some permanent lands they can use in other countries to have a guaranteed food supply where they won't be beholden to the whims of the next U.S. president.
That was a fascinating talk. I wondered if you could say something about all of the land that we're using to grow biofuels instead of sustainable.
I could have paid you for that question. I didn't think I'd have time to do this, or I would have shown you. Right now, the corn, the maize grown in the United States, 40% of our maize becomes ethanol for cars. The calorie content of that maize would feed a little over 1 billion people all the calories they need for a whole year. So every year we turn that amount of food, if you will. Not that all the corn is eaten, but corn actually is a grain that many people eat as their main source of often protein and calories. And instead we feed it to cars. And in Indonesia, 40% of the palm oil they grow is used to make biodiesel for vehicles and is sold around the world. To me, these are both unwise environmentally and absolutely immoral. Because when you sell more of a crop, everything we know in economics says the remainder part of the crop that does go to the food supply is smaller, and that increases the price. This happened after the United States adopted the 2006 America Energy Independence Act, I think it was called, where we imposed a 10% requirement for ethanol in gasoline. It meant a lot of corn went to ethanol, and because of that, it wasn't on the market. It caused all grains, since you can grow wheat, corn, and soybeans on the same land, they all went up in price. We had food riots in countries all around the world. We had countries, governments that were destabilized because the very least the government has to do is feed its people for its people to accept governance. And so it's, to me, biofuels are an immoral issue, but they also are environmentally unwise. We wrote some papers on this, and I had a guy who owned hundreds and hundreds of gas stations in Minnesota and Wisconsin who I met. And he said, "You're my hero." I said, "Why?" He said, "You found something worse than gasoline, ethanol." Yeah.
Well, I couldn't agree more or less on that matter. Question about your projections. Did they take into account the climate change and the increased heat stress and drought that will happen over the next, say, few decades?
Would you do those calculations for me? We've written some papers trying to guess at that. The dilemma is what economists call the dumb farmer. If you assume they'll keep growing the same crop in the same variety in the same way, yields are going to go down. If they switch to other varieties and other crops, yields might not go down so much. At some point they will, when you run out of water. But actually, warmer Earth gives a lot more water in the atmosphere. So where we're heading there, I'm not sure. John?
And what if they intercropped?
Oh, intercropping is a brilliant idea. Championed by this gentleman here for a long time, and I knew you were doing that, and I always was interested in it. Now one of my PhD students is doing an intercropping experiment with real agronomists at the University of Minnesota, looking at corn, soybean intercropping being planted out with normal mechanical machines and be harvested that way and so on. We'll see what happens. But on average, across hundreds and hundreds of intercropping experiments that you know, there's about a 25% increase in total yield by growing alternate strips or rows of two different crop species. And the more ecologically different the crops are, the higher the yield increase, as you would expect.
No time for more questions. Thanks very much.
That's very hard to follow. My name is Max Witinsky. I'm a PhD student in Ben Winger's lab. I study life history evolution in the context of seasonal migration in birds. It's my pleasure today to introduce Dr. Trevor Price. Trevor graduated with his PhD in 1984 from Michigan and is now a professor at the University of Chicago. He's been a leading scholar on the field of speciation in birds and has a remarkable research program that combines Himalayan fieldwork, ecology, behavior, and genetics. Ben, Matt, and I were lucky to interview Trevor and Dolph Schluter about their time at Michigan working with Peter and Rosemary Grant for a long-form interview that's on the EEB website. So if you're interested in that, you can read it. Dr. Price has been the recipient of many awards and honors. He is a fellow of the Royal Society and the American Academy of Arts and Sciences, and also a Guggenheim Fellow. And he's also been the recipient of an E.O. Wilson Award from the American Society of Naturalists and a Mercator Fellowship. So please join me in welcoming Trevor back to EEB.
Yep, this is going to be a hard act to follow. Right. Let's see if we can get going here. Yeah, well, thanks to Nate, and thanks to all the great talks we've heard so far today. I'm going to be talking about my time here in Michigan, which was from 1977 to 1984, but I acknowledge that actually the Finch investigation unit lasted 2 years longer, and Lyle Gibbs is somewhere in the audience. He was the person who carried on. So in 1973, a postdoc to work with Peter Grant, Ian Abbott, and did his project in the Galapagos trying to figure out the involvement of competition in the adaptive radiation of Darwin's finches. A couple of years later, Peter, realizing what a great place it was to be and what a gold mine, what he wrote to me and said, we've got an ecological gold mine here, he set up a project on this little island called Daphne Major, which is just off the island below it of Santa Cruz. And he basically decided he was going to do a population study there, testing Lack's famous idea that this was known as the classic case of character release. So on Santa Cruz, there's a small ground finch and a medium ground finch. But on the small island of Daphne, there's only the medium ground finch, and it's intermediate in size between those two. So this seemed like release in that the medium ground finch had flown over to Daphne. In the absence of the small ground finch, had evolved to be a medium size, so it could consume the resources of both species. But people had suggested, in fact, that's not what's gone on. There were two other possibilities. One was it was a hybrid swarm, which Lacker had considered and rejected. And the other one is that, in fact, the resources are different. And this has been always a problem between testing character displacement and character release. So he set up this project, and the first graduate student to work there was Peter Bogue. And this was done while Peter was still at McGill. And they published, believe it or not, in Nature, a paper called The Heritability of External Morphology in Darwin's Finches, a bunch of parent-offspring regressions in Nature. So I was looking around for a PhD, and even though it was just a bunch of parent-offspring regression, I was pretty impressed that they were actually studying individuals. And actually, Peter and Rosemary Grant had already, with Ian Abbott, started looking at color-banded individuals and showing that they were actually different in what they were doing. So this was pretty big, because before that, everyone was just sort of looking at species means, or considering every individual within a species to be similar. So I was enamored with that. So I said, okay, well, sounds like a great idea. I'll spend the winters in the Galapagos and the summers in McGill for 5 years studying Darwin's finches. And before I got to McGill, Peter had moved to Michigan. So myself and my other more famous big buddy, Dolph Schluter, in the middle there, were, I think, maybe the only people never to take a GRE. We just came as part of Peter's startup funds. And we had to fill in an application, and Dolph just left it blank. And of course, I filled in all sorts of stuff, but it was completely ignored, and we were enrolled in Michigan. So this, we stayed in this 13-person house, and the person on the right there is Nancy Heckman, who ended up marrying Dolph. But it was a great experience outside of the department, because she was a mathematician. So we actually published some papers together, the three of us, as a result of her math expertise. So I don't know what I was doing with the balloon, by the way. I've got absolutely no idea. During that time, or when I arrived there was actually a second paper that came out of Peter Bogue's thesis, which was very high profile. And that was the discovery of intense natural selection in this population. What had happened was, once we started to, or Peter started to measure the resources on Daphne, was you've got this variable finch, the medium ground finch, and you've got small seeds and you've got large seeds, basically. Two kinds of large seed, one of them being a cactus and one of them being this tribulus, and a bunch of small seeds. And Peter Bogue went back to Daphne for his second year, and there was a major drought in 1977. 85% of the birds died, and of the 15% that survived were much larger than the ones that died. So they had this selection event, and what I've got here is a plot of beak size. It's actually a principal component, but it's going to appear again and again. And you can see that from across Peter Bogue's study, beak size went up substantially. And because they got this cohort of banded birds, they could actually measure the intensity of selection on the birds. Great. So, oh yeah, and I didn't put the data in, but because they'd been measuring the seeds, Peter Bowe could show that the small seeds have basically disappeared from the island, but not completely, and that will be relevant later. So they had a mechanism, and they had this strong selection, and you could work out that a few more bouts like that, you turn a medium ground finch into a large ground finch. So this is pretty good. So I was able to write my thesis proposal to go ahead and ask what will happen when it rains again. Surely there'll be selection back, and we could work out why. Dr. Adams was on my committee. Pretty skeptical he was at the time. And with good reason, it turns out, because I was on the island for 3 years, and it rained a little bit. What that meant was that nothing died and nothing reproduced. So you can laugh about it now, but I would say that, you know, just talking to graduate students, the problem with being a graduate student is that your whole life depends on your one project. Once you become a professor, at least one person in the lab everything's working out on, and you just ignore everyone else. So there I was on this island. I mean, it was a great place to be, I don't deny that, but I was getting pretty depressed about the whole situation. So I had my 3 years, and then Lyle arrived. So yeah, in my 3 years, absolutely nothing happened. I was having to write a thesis. My 3 years are up, that was clear. Lyle was coming. I don't know if you'll recognize him, he's in the audience somewhere. But here he is on Daphne with the boobies in the back. And this is what happened. So there's Bogue, there's Price, and here comes Gibbs. Yeah. And actually this El Nino in 1983 transformed the island. So it went from being essentially a large seed island to a small seed island, and it stayed that way for years afterwards. Incredible change. So, as you might expect, we had a return, a decline in body size back towards where they were. So, Lyle had his Nature paper as well, oscillating selection on Darwin's finches. So what was I going to do? I mean, he was already there. He didn't write this when I was there. It was a bit later. Thank you for doing that. But two things happened. One is that Peter Bogue and Lyle were great collaborators, so they let me use their data. So I had Lyle's first year of data. And in fact, this was associated with three droughts, the big one you saw, a minor one in my year, 1980. We did have some mortality, and his first year. So I had 3 selection events I could study. And the second thing that happened was at exactly that time, this amazing paper by Landian, an alumnus of here, Arnold, came out in Evolution. I must say I used up about half my brain trying to understand it. And then Russ used some of his retention money to bring me to Chicago to explain it to me. So I now had 2 things. I had data from other people, and I had the theory of Landy and Arnold, so I was able to get my Nature paper, basically. With Lyle and Peter, we showed that the selection differential was always for larger size, but it was always a selection on shape, where big beak depths were favored, and actually beak width was under selection to decline, but because it was correlated with beak depth, it got bigger every time. So that was pretty cool. And of course, that study is carried on. After that, Peter and Rosemary made it clear that no more graduate students. In fact, anyway, we won't say anything more about that. And they've written a book. They went for the next 30 years and wrote a book on it. And they had this amazing drought in 2005 where the birds got smaller for reasons I'd be happy to talk about. And this is really what I think is really remarkable. When Peter and Rosemary when Peter Bogue wrote this heritability paper, a bunch of genes, it just seemed completely amorphous. But just a couple of years ago, absolutely brilliant paper by Eric Enbody and others. There's that plot at the top. They'd used a GWAS to identify genes that affect beak size. And look at all these beak genes. They all increased during that drought. And this is actually a reflection on the fact that Peter and Rosemary had started collecting blood from 1985 onwards. Absolutely amazing to see the genes moving, really show that it was genetic change. Right, well, let's just, finish up by, I was in the Galapagos, and so was my friend and colleague, Dolf Schluter. So what was he doing? Well, I was sitting on Daphne waiting for rain. And basically, he set out to really rigorously test the Ian Abbott, Peter Grant original study. And he came up with this, what I still think is I wouldn't say it's the best paper on Darwin's finches, but I think it's, the most phenomenal paper on Darwin's finches I've ever seen. And it's an interesting point, actually, because it's by far not one of the most quoted, but if anyone wants to do anything interesting, they should read it. So what Dolf did was he managed to use empirical data to say if we had a single species on an island of a particular beak depth, how much, what would be the biomass of that species? So on Daphne there, we've got some small seeds and some large seeds, as you've seen. So what happens is where you've got small seeds, you could support a small finch, but as the finch gets bigger, you can't support as many of them for a couple of reasons. And then you suddenly get to a beak size where you can exploit the big seeds, and then it goes down again. So this was a way of translating seed size distributions into beak sizes, and he did it for all of the islands that he'd visited. And one of the things he was able to show was that there's one finch for one peak. So we have an adaptive surface, and he was able to, I'm running out of time, but Peter Grant was talking about the great Daniel Simbaloff, Jared Diamond debate, and he said, this shows that some of Simbaloff's null models may not be null, although they could be a bit void. So anyway, just to quickly finish up, we could then return to this Daphne situation. So we went back to the classic case of character release, and Dolf and I were able to write a paper together using his data and mine together. So remember, we've got a small and a medium ground finch, and on Daphne we've got a medium. And what Dolf showed was that on Daphne, the medium ground finch is closer to the peak than it is on any of the other islands. So after you've controlled for food, there's clear evidence for character release. So he controlled for the food and showed they'd move towards character release. And I was able to use the Landy-Arnold technique on the big drought that Peter Bogue had experienced to show that we actually had, there's some problems with it, but we had disruptive selection whereby the smallest finches on the island were doing better than the ones that were slightly larger. So we had the two niches were being exploited, and that was the reason that the medium ground finch was closer to the peak on those islands. So that basically summarizes my time here. So that paper was written up in Science News, and it said that a team led by Dolf Schluter counted 12,000 seeds and measured 2,000 finches. And that was, of course, Trevor Price and Peter Grant. So here's the team. I think you'll be impressed with the Landy equation, because this was, a first formulation. My brain cells were being destroyed trying to understand it. So I didn't quite know how to work this in, but Peter said, I'm sure you can point out that we did consider an alternative career in Banbury, Oxfordshire, England. We opened a shearing shed, but it was much harder work and not as much fun as being a scientist, right? Thank you.
Hello, my name is Emma Carlson, and I'm a PhD student in Dr. Louise Simon's lab. I use experimental evolution to study host-parasite coevolution in bacteria and bacterioviruses. It is my pleasure today to introduce Dr. Joan Strassman. Dr. Strassman graduated from Michigan with a Bachelor of Science in 1974 and is now the Charles Ribstock Professor of Biology at Washington University in St. Louis. Among many other accolades and awards, Dr. Strassman is a John Simon Guggenheim Memorial Fellow, an American Association for the Advancement of Science Fellow, Animal Behavior Society Fellow and later President, and then elected member of the American Academy of the Arts and Sciences and is a member of the National Academy of Sciences. Please join me in welcoming Dr. Strassman.
Well, this has been a lot of fun. I just love the mix of memoir and actual science. I'll be taking you back to the memoir side of things here. I was an undergraduate at Michigan, and I think that many of you have been more on the graduate student side. Although I was an undergraduate here, I was completely formed by Michigan, and when I got to Texas for my PhD, there was really nothing new left. So, yeah, this is really all about Michigan, and honestly, my entire career has been all about Michigan. So here's a few details. I used to be a redhead. It's really tough on my identity to no longer be a left-handed redhead. That I have a redheaded sister and another sister, and someone once wrote Hellcats on our mailbox at home, and we kind of embraced it. So I started at Michigan as a French and linguistics major, and I finished high school in Switzerland, so I already knew French and linguistics. I placed highly, and I thought, what else is there than the 4 more courses I needed for both majors? Maybe that's an exaggeration, as all of this will be. But it turned out that I braved Bio 106 as distribution instead of Bio 100. And that was a fabulous course. And I heard about this place called the Biostation. Actually, that was the next year. And I went there, and I discovered that the kind of stuff we did as kids with the forests I grew up in East Lansing, mostly, you could get a career for mucking about in the fields and woods. And I was just amazed. So I was there for the summers of 72 and 3, and the winter course of 73. Actually, I took that. It was a January course made for these fancy schools that have January semesters. It meant I had to just skip the first 2 weeks of UM. And once I realized I could do that, I kind of skipped most of the rest of this whole semester to study a vernal pond. I was also there in the winter. The courses I took at the Biostation were life-changing for me. I took Boreal Flora with Ed Voss. I think I was in the bottom third of the class with my 96 average. Even more influential was Insects with Jim Lloyd, who told me when I got back to Ann Arbor I needed to go look up this Dick Alexander guy. Winter Ecology, I think I have a couple pictures of that. Anyway, I took Pleistocene Geology with Bill Ferrand. I heard at some point Michigan was saying it's the first time a geology course had been taught at bug camp, and it wasn't. I did an independent study on a vernal pond for many, many months. And this is the one negative thing I'll say, and that is I learned what it was like to not be properly mentored. And I have ever since paid an awful lot of attention to how I am mentored and how I act as a mentor. And one of the things I really know is that you can't predict who's going to be worthy of your mentoring and who's not, but you can crush people. So you just don't ever want to crush or judge anyone because you can't tell who's going to end up how. So here I am in winter ecology with a friend. We were doing, in the winter, a lot of the interesting insects are in the streams. So it was really the only time in my life I've worn both waders and snowshoes. We are shown here entering the Maple River. We didn't just stand on the shore. We went right out in there and started seining and stuff. And anyway, it was an amazing course. And then when I got back to Ann Arbor, I found Dick Alexander. And basically, his philosophy is still my philosophy, and his questions formed certainly my first questions. I think we've gone in directions he might have gone on had he known what amazing things we can now do. So, at that point I was thinking a lot about mentorship, and I thought, well, I should meet with you every week for an hour. And he said, okay. I was like, I kept up with him his entire life, and later I was like, Dick, an hour a week with an undergraduate? What were you thinking? My husband was a grad student. He said, maybe once a month, if that. But I met with him once a week, and Dick kept giving me more and more stuff to read. And I now realize, if I ever said I can't make the meeting, he never seemed sad. Anyway, so this continued all through grad school. I graduated. I went off to Texas. I was still answering Dick Alexander questions. Somehow I never let him, so I'd come back to visit my family. I never let him know when I was showing up. I knew if I showed up around 11, he'd take me to lunch because he was always looking for someone to go to lunch with. And by then, Bill Hamilton was in town, and so I could go to lunch with Dick, and then I'd spend the afternoon with Bill. And honestly, they were both incredibly critical and never really supportive at all. I remember vividly one time when it was 1977, which was a big drought also in Texas, and a third of my wasp nests had died. And I was talking about it, and Dick said, well, Joan, if you don't know why, what is it you do know? So it wasn't exactly supportive, but he spent all that time. Anyway, could go on about that. Little bit of science. This is something you all know, and it was really kin selection that I was looking at. This is a extreme simplification of what I worked on. Spent a lot of time using molecular methods to get at relatedness. Costs and benefits are what actually turn out to be where all the ecology and biology that's a lot of fun to look at is. So there's a tiny bit of biology for you. I had a hard time picking a thesis topic. I wasn't one of these people that logically figured out the best thing. I kind of more was of the desperate flailing, and maybe if I look at something will turn out okay. So I knew Dick. I knew Mary Jane West Eberhard. She seemed to do okay with her wasps, so I thought, well, maybe if I go to Texas, the wasp is subtropical, the wasps will be different and I'll find something. And so that worked out rather well for me, although I have to say I never got rid of my kind of pathological fear of wasps. Dick had advice. I still have this on my field supplies cabinet. I retyped it for you. how to stay optimistic while living in the field. I like a few of them. Do the easiest things first, have several things going at once so you can afford failure. Grasp at straws, I love that. And never give up. So field work is my jam. So here I am using a mirror to look at a wasp nest. We did a lot of this. By this time, I'd finished at Texas and had a job at Rice, and we hired a junior faculty member, a Huxley Fellow, another Dick Alexander student, this guy, and he had worked on plants for his PhD, but had done a WASP project with Katie Noonan while at Michigan. You'll hear from him later. And it's Dave Queller, in case, for you, those of you that don't know. Anyway, he said, well, if he works with me, he had this experiment in mind, and it would require that we find about 80 nests of foundresses in the field, you know, in a short period, and then we could do this experiment and all. And I said, oh, that would be no problem at all. And we went out, and along with grad student Colin Hood, we found the 80 nests, which I have never done before or since. So I guess the gods, the stars were aligned. What can I say? As long as I'm talking about the huge family impact that Dick Alexander had on us, it would be appropriate at this point to mention another family member who worked with Dick, has done some really fabulous work on the Dogon. It's a human group in Africa, first as a behavioral ecologist, now really complicated epigenetics with a data set of 4 generations of human. Beverly Strassman, she's here, I'll embarrass her, she's right back there. So anyway, I did get to visit her there once. Well, I said I was afraid of wasps, and that certainly wasn't why we stopped working on wasps in the field in Tuscany to move on to social amoebas in Texas and in the lab. But we were intrigued by these amoebas because they have a group 20% die to help the others, so it can be like a wasp colony where the workers are sterile. But these form from aggregation, so they might not be as related as one could figure out in wasps. There was a collection of these amoebas from the wild that turns out to be very friendly Dicty community gave with them. And we were intrigued by them because this was a system with a genome when we needed DNA microsatellites to look at relatedness. I didn't have to make libraries and probe them and all of that. We just had to go to the genome. And there were all these fancy molecular biology techniques where we could knock out genes and really get at the molecular basis of things. So Dave and I, about 25 years ago, largely switched to this system. And we're still working on it. We're not actually working on the kin side. We're working more on the predator-prey side, unless you think I'm only history now. Our current grant is a grant for predator-prey, and I just got an email from our postdoc yesterday showing that it looks like the way that the Dicty predates on different bacteria of different edibilities is what Dave calls the Anna Karenina hypothesis, which they tend to eat the good ones in all. This is a RNA-seq type project. The gene profiles are the same, whereas the bad ones turn on different kinds of genes. So we're still at it. So my advice is simple. Go to a great university if you want to find a great question. Find a great mentor and listen, although if any of my mentors were in the audience, they would get a big sarcastic laugh at this point. And work in the field. So I'll just close by saying my other interest while I was at Michigan was writing fiction. And I did this the whole time I was here. I wrote short stories. I even won Michigan's top undergrad fiction prize, the Hopwood. Took me a while to get back to writing, but I'm a believer in public communication of organisms people actually want to read about. And my last thing I'll say is the thing I should actually be most known for, I'm actually not known for whatsoever. And that is, I have through the years had my behavioral ecology students write pieces on single organisms, on single species. Fill out, this semester we picked crustaceans. Fill out the encyclopedia of the world. I've had my students doing that for about 20 years, a little less than 20 years. And the Wikipedia education people just told me that the articles my students have written have been accessed 60 million times. So that is, to me, far more significant than anything else that I've done. So look at wiki.edu and see what you can do. Thank you.
Hello. My name is Teresa Sauer. I'm a PhD student with Megan Duffy studying natural multi-host, multi-parasite communities in freshwater systems and zooplankton. I'm so excited to introduce Dr. Carla Cáceres today. Carla graduated with high honors in 1991 with a BS in biology, and then went on to Cornell to get her PhD with Nelson Hairston Jr. She is now a professor and the director of the School of Integrative Biology at University of Illinois Urbana-Champaign. Among her many awards, she's a fellow of the Ecological Society of America, as well as the Association for the Advancement of the American Association for the Advancement of Science, as well as a sustaining fellow of the Association for the Sciences of Limnology and Oceanography. She's also a recipient of the Presidential Early Career Award for Scientists and Engineers, and overall just an incredible and influential aquatic ecologist and evolutionary biologist. So please help me in welcoming Carla.
Teresa, thank you so much for that kind introduction. I am so honored to be here. And again, I was an undergraduate here, so my story is a little different, and I hope that this resonates with some of the undergrads in the room. And so, how did I get to Michigan? Because I grew up in a small rural town in northern Illinois. And we moved there when I was 6 years old, and we were fortunate enough to have lake access. And I loved this lake, and I had so many questions about it. What were those fish that were biting my toes actually eating? Why did the plants look different in this side of the lake and that side of the lake? What was the slimy stuff that I was mounting on my microscope slides? What were those things actually called? But there were no scientists in my family. It was rural, the school districts were bad, so I thought if I wanted to study water, I had to be a marine biologist. When I got to high school, I actually did meet two scientists. They were parents of some of my friends, and even though they weren't ecologists, they both had gotten their bachelor's and PhD from Michigan. And so they helped me understand what a PhD was, how you might want to pick a school that could take you to do the career you wanted to do. But the real reason I came to Michigan is I was a band geek, and I really, really, really wanted to march in the Rose Parade. And so there's me leading my line. We did go twice, once in '89, once in '90, one lost once. And so this was the reason 17-year-old me made the decision to come to Michigan because of the marching band. years. But meanwhile, I was a biology major, and I was going to be a marine biologist, right? And so as soon as I could, I registered for marine biology, which was the winter term of 1989. And marine biology that semester was taught by Killam, Wynn, and Lehman. Killam started by teaching the course and then went to Kenya to do field work. And unfortunately, that was the semester where he passed unexpectedly. And so I never really got to know him that well. I just had those few weeks of him lecturing and the tests that he wrote before he went to Kenya. But the rest of the semester was finished up, and Lehman came into this class that had about 80 people in it and said that he had an REU opportunity to work with him on the Laurentian on Lake Michigan during the summer. Who was interested? All 80 of us raised our hand because we were going to get pay and course credit for it. I almost didn't apply. I was a very serious student, but I came to Michigan completely underprepared for a STEM major. So no matter how hard I studied, no matter how many times I reviewed my notes, I just couldn't get that A. My mother forced me into it, I was selected, and that changed my life. So I was given a choice for that first summer. The Laurentian had just acquired a new CTD, conductivity, temperature, and depth probe, and I could calibrate that against the other instruments that had been previously used so the long-term data set could be maintained. Or I could study this. This is Bythotrephes. It had just recently invaded the Laurentian Great Lakes. It had one eye. It carried its babies around in this brood chamber. It's a predator, so it has this giant trap basket, and I was just enthralled. I was particularly enthralled by its life cycle. So I didn't really understand what cyclic parthenogenesis was at the time, but I was told that when conditions are good, there's only females in the populations, they reproduce clonally, make more females, and then conditions get bad, some females will make males, others will make these haploid eggs that once they're fertilized, enter a state of dormancy, and they can remain dormant for who knows how long. And I was just enthralled. How did they know when to switch? How did they know when to hatch? How did they know when to find their mates? There were also these other related invasive species that came in later, and they just were the weirdest thing at all. So I chose to study Bythotrephes and had this wonderful year-long experience. I also want to acknowledge the importance of graduate students as mentors. So Kim Schultz was my graduate student while I was here, and she helped me put together my poster. This is what we used to have to haul to meetings, not some rolled up tube. And I had 2 different publications that came out of that. But at the start of my second year, I was told if I was going to be a real limnologist, I had to do something other than plankton. I had my entire career to study plankton, and so I needed to do something else. So I was told to go get all these papers by this guy named Tillman and read all about R-star theory, and I had to filter so many water samples and size fraction those. I was also told to reach out to Sue for help, and Sue was so amazing to me those years. And it was, Peter had just died, she was dealing with having to move, but she still always had time for me, and she was always that smiling face in the audience when I was terrified giving my talks at national meetings. So Sue made just such a difference in my career. I also, Bythotrephes, there wasn't much written in English about it, and so I had to find other zooplankton papers, and I found the series of papers by this guy named J.D. Allen. There's my actual photocopy that I had read over and over and over. I thought he was at Maryland because that's what that said, but actually he had returned to across the street after completing his master's and PhD on zooplankton here at Michigan. So I left Ann Arbor, but Michigan's influence continued. So I wanted to study dormancy in zooplankton. So I went to work with the world's expert on dormancy in zooplankton, Nelson Harston Jr., who is the younger looking guy in the photo. Next to him is Senior, as we've heard a lot about, and then next to him is Bob Payne, who actually I didn't interact with, but I asked Nelson for a picture for this talk, and I thought it'd be rude to crop Bob out. But you can see, so Nelson Jr., my advisor, got his degree here in '71, and you've heard the stories of the other ones. And also, but senior was on Bob's committee, and Bob was on junior's committee at Washington. And I had junior every day in the lab, and then about once a year, he would announce that father is coming, and it was terrifying to us graduate students. And then senior would show up in the lab for about a week. And so I had both the direct effects and the transgenerational effects for what happened at Cornell. And at Cornell, I also want to point out somebody who I feel like I learned so much from. So Dick Root was an undergrad here with Frank Smith while he was doing his undergraduate work in the late 50s. And he taught the Intro to Graduate School class, and his grad student Walter Carson was the TA, and they would get into these arguments that I was too young to understand at the time. You can't do an ANOVA on it, Walt. And then he also taught the field ecology class. And so spending time, and he was an amazing naturalist, and also taught me to appreciate terrestrial environments, which up until that point I thought probably weren't all that interesting. But what Cornell taught me, both my official and unofficial advisors, were how to combine my interests in life histories with long-term dormancy and community ecology. In other words, we were doing eco-evolutionary dynamics before we were calling it that. Then I moved to Urbana, and my Michigan connections continue. So when I moved to Urbana, as we've heard from other talks, not so many women on the ecology faculty, but Carol Augsburger quickly became a mentor and a good friend. And I was looking up her dissertation, and you can see who was on her doctoral committee. And I also found her papers that said Department of Ecology and Evolutionary Biology. And so I emailed her on Monday, and I said, Carol, your PhD's in zoology. Why do your papers say Department of Ecology and Evolutionary Biology? And she said, I don't know, but I was terrified because my degree was in zoology, and I'm a botanist, and I did not want to be found out. So now that I've been in Urbana, I have two different study systems, both with connections to Michigan. So I still work on Cladoceran zooplankton, and this picture of our host parasite system is in collaboration with Megan Duffy, who was a graduate student with my collaborator Alan Tessier. And then my midlife crisis was a shift into mosquitoes. Because I thought, those are close enough, but I didn't know anything about working with mosquitoes. But I had this wonderful colleague, Brian Allen, who also got a Michigan degree. He got a Michigan degree because his father is David Allen, and he lived here as well. And so there's my second father-son pair, and we've been working together on mosquitoes ever since. So I don't want to talk too much about the current research, but just that Michigan taught me, both Michigan and my Michigan mentors taught me to think big and to think about the connections that are there and how we make different connections. And so our lab right now, still working on some of the same organisms, but adding things like phenotypic plasticity, anthropogenic stressors, microbiomes, and disease dynamics. And I have these amazing students. And so I want to just talk a little bit about especially the undergraduate students. So Nina, who's on the left, was an undergraduate, now master's student, and she and her mentor developed this whole system on Culex mosquitoes and their intercellular symbionts, Wolbachia, to ask questions across multiple scales of organization. I have graduate students and undergraduate students asking what happens to aquatic food webs when you live in the middle of corn and bean fields and all these broad spectrum fungicides and insecticides end up in those systems. Things like what are the net effects when you add a fungicide to a host fungus interaction, and how do insecticides influence temporary pond ecosystems. And then I've had undergraduates and graduate students working on the microbiome, because why not? Everybody's doing it these days. And so just quickly, as I reflected, and this is where I'm talking to the undergrads and graduate students, reflecting across the arc of my career to date, there are some things that Michigan taught me, like Cladastra and limnology and life histories, that have been a through line my whole career. The one thing I stopped doing is invasion biology and Great Lakes limnology. Great Lakes are just too hard to work on. And so thinking about opportunities to add new directions or new dimensions, I started adding phenotypic plasticity and dispersal and community assembly. Then Meg, Spencer Hall, Ellen Tessier, and I moved into disease. My first omics paper was actually under a collaborative with Kim Hughes on Drosophila. Did not understand a word of it when it was actually that time, so don't let that stop you. Have good collaborators. I took on an administrative position, which I still hold in 2013, and that kind of shifted the career a little bit. But we've still been thinking about mosquitoes and microbiomes and ecotoxicology. So in thinking about what Michigan made possible, this is my little undergraduate me with my mentors from Michigan, my 2 friends and colleagues, and then the transgenerational effects. I was given a chance as an undergraduate, and that then has given me the opportunity to give 141 undergraduates opportunities to help them become the scientists that they are meant to become. And I am so incredibly grateful to Michigan for that. Thank you.
All right, thanks everybody. We have time for a break, and we'll reconvene at 3:15 for Judy Bronstein.
I ended up sticking my flash drive in, and that worked.
Okay.
So now let's see if I can.
Very interesting. Yeah. Bring back a lot of memories too. Good last night.
Yes, very excited.
I was wondering where you want posters.
It's anywhere outside. Yeah. Sure.
to look at it. But if somebody serves me, I'll never say a word. I'll make it much and hopefully after the rest of it.
minute a little longer. She has too many accolades, okay?
All right, find a seat, folks. We're going to get started. Judy Bronstein, please come to the front. Judy Bronstein, please come to the front. Judy Bronstein, are you here? Great. All right, we're going to get started. So if your name is Hernan or Luis, you should find a seat. Hernan and Luis. All right.
Do I put this in? And this is
Hello. My name is Abby Soul. I'm currently a fifth-year PhD student in Marjorie Weber's lab, and I'm also a proud alum of this amazing department, having earned an honors BS in EEB in 2018. I study chemical defense and species interactions across space, time, and trophic levels, and if you're interested in hearing more about some of my research or research of my amazing lab mates, please check out the Weber Lab poster out there, and also Rosie's excellent talk that is coming at 4:30. It is my absolute pleasure today to introduce Dr. Judy Bronstein. Dr. Bronstein received not one but 2 degrees from Michigan, earning her master's in 1981 and her PhD in 1986, with Dr. Beverly Rathke serving as her advisor. She is now a university distinguished professor at the University of Arizona, where she has worked since 1989. If I were to list all of Dr. Bronstein's many accomplishments, we would be here all night. So I will stick to just a few. Among many other awards, Dr. Bronstein is an elected fellow of the Ecological Society of America, an elected member of the American Academy of Arts and Sciences, and has received the, Distinguished Achievement in the Conceptual Unification of the Biological Sciences from the American Society of Naturalists, where she previously served as president. Dr. Bronstein's work significantly advanced our knowledge of mutualism and cooperation in a time where the field of ecology was still dominated by research on competition and predation. Today, she remains a clear leader in ecological research, having quite literally written the book on mutualism. And I am so delighted that she is here today. Please join me in welcoming Dr. Judy Bronstein.
Hi, everyone. It's great to see old friends and new friends, and let me just say that the Weber Lab totally rocks. And so be sure to check out Rosemary's talk and then the poster later. You know, since Nate invited me to do this, I've been thinking a lot about those years and what graduate school was and what graduate school did for me, how it got me to where I am now. And it's always interesting when you look over your past, you can make a storyline about what happened, right? You can make what I call a coherent but not necessarily correct, accurate retelling. Maybe it's just a just so story. I'm going to tell you a story of how I got from there to here. Is it right? I don't have a clue. But it's been really fun trying to process all these thoughts about how I got to this point from where I was in 1979. So I want to go back a little further than showing up at Michigan and tell you about my undergraduate years. I grew up in suburban Boston. I ended up at Brown University in 1975. And this was the early days of environmental consciousness, the first Earth Day, remarkable levels of pollution in the United States, and I wanted to go do environmental policy and clean up the world. And so I went to Brown and I made my own major that was all about environmental policy. And I discovered, in part through work experiences for things like the Rhode Island Department of Transportation, that this was totally not for me. Did not like it. But I took some field biology courses, and boy, was that fun. I mean, I like field work, but mostly I liked pondering. I liked thinking. I liked trying to make connections. And then I was, working in the science library at Brown, and I discovered the work of this guy, Dan Jansen. I would read papers like Why Fruits Rot, Seeds Mold, and Meat Spoils, 1977, and I was hooked. I had the thought in my mind, That's a job? I can have a job doing that? And I was hooked. So I took more field biology courses, and I ended up at the University of Michigan Biological Station in 1978. And this was my first encountering of really super smart ecology students from around the country and graduate students who we didn't have ecology students and graduate students at Brown. And it was a real test for me. It's like, can I do this? Am I okay? And I had such a blast. I took courses from Conrad Istok and John Cairns, and it was absolutely formative. I found a great topic for my honors thesis, came back to Brown, and met Beverly Rathke. Beverly was a faculty wife at Brown who was given a small room in a building and allowed to write her papers and allowed to teach some courses. Her husband was a faculty in the department. And I came under Beverly's wing, and it was incredibly formative. Beverly was absolutely at the cutting edge of pollination biology, community ecology, and this is the environment from whom I learned. So for all of this, it was fun. It got to think a lot. Beverly was an influence. I loved bug camp. Time to do a PhD. It's like, okay, I'm ready. I can do this. And the question was, where am I going to go for a PhD? And then I came under the influence of this guy. John Waggy got his degree in this department in 1971. And John said to me, in these words, if you're any good, you'll go to Michigan. And I wanted to be good. I wanted him to think me good. So I went to Michigan, and Beverly left her husband and went to Michigan. So she had arrived at Michigan a few months before I did, and I became her first graduate student. So 1979, it was not EEB, it was the Division of Biological Sciences. A lot going on in the world. It was a very, very turbulent time. I, honest to say, did not pay very much attention to any of it, except for the Walkman. The Walkman was actually kind of important in my life. But that was our corner of the world, and it was all-consuming, and it had a huge influence on me. Now, this was all totally terrifying. I think Deborah was saying it was a scary time. Yeah, 1979. It was tough out there in EEB because it was a lot of smart people in the same place, talking intensely about science. I loved it, but I was terrified, right? It was smart faculty who weren't even talking to each other anymore because they were so passionate about their ideas. Grad students that were so much smarter than me. I mean, the whole Peter Grant group was terrifying. Stupid things came out of my mouth when I tried to talk, and everything seemed known already. So like, what was I gonna do? I was pretty tense. And Steve Frank, who ultimately was my office mate, said this to me. Steve was an undergrad at Michigan. He then went and got his master's at Florida and came back. And he said, Judy, I decided not to go to grad school because we were in a seminar together and you didn't smile once all year. I thought grad school must be awful. And Deborah will attest, I was pretty buttoned up. So how did I get from that point, or this terrified first-year grad student, to like where I am now? So I've been thinking about that a lot in the last several weeks. So I think it was these four things, and I'm just gonna go through these each one by one and tell you what I think maybe sort of happened. So the first thing was it helped me find the cutting edge. Well, what do I mean by that? I mean, it helped me find the big unanswered questions and thus what it was worth devoting my life to. In other words, anything you learn is new. It's like, fine. It's important to get that stuff, to know that stuff. But what we were all trying to do at Michigan was to find the big stuff. The places where, we had a passion and where we could actually answer big questions. And this was Beverly's mantra all the time. Whenever I would go to her and spout off about something, she'd look at me and say, but Judy, what's the big question? Right? It's like, What does this add up to? And that's now my lab logo is the Bronstein Laboratory. What's the question? So how did Michigan do that? You know, how did it help us find the cutting edge and make us who we are? Well, I think it was a bunch of things. The first thing was seminars and lab groups and courses. I took, along with, I think, many of you, Bill Hamilton's seminar over in Ruthven. Didn't understand a word of it, but it was very impressive. Had field experiences. I went with Beverly back to her study sites in Rhode Island for a couple of summers and got a lot of field experience in pollination biology. And then very intense, very challenging conversations. This was my doctoral prelims, definitely the low point of graduate school. That's Dick Alexander, the late, great Deborah Rabinowitz, and the late, great, well, they're all late and great, I guess now. So sad, Beverly Rathke, who's looking like she's laughing maniacally at something that just came out of my mouth. I was really stuck trying to figure out a dissertation project because I was so question and concept oriented, and I couldn't find a good one because I had great questions and I couldn't find a system that made any sense. I was really stuck. So what do? Get the heck out of town, right? So I went to the OTS course. I went to Costa Rica, and that's when everything changed for me. And a lot of it was due to these three guys, all of whom, of course, have Michigan connections. Find a system. It never occurred to me to start with the system and then change my questions. Like that, I didn't think that's what we were supposed to do, but that's what I did. Bill Hamilton had introduced me to figs and fig wasps in his courses. You guys, you know, many of you know him as the brilliant, amazing theoretician he was. I knew him because Bill was a field maniac and really, really knew his biology. And he loved figs and fig wasps, and he couldn't stop talking about it that year. Steve Hubble, who I met on the OTS course, and I said to Steve something I normally wouldn't do, which is, I have no idea what to study. This is what I'm interested in. Do you have any ideas? He said, have you ever thought about figs? And then he said, come down and work for me in Panama for the summer, and you can start working on figs. So I did that. So I found my system. And then Doug, this is obviously a very recent photo of Doug and me in Costa Rica. Doug was the one who planted a question in my mind. Actually, after I'd been working on the figs for a while, Doug planted the question in my mind that I've now been studying for the last 40 years. So I came back to Michigan. What happened next was I was on fire. I knew what I want to study. I was so excited. And the best thing that Michigan could do for me at that point was to get out of my way, honestly, and let me do it. So I had kind of defined an empty niche, which was sort of like, wow, we know almost nothing about mutualism as a whole. So I was inching towards this sort of general perspective on mutualism seen through the lens of this one extremely strange interaction. So I was thinking about things like that. How do mutualists find each other? I mean, this wasp is like the size of cosmic dust. How does it find fig trees? What are the costs and benefits of specialization, coevolution, cheating as a destabilizing force? And then the big idea that Doug planted in my mind, the idea that there must be conflicts of interest between mutualists, that interests are not aligned between partners. Brilliant insight, and really set me up. So now, you know, the EEB department really paid off. I had a great committee. This is Beverly, Deborah. I'm so sad not to be the first Deborah to show that photo. Herb Wagner, we've heard a lot about, and George Estabrook, who there seems to be no photo of George on the web. So I put a picture of me playing recorder because every week George and I and a couple of others would get together and play music. So they were interested in what I was doing, but they also just backed off. I must have been terrifying. And they gave me help when I needed it, but not too much. They made me a better writer, infinitely better writer, and I take writing super seriously, and I edit and things like that. And I put here, they built me up and calmed me down. This was the line I put in the acknowledgments of my dissertation. My committee taught me to have a better time doing better science. Better science, absolutely. Better time, well, there were a lot of dance parties they took me to, and Deborah knows. Yeah, they loosened me up a lot, and I was badly in need of that. So it got out of my way and let me start doing my thing. But of course, you never just let a grad student do their own thing. That is very dangerous. They kept me on a short leash, and they taught me how to argue. They taught me how to stand up for myself, how to express my ideas, and do it in a coherent way that was true to myself. And it's kind of hard to explain how important that was. Every Sunday morning, a lot of us would be at Nat Sci up on the 4th floor. I'd bring the Sunday New York Times, and we would pretend to work and do a little bit of work and talk science all the time. There were lab groups. I actually used to go to the group that Rick was talking about, the mammal and bird and herp group, because that's where some of the best discussions were happening in the department. Everybody had an open door. I mean, I would just wander and talk to Beverly, talk to Deborah. I was very scared. I went off to the ESA meeting and gave a talk, and I was very scared, but I stood up for myself with journal editors and fought back against some decisions. I interacted with speakers. Actually, when I came to interview here at Michigan, Dan Simberloff was giving the seminar that day. And I knew I had read a lot of Simberloff papers at that time, and it was like, wow, I can go somewhere where someone like this comes as a speaker. Some of the speakers were honestly remarkably rude to graduate students. I went out, things have gotten better, but I went out with one. Deborah and Beverly dragged me out with a very prominent plant ecologist who was speaking, and I, timid me, finally asked him a question and looked at him, and he looked at me and said, well, I suppose that would have a certain superficial interest. And then turned and went back to talking to them. So, there was a lot to overcome. But little by little, I learned to push back, and I became less timid, and I began to gain that level of self-confidence. I've thought a bit about the scientific milieu that kind of shaped me during those years, and I think there was a lot going on. There was a group of plant ecologists, so I was definitely influenced by plant ecology, pollination biology, community ecology. This is just a random assortment of names of people at Michigan who influenced me. And then there were all these evolutionary biologists. I wish I'd learned actually more from them. I didn't know what they were talking about a lot, but that was the milieu. And so it's not surprising that, like, if I was going to define who I am now, I'm an evolutionary ecologist who studies plant-animal interactions in community settings, right? And that's a way of picking and choosing all those themes. But it could have been otherwise. I was thinking when I was putting this preparation together, there could have been a whole different nexus of what had shaped me. There was a group of us in various ways who were studying mutualism, between species cooperation. And there was a big group of people that in one way or another were studying within group cooperation. Dave Queller, Richard Connor, Bill Hamilton, Steve Frank, Richard Alexander, all these people. Wouldn't it have been cool if we had all gotten together and started building a synthetic theory about cooperation that extended across the inter-specific divide? That's a divide that still marks our field. We could have worked to bridge it. I mean, it could have been the moment. It was not the moment. This did not happen. But it's really interesting that that's what happened and not that's what happened. I think the time wasn't right for that. So the last thing is that Michigan really encouraged me to teach, to mentor, to give back, to not stop at doing the biology. They modeled, my mentors, the other professors, the graduate students modeled for me what it meant to be a well-rounded academic. I was coming in actually with a lot of children's theater experience. So standing up and giving talks is like no problem whatsoever. And I built on that a lot in becoming an instructor. Every time I went to Costa Rica, I got voted onto something like being grad rep of the, to the faculty meeting. So I did a lot of that. And then Beverly Rathke absolutely modeled truly outstanding mentoring to me. She knew us each, she knew what each of us needed, and it was not the same among students. She gave us what we needed. She did not get in our way. She was vigilant, but she also gave us the freedom we needed, and that's something I've tried to model. So yeah, so I started out, kind of plant reproductive biology, very plant-oriented, moved into ecology and evolution of obligate pollination mutualisms, and then well, what happened next? Okay, what happened when I finished up? Well, 1986, I got my degree from here. 1987, I did a postdoc, a one-year postdoc in France. 1988, a one-year postdoc in Alberta. Landed in Arizona in 1989, and I'm one of the relatively rare faculty who's actually stayed in the same place their entire time. Arizona's an absolutely amazing environment and place to work. So, what happened next career-wise is I continued to work on mutualism, but the questions have expanded, and I'm super interested in conceptual development for the field as a whole, really building a body of theory and concepts that stretch across everything from the microbiome and the symbioses that Carla was just talking about, to pollination, to the mycorrhizae that Aimée was talking about, and so on. So that's kind of ultimately the goal. So my lab is probably the biggest in the world that's dedicated to the study of big questions about ecology and evolution of mutualism. It's such a young field that, many people trace their lineage to me and therefore to Michigan beyond me. This is in general what I think about. I mostly am a field biologist along with doing experiments. I've worked on collaboratively on a lot of theory, worked on a bunch of different systems, but with the general goal of understanding the rules underlying the mutualism structure and dynamics. I'm still very much involved in teaching and mentoring, and that's really the Michigan spirit. This is a page from one of my students, Paul Caradonna, who's a professor now, and he's written all over his students' paper, "What is the question?" which he picked up directly from me, which I picked up directly from Beverly. This is my current lab group. My newest graduate student, Nick, got his bachelor's degree here. And it's great to have that new infusion of Michigan in my lab. I do a ton of service for me. I love it. It's a passion. And actually, I know a lot of you through one or another of these connections. So, that I think is the stuff that Michigan did for me. It made me who I am. But it's not just me. I think it did it for all of us. It did it for all of us in our era, but I think it did it for all of us probably in all our eras. It helped us find our cutting edge. It got out of our way. It taught us how to argue respectfully with smart people, and it encouraged us to teach and to mentor and to give back. I think anniversaries are really important things. I mean, I loved that picture of, critiquing looking back. But you look back, you look forward, you try to figure out where you came from and where to go next. My department just had its 50th anniversary. It's because we were one of the first departments that was called an EEB department. That's why we're twice as old as you. But, and I was organizing it, and we had a small group of speakers talk about the future of the field. And when I looked over the photo, this was just last month, I realized that two of us were from Michigan. Of course, that's Michael Nachman, PhD 1990, and Marlene Zook, PhD 1988. And then there's me. So that's one department and how it's been so strongly influenced by this one. So I want to say thank you very much. This summer, I got the amazing opportunity to go back to the Michigan Biological Station, and I did indeed find the exact bracken patch that photo was taken in 1978. And I have to say, it was pretty hard to get down on my knees and back up, but it was an amazing opportunity to be back, and it really feels like coming full circle. So thanks a lot. And I finished early. See, Joan, I finished early.
Great, thank you.
Just grab mine.
Yes. I don't see who is introducing you. Maybe I'm self-introducing. Is KE here, anyone? Oh, there. Great. That is it. There you go. Cool.
Hi everyone, my name is Kei Feng. I'm a 6-year PhD student working with Dr. Stephen Smith on plant phylogenomics. And today, this is my pleasure to introduce Dr. John Freudenstein. He graduated with a bachelor's degree in botany in 1985 from Michigan. And then now he's a professor in the Department of Evolution, Ecology, and Organismal Biology at Ohio State. He's the former chair of the department, and he's the current director of the herbarium. Among many other awards, Dr. Freudenstein is an AAAS fellow. He is a highly respected specialist and field leader within plant systematics, orchid evolution, herbarium-based biodiversity science. Please join me and welcome Dr. Freudenstein.
Can I do this? Oh, we have to do that first, but then, yeah. Thank you very much. It's such a pleasure to be here. It's a real privilege and pleasure to be part of this recognition of this institution, this department that's meant so much to me, as well as obviously so many here. So my time at Michigan was very much an undergraduate experience. 1981 to 1985, we had a thing at that point sort of called the botany department, and those were the people that I mainly interacted with. When I think about what was most impactful for me, it really comes down to a few things. It's basically the people and what they did and their interactions, the places and the facilities that were part of the mission experience, and then some lessons that I can point out that I learned here that ended up being important and remain important to me even to this day. My first faculty connection was with Ed Voss. Ed Voss, as you know, was a big person in the herbarium for many years. Basically was hired to write the Michigan Flora, which was supposed to be, I think, a 5-year project. Turned into a career-long project, actually, for him, which benefited people like me because we were able to be involved in this work, actually. But my connection to Michigan and to Ann Arbor actually began a little bit earlier than that, because I was actually a high school student of this man, Fred Case, who was also a Michigan alum. He got his bachelor's degree and his master's here. He was an expert on the orchids of the Western Great Lakes region, an amazing gardener, and he transmitted that enthusiasm to his students. And so the first time I actually was in Ann Arbor this was in Saginaw. I'm from Saginaw. We came down, this must have been in my senior year, and we went to a Michigan Botanical Club meeting. And I was introduced to people like Ed Voss, Bill Benninghoff, who was the director of the botanical gardens at that time. So then in the fall I came down, I had my first days of class, and I thought I would do something the previous summer, a way that I could contribute and begin to work in this area that I thought was important and I was interested in, and that was to make a few plant collections. And so I looked at Dr. Voss's flora, which I already had, And we had distribution maps there. And I thought, well, where are some holes I can fill in? And so, okay, I'll do this. So I collected a few plants. Now, one thing I collected was Jack in the Pulpit, which you're probably all, or many of you, familiar with here. This is a common wildflower in northeastern part of this country. And I thought, well, I'm going to take the biggest one I could find. So I found one that was about 2.5 feet tall, 2.5 feet wide, and I pressed it flat in a sort of cardboard contraption I constructed, as well as with a few other things. And so came down to Ann Arbor. I was here this first week. I'll go talk to Dr. Voss. So I walk over. I was living in Mary Markley, which maybe doesn't exist anymore. So I brought these over and bring these in. Dr. Voss takes one look at this gigantic thing that I have and says, So snap, snap. He snaps the leaves and gets it down to a size that, oh yeah, it needs to fit on a herbarium sheet. That's what herbarium sheets look like. So that was my first lesson that was learned. And this is from your collection. This is my JVF number 1, actually. It is in your collection now. Still, I just pulled that image down the other day. So when I was here, I spent most of my time in the herbarium. And the herbarium was at that time, what was called the North University building. Now, I know you have a North University building now, but that's not the same one. There was a different one, basically under where we are standing right now, and we called it NUBS. And not only was the herbarium at NUBS, but also there was a computing center, English Language Institute, and the ROTC firing range. So you would be working in the herbarium, and suddenly you'd hear bang, 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 which of course, if any of us heard this today, would mean something very different to us, right? We would all be upset. But at that time, you knew, well, it's just the firing range and adjacent to the herbarium. While I was there, which was 40 years ago now, a little more, some of the members of the faculty from the previous generation were still there too. So people like William Randolph Taylor, phycologist, who was quite advanced in age and would come in with 2 canes and walk slowly down the hall of the herbarium. Alexander Smith, the mycologist, who did so much for our understanding of fungi, particularly basidiomycetes in North America. And then Rogers McVaugh, which was the only one of these that I really interacted with. Dr. McVaugh had worked in the flora, especially of Western Mexico, Nova Galiciana, and I actually was working on a project that I'll get to, and I consulted with him on that. So I had some connection with him, but these were some of those people who were still here at that time. Well, Ed Voss, because he was working on the Michigan flora, there was an opportunity for me to become involved with this as well. So he was working on volume 2. This is volume 2 of the Michigan flora of the 3. And so someone needed to assemble the plates that went into this. And so he had borrowed images, actually what we used to call photostats. People don't even know what those are today. From the New York Botanical Garden. It's a reproduction, basically. These were then cut out, and I glued them up into the plates that appear here. And somebody had to put these dots on the maps, too. And so I was also involved in this work. And so it gave me a sense of what's involved in publication. And of course, Ed Voss's, his sense of careful work is something that was transmitted to me as well. My first course, a seminar course, and this was not that common for undergraduates to do, was in nomenclature. And that's because Ed Voss was heavily involved in botanical nomenclature at this time. This was the Sydney Code, so the one that resulted from the Sydney Botanical Congress. And you can see his name here. He's the chairman of the nomenclature committee. And so we had a seminar on Monday evenings. It was in the herbarium. I was the only undergraduate. There were some graduate students. And we would go through the code and talk about the implications of those rules. And then because Ed Voss was also the head of this thing, he ended up getting the proofs of this. And so I ended up reading this. So I read the whole code, actually. I basically read it with him. He would read, I would read out loud, I would read, and then we'd reverse, trying to catch errors, which we did find. Okay. Another person I need to mention is Herb Wagner. I took Botany 422, which was systematic botany at that point, from Herb Wagner. I really loved this picture of him, sort of peeking out with this sort of impish grin from behind the ferns here. And as you may know, he was a fern expert. But he had a bigger impact than that. He was really a model as a teacher, and some of the I realize that when I'm teaching, I sometimes say, oh, this is something Herb Wagner would have done, a sort of mannerism or a way of communicating that you felt was something that was effective. One of the things we did in his class was an exercise, and we've heard about sort of rabid cladistics a bit today. Prosanta mentioned that earlier, but so Herb Wagner had created this set of diagrams of individuals called dendrogrammacea, these imaginary plants. And an exercise in the lab in systematic botany was to construct a phylogenetic tree using what was at that point called his ground plan divergence method, which was sort of like a parsimony construction of a tree, but he really had produced it as a teaching tool, although some people then went ahead and created publications using it. I also did what Herb Wagner called batriculating with him. So batriculating, so you may know, Herb Wagner was an expert on the genus Batricium. It's now been sort of separated. This is now Septridium, and these are still Batricium. But anyway, he would go out in the field, and sometimes I would go with him. And we would look for these plants. And then he'd get down on his knees and scrutinize them carefully. And there are people to this day, I think, who believe that some of those species still only he could tell apart, that he had named, actually. Anyway, this was, it was an interesting experience. The most interesting time that we were batriculating was one time in deer season, hunting season, when he thought, you know, we need to be a little extra careful here. So he insisted we drape ourselves with flagging tape as we walked through the woods, and he carried a bullhorn that he had set to a blasting sound. So here we were walking through the woods, draped in hunting tape, or flagging tape, and he was going, meep, meep, meep. I was kind of surprised we weren't just shot to get rid of us, basically. So if there were any hunters there. Obviously a memorable experience. I need to mention Tony Reznicek. Tony hasn't been talked about too much today. Tony arrived at the U of M shortly before I did, and has still an amazing understanding and appreciation for especially the native plants in North America. And at one point I was looking for an undergraduate thesis topic, something to do with Ed Voss, and so Tony suggested that I work on a particular problem in this genus Coralorhiza. This is Coralorhiza, the coral root orchids. They are one of about 40 separate innovations of leaflessness that has occurred in the orchid family. It's only a genus of about 12 species. It's not a huge genus. And what he noticed was that there was some variation in maculata. Some of them had a somewhat different lip morphology, flowered a little earlier. Others had a somewhat different there, flowered a little later. Anyway, this developed then into my thesis, my honors thesis with Ed Voss, based on herbarium specimens. We actually borrowed things for me to work with. And I did field work as well. This then led to my first talk at a meeting, which was the Michigan Academy of Sciences, which I don't remember where it was, but I remember going with Ed Voss there, and one of my first publications, actually, too. But the thing about Coralorhiza and this idea of a kind of through line of experiences sometimes is that somehow I'm still working on this genus. So this was my gateway basically into orchid systematics, and it's worked up to the subfamily level basically. I've worked across the Epidendroideae quite broadly. But when I went off to graduate school, which was Cornell, I ended up doing my dissertation on this genus. But we've come back to it time after time. Sometimes, well, one of my graduate students actually worked on it specifically. And in fact, one of my grand students, what I call my grand student, so his PhD student, just defended a thesis on this same group, a dissertation 2 weeks ago. So it's still trickling down. This was one of the first papers that we published that was an early early demonstration of changes in plastid genome in a leafless plant. And this is something we published 3 weeks ago. We've demonstrated this as the first pseudocopulatory orchid in North America, and we're still working on the chemical ecology of that. Need to speed up a bit. My first seminar here was to hear Harlan Banks, who came from Cornell. Banks was in the Rackham Auditorium, which I'd never been in before. Banks was an expert on Devonian land plants at Cornell, and Charles Beck, who was the paleobotanist here at that time, was in the audience, and Banks called him Mr. Progymnosperm. I didn't know what that meant. I didn't think it was because he was sort of an advocate for gymnosperms or anything like that. But I later found out that basically it was Charles Beck who realized there was an organic connection between these stem and leaf structures called Archaeopteris and this wood, which is called Calixylon, that those two things were the same, thus uniting gymnosperm wood with fern structures and giving rise to a sort of big burst in our knowledge of vascular plants. Can't not talk about the biological station here. Who wouldn't want to go and live in a place like this for part of the summer? Comes complete with a stick to keep the little window open. Well-dressed faculty. Somehow purses were like de rigueur for field work that summer. People started wearing their underwear on the outside, including poor David Gates, who was the director. And I just don't really know what's going on there. So anyway, all joking aside, my experience at the station was a very good one. It's great to be sort of submerged in that milieu for several weeks. Let me just hit a couple of course moments here. In genetics class, I first heard about the use of DNA in systematics. I knew I wanted to go into systematics and plants. It's like, oh, I think I want to use that, molecular systematics. Bill Benninghoff's phytogeography course. I first learned about cladistics and phonetics. The book we used didn't do a very good job of distinguishing those, but at least I knew the words. And then finally, Larry Nuden's Botany 488, his plant secondary compounds course. I learned about things like Ophrys orchid pseudocopulatory pollination, which it turns out I'm working on now, this kinds of thing. So why Michigan mattered to me? These things, the people, the places and the facilities, and it provided this great preparation for the next step. And really what it's about for an undergraduate, I think, is revealing the possibilities, revealing to them possibilities they never knew existed. And let me just finish here by saying, being at Ohio State, people will say, what are you? Are you a Wolverine or a Buckeye? It's like, well, one thing I can just point to is I live with this all the time. Basically, undergraduates before the Michigan game will go and put X's through any M's they can find on campus. Unfortunately, they do not go back and take these M's off. So they're up there all the time. So I'm reminded of this. But really what I am is a hybrid. I'm both at this point. I asked ChatGPT to come up with an image of a hybrid mascot. That's what it is. Thank you very much.
Thank you. Hello, everyone. My name is Joey Caldwell, and I'm a WASP enthusiast in the Tibbetts Lab, and that's why it's my great honor to introduce Dr. David Queller, who is also a WASP enthusiast. Dr. Queller comes from Washington University, where he is a professor and is one of the most highly regarded evolutionary biologists in the field today. He's also a member of the National Academy of Sciences. Now, I don't know Dr. Queller personally. In fact, I just met him this morning. But he has been unknowingly communicating to me through his prolific work on social behavior. And he also did his PhD on milkweed at the E.S. George Reserve, which is where I work in the summers. So not only do I stand on his giant shoulders in the literature, I also walk in his giant footprints every day at work. So join me in welcoming Dr. David Queller.
thank you. It's great to be here for the 25th anniversary of the department, and even greater for me because I've got an anniversary too. It's 50 years since I arrived as a graduate student at the University of Michigan. And at that time, I'm afraid I didn't really know what I was doing very well. We talk about chance and necessity in biology, and my choice of graduate school, I had my reasons for coming to Michigan, but I'm listening to all of you, and you had great reasons. Mine were kind of, so-so. I flipped a coin, sort of, and came here, could have gone somewhere else. But the chance played a role, and it turned out to be an extremely fortunate choice for me. It turned out to be the perfect place in a variety of ways. Now, I didn't take a lot of pictures in those days, and so I'm not going to do the PowerPoint thing here. It's just going to be, me and you, and this piece of paper here. You can all read that, right? Instead of the PowerPoint. That's just a crib sheet for me. This is how we used to teach, right? Before PowerPoint, we had to have notes and kind of glance down at them. So I'm going to go with that. The first thing that happened, one of the first things that happened to me when I got to Michigan, and I wasn't quite sure what I wanted to do, but certainly evolution, maybe social behavior. But you met with a small committee of faculty who were supposed to advise you on your coursework. I don't remember everybody that was on the committee, but I remember John Vandermeer was on the committee. And they listened to my interests and what I'd taken before and said, you should take Dick Alexander's course, which was a little bit surprising to me because the year before, E.O. Wilson had published this sociobiology book and had ignited a firestorm, and Science for the People was on the other side of that. So anyway, thank you, John. It was important for me. Probably would have taken it anyway, but there you go. It was an eye-opening course for me and for many other people too. The way he taught the course, he gave us lots of literature readings. It might be something that was published the month before. Now that's not so uncommon for a graduate course, but this was a big course that was more undergraduates than graduates. And he told us, both in the syllabus and throughout the course, that we were supposed to read these things. Not only that, we were supposed to understand every word of the things we read, or at least understand why we didn't understand it. And that was an impossible task, pretty much. But one thing I didn't realize until I was kind of putting this talk together is that he modeled that for us. That's how he did his lectures. He didn't get up there and present science as a done thing. He got up there and ruminated, basically. He'd grab onto an idea, he'd chew it over, swallow it down, move on to something else for a while, and then cough it back up again and chew it over some more. And we got to see the process of him thinking about these things, debating with himself, debating with sometimes his adversaries who weren't there to defend their points. One case I remember in particular was Trivers had come out with a couple papers on parent-offspring conflict, and at that time Dick Alexander didn't buy it, didn't think it would work. So one of Dick's arguments was, okay, you do something as an offspring that's contrary to your parents' interests, okay, you could get a benefit from that, but then you grow up and your offspring do things contrary to your interests. And so he thought it couldn't work. And there was a flaw in that. I was not able to point it out to him, although I thought Trivers was right. A different graduate student convinced him of that fairly quickly. But you got to see how he was thinking about these things. Now, initially I was just in his class. I didn't really know him. And my first contact, Doug had a story of first contact with mentor. This one's even a little bit better. After class one day, he asked me to come into his office the next day, wanted to talk to me about something. So I did that. I couldn't imagine what it was. I didn't really know him yet. And I sat down and he started asking me about one of the essays I'd written for the course. And he never really said this, but I eventually figured out he was trying to figure out if I'd plagiarized it. And so that was my first contact with the person who was going to be my advisor. I think I convinced him because pretty soon after that he offered to be my advisor if I wanted to do that. So that was one bit of luck. I knew Dick Alexander was here at Michigan, but I didn't know that much about him, and he turned out to be a very inspiring mentor. Next stroke of luck, something I couldn't have known about, was that, as I understand it, when Don Tinkle took over directorship of the museum, he negotiated for his former salary to go to the purpose of bringing in guest faculty members. And so I arrived at Michigan. I discovered John Maynard Smith was going to be there for the semester teaching a seminar. John Maynard Smith, who was foundational to so many things, had just published a book on applying game theory to evolution and so forth. So I got to take that seminar and begin learning some mathematical biology as well. And then in the second year, the fall of the second year, it was Bill Hamilton. He wasn't at Michigan yet as a professor. We brought him in as a guest professor, and I got to take his seminar. The following year after that, it was George Williams. So George Williams was the guy who sort of set in motion the whole, you could say sociobiology, the idea of thinking more critically about how social evolution evolves. So that was just kind of unbelievable luck. I got exposed to all of those people in a way that I could not have imagined. There was more beyond that as well. Tinkle and Alexander organized a symposium in my second or third year in which they brought people working on social behavior from all over the country to give talks. And Joan mentioned that Dick never seemed to value her work, but he invited her to the symposium and put her in a prominent position. The very first session in the symposium was on social insects, and it had Mary Jane West Eberhard and Katie Noonan and Joan Strassman, and they all just knocked it out of the park. And they were followed by Bob Trivers, who kind of mumbled around a little bit, and then later accused Dick of having set him up by putting him behind all these forceful women. So I didn't actually meet Joan at that time. That would come later, but I had encountered her work. There was also a guest ecology course in my first year where people came in for 2 weeks at a time and gave talks. Peter Grant was one of them. I don't remember all of them, but one of them was where I for the very first time saw that little graph of CO2 on Mauna Loa ticking up every year, which defines what a lot of people do in the department these days. Another amazing bit of luck was just the graduate community here. You've heard some people talk about it. Overlap partly with Trevor Price and Judy Bronstein, for example. Actually, another of the first things that happened when I got here, you get assigned an office. I had an office mate. I walk in there and it's Nancy Moran. And we were both kind of quiet people. It took me a while to figure out how smart she was, but she really was and has gone on to an amazing career. She eventually worked with Bill Hamilton, moved over to the museum. I stayed in the Natural Science Building and moved me up to the third floor, I think it was, where in just one hallway, for a while at least, we had not just Judy and Trevor, the rest of the Finch unit, Dolph and Lyle, for example. We also had Steve Frank and Bernie Crespi and Marlene Zuck. It was just an amazing group of people. And if you couldn't learn a thing or two talking to those people, you weren't going to learn a thing or two, ever. So fantastic luck all around. Now, as far as research, it was difficult finding a thesis project again. We were kind of left to our own devices to a certain extent. The first summer, I tried a project on Polistes wasps. Didn't work out too well, plus you get stung. I got stung 6 times that summer, and actually only one of them was when I was doing my research. The other 5 were just random. But it did, between that and the project not working all that well, it set me to thinking about trying to find something that would work better and had a less evil organism involved. And I started to think about plants and whether there was some room for plant sociobiology of some sort. And so for my second year qualifying exam, the way they did it in those days was to have you write a review paper on a topic of your choice, and then they'd grill you on it. I chose to write about the evolution of breeding systems in plants, monoecine, diecene, so forth. It was really hard. Maybe I put too much pressure on myself, but I was taking courses, not having enough time for this. I got an extension on it. So I really had to finish in that week of the extension, and in those days we couldn't cut and paste. We didn't have autocorrect. What I had instead was a manual typewriter. And some of you don't know how that works. You sit there and you go tick, 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 and then damn, and you roll it up and you white it out, roll it back down, tick, 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 damn. And so that last week between trying to put the ideas together in my mind and trying to get them actually down on paper properly, I think I got 11 hours of sleep in the 5 days before it was due. But I got it done, and it went okay in the end. Then in terms of coming up with a thesis project, a little bit of luck again. Part of it came from listening to somebody else's thesis defense. There was a student named Forrest Hartman who had worked on a milkweed, Asclepias syriaca, questions of why they produce so many flowers, why they abort so many of their fruits. And he had this 600-page dissertation. He'd been there for 10 years, I think. And showed some very interesting things, but was missing something that I thought might be the key, which was that the floral display may be largely for the male function. You need to have the flowers out there in order to get your pollen dispersed to other plants. And a smaller display would suffice easily for getting enough pollination for the female function to raise as many seeds as they could. And so that's half of what my thesis was, is working on that question, which I did out at the George Reserve, as was mentioned. I put together a hybrid thesis committee, the likes of which hadn't been seen before and probably won't be seen again. On the social behavior side, I had Dick Alexander as one of my thesis co-chairs, and then I had Bill Hamilton. And on the plant side, I had Beverly Rathke as my other chair. She had worked on milkweeds previously, and also Warren Wagner. So the milkweed stuff turned out pretty well. I did a lot of work at the George Reserve, and it turned out my hypothesis came through pretty well. That doesn't always work. That time it did. The other piece of my thesis came out of thinking about the question I brought up earlier, parent-offspring conflict. And I thought, how does that work in plants, where, especially flowering plants, where you've got not just the parent and offspring, but you've got the haploid gametophyte and the triploid endosperm. And so I kind of worked out the kin selection consequences of those. How were each of these tissues related to our own embryo to get resources for, and how were they related to other embryos on the same plant? And I think yielded some insights into the evolution of the endosperm that have, I think, stood the test of time. So that was the thesis procedure, and went nicely, I think. I didn't actually stick with plants that long, although I've touched back on them once in a while. What Michigan really gave me, and gave us, I think, was an ability to think about things, to generate new ideas, to move on to something else if it seems profitable. So for a postdoc, I got a postdoc at the University of Sussex with Brian and Deborah Charlesworth, but Maynard Smith was also there. I began developing more of my theoretical skills, that I'd learned something from Maynard Smith, something from Hamilton, a bit from Julian Adams in his population genetics course. And so I have a theoretical line that's continued to the present. And then got a temporary faculty position, as Joan, initially temporary faculty position, as Joan mentioned, at Rice University. And she drew me back to the evil wasps. And we spent about 15 years on the evil wasps, working on kin selection questions. And then she's presented this already. We're attracted to the prospects of working on social amoebas, which the questions were very much the same, or at least very related to the ones we'd done before, but the techniques were very different. I mean, we were running a microbiology lab that's doing genomics, and it's doing cloning, and it's doing phylogenetics, all these things that I never would have even thought of doing way back in graduate school. I think since I have a minute left, I think I'll end with another short anecdote. Another person that came to visit, although only for one lecture, was Sewell Wright. And Sewell Wright had been there since the beginning of genetics, essentially, and he was over 90 at the time, I think, and he filled a large auditorium, I think it was the Rackham Auditorium. And a couple of things I remember about it. One was that it was not a scintillating lecture. He was over 90, and he talked in a low voice. He had a lot of picture grabs from his 4-volume masterpiece on population genetics. And then he went on and on. You get to 5 o'clock, and normally people start dribbling out. He got to 5:10, he got to 5:20, he got to 5:30, maybe even 5:40. And huge auditorium, almost nobody left. It was just an amazing experience. It was just out of respect for the man. And I know I'm no Sewell Wright, so I'm going to quit right now and stop there. Thanks.
Hello, my name is Diana Medellin. I'm a PhD student in Chris Dick and Stephen Smith's labs. I study the biogeography, evolution, and chemical ecology of tropical plants. And today it's my pleasure to introduce Dr. Chuck Davis. Chuck graduated from Michigan with a bachelor's degree in science in 1997. And then he graduated from his master's and doctoral degree from Harvard. And now he is a professor of organismic and evolutionary biology and curator of vascular plants at the Harvard University Herbarium. Among many other awards, Chuck was elected as a Michigan Fellow in 2002. And he has working extensively by highlighting the incredible value and importance of collections of natural history for research, applied research. So please join me welcoming Dr. Chuck Davis.
Thank you. Thanks, everyone. It's just a tremendous joy and incredibly nostalgic for me to be here with this incredible group of people. So I thought about this reflection and what I would present, and for me, the Michigan experience wasn't about the credentials, the degrees, wasn't even really about the department, honestly. It was about the place, of course. I think many of us love Ann Arbor, love the university itself in a whole variety of ways. But what really rooted me here were the incredible mentors that I shared time with as an undergraduate. And it was also the museums and the collections that were part of my experience. And the combination of those two things shaped what I call a plant vision, an ability to see, to recognize, and to distinguish plants in our world. And that really set off my trajectory in both research and teaching. And so earlier this week, I sent Nate on a scavenger hunt to find the Herb Wagner Memorial Ginkgo, which I remember more or less when it was planted, and it was like that big in the early 2000s. But there was an inscription at the bottom of it. And I told Nate it was about throwing a rock into water, and the ripples go everywhere, and they're never ending. And boy, was I wrong, because it's sort of like that, but it's actually a teacher affects eternity. They can never tell where their influence stops. But you'll forgive me that I'm going to use the ripple and the stone in the water as the metaphor for this talk, okay? So that's the thread that I'm going to weave through this. So this is a thought exercise and a little bit of personal amusement, but it's the first slide that I give to all three courses that I teach at Harvard. So these are to first-year students, to intro concentrators, and even to graduate students. And I ask them, what do you notice first? And I want like first impressions, first instinct. And invariably, the majority of students say the lions, the animals. And almost never do they point out the magnificent sausage tree with this incredible branching and these amazing fruits, which you can see up near the top. They're like a foot long. And so they miss the plants. And of course, that begins another series of questions about how most of us operate and move through the world. We don't see plants, despite them being, forming the terrestrial matrix of life on Earth and separating us from starvation. And that kind of attentiveness to plants, that ability to see plants, was what I got here at Michigan, and what I try to bring to bear in both my research and teaching life. So I'm from Michigan. I grew up in Flint, just north of here, and that is my mother on the left in the first row, 3 from the left. And this is in the mid-'80s, and my mother was a student of the ornithology class at the University of Michigan Biological Station. So she was seeking professional development credit for, she was a teacher in the Flint Public Community Schools, a biology teacher. And I was sort of a tag-along. I hung out at the camp, at the biological station. I was what we called at the time a camp kid. And this was just a completely and totally intoxicating experience. Nature, biodiversity, and people having an incredible time. And I would later, in the mid-90s, go as a student. And so my mother and I would share the same instructor. That's Francie Cuthbert. And to this day, we bird together, and there's just a lot of joy that we share around this experience. But my connection with the University of Michigan happened very early and during very formative years when I was like 10 years old, okay? And then I made it to Michigan, where the ripples really began, okay? And it began with these mentors. Again, they were really the greatest gift to me, and there were 4 that were central. John mentioned Ed Voss, and he was, of course, wrote the Michigan Flora. He had meticulous attention to detail, okay, just extraordinary. Bert Barnes loved plants, could identify them, he saw them, but he also saw them in this broader ecological context, right? So a slightly different perspective, but an important one. Herb just brought enormous amounts of enthusiasm, excitement, and passion for absolutely everything he did. And Bill Anderson, who I would ultimately spend the most time with, would publish papers with for the next 20 plus years, really kind of made me aware of field work and herbarium practice and herbarium science in a way that I just, had never knew was possible. Right? And so these were the incredible people that shaped this plant vision. They weren't always easy people, as some of you in the audience that may know them, but they were truly incredible and lifelong friends in all these cases, and mentors as well. Now, they're all past. Their legacy remains, but there were a few others that are still alive and were also here and formative for me, including Tony Rezicek, easily one of the top field botanists in all of North America, probably in top 5 easy. Melanie Gunn, who was my teaching fellow in woody plants, which I took with Bert Barnes and Herb Wagner, and Chris Anderson, who I saw just the other day, and we're actually working on finalizing a manuscript together. So that kind of legacy continues. Now at the biostation, I took courses in birds and insects, and I was very much animal-centric. But I ended up taking intro biology with Bill Anderson, and I kind of moved into plants fairly quickly, And after leaving the station, I really wanted a field experience, something beyond what the biostation offered. And Bill lined up an arrangement through John Beeman, who was at Michigan State at the time. And at 19 years of age, I was flying off to northern Borneo, leading expeditions, collecting on the north side of this mountain, hiring tree climbers and guides and cooks. It was extraordinary. It was also trial by fire. I was literally living in the field for weeks on end, and sending out specimens to Kew and to other places, and truly extraordinary. This was also right around the time that email was kind of emerging. So Bill and I had arranged a plan that I would have additional coursework that was on the books, as it were, and it was sort of a floristic family level treatment of the plants of Kinabalu. Now this is an area, it's the highest mountain between the Himalayas and mountains in New Guinea. It has just astonishing diversity. So I was kind of capturing a flora of the family, learning many of these families for the first time, and just having a tremendous amount of fun, okay? And one of those plants that I would see just outside of the Kinabalu Park boundary was that plant on the right. This is a Rafflesia. This is Rafflesia pricei. It's one of the world's largest flowers. They can be upwards of 3 feet in diameter, look and smell like rotting flesh, and they're holoparasitic. So they have no stems, no leaves, and no roots. It is our best case of mega charismatic flora, okay, as it were. And it was extraordinary to see this plant. But I had no idea that 10 years later would kick off a 25-year research program that focused all around Rafflesia. I mean, and there was really in many ways serendipitous, but I started working on phylogenetics and phylogenomics. Lo and behold, we published the first case of host-to-parasite gene transfer in plants, facilitated through the intimacy of host-parasite connections. We then placed them more thoughtfully phylogenetically so we can infer the astonishing increase in floral size, and that led to floral developmental genetics to characterize the homologies and indeed to think about, at the very reductionist level, how these flowers are constructed. Not too long ago, we published the first genome of these things. They have really difficult genomes, and they've lost half of their genes, along with their stems, roots, and leaves. We also use that genome to take those horizontally transferred genes and use them as what I call the ghost of horizontal gene transfers past. So to really look over tens of millions of years at host shifts and to use those genes that were transferred to actually trace those host shifts. And then finally, to look at convergent trends and what may be motivating these independent instances in increase in size. And so here I am. This is in January. I noticed my Michigan hat there, by the way. This was I was back in Sarawak, and we're working with a postdoc on pollination, on fragrances that are produced by these plants, and also on the microbial diversity. So stay tuned for more, but that work continues, and I cannot stress how I would have never imagined that I would actually be working on this group of plants that I saw there in northern Borneo. So the other thing outside of the mentorship were the museums, and especially the herbarium. And the herbarium, as John mentioned, was kind of right here-ish, slightly lower. I don't remember the gunshots, but it opened my world and my eyes to a kind of science that I've never looked back on. And so, which is to say, like, I've spent 30 years in an herbarium. When I was here as an undergrad, I spent years in the herbarium. 7 days a week. On Saturday mornings, it was the Met Opera broadcast, okay? And it's also where I learned to love opera, I guess. But this is just so kind of astonishing at how central these are to initially the more classical work that I've done, but herbaria and natural history museums more generally are entering into this kind of Renaissance period. And so that's due to kind of the digitization of these collections, and so scientists around the world are actively curating what I call the global metaherbarium, this interconnected, digital, virtually accessible herbarium for scaling science in ways that we never thought possible. But beyond that, we're applying all kinds of approaches that were never even imagined when these things were collected. So they range from eDNA to a whole host of multi-omic investigations, even de-extinction. So now when I give tours and I talk about herbaria, I think way more about the possibilities in these collections that we haven't even imagined yet. Makes it all the more imperative that we become excellent stewards of these collections. And so I found a group. I fell in love with a group, as many of us do, who hold a systematist card. And in this case, it was the Barbados cherries, the Malpighiaceae, and herbaria made that research possible in a whole variety of ways. But I think what I've learned more out of this than anything else is that it's good to be rooted in a clade, in a group of organisms that you spend a lifetime studying. And for me, this is kind of a model clade. All right, so we've learned studies on taxonomy, on evolutionary ecology of plant pollinary mutualisms, and on global biogeography. And finally, just a plug to say museums and herbaria matter more than ever today. Again, they just continue to give back, right? They preserve the past, but they make new science possible. And this is just a scattering of work, some of which I wouldn't have even imagined doing 5 years ago, let alone 30 years ago. So characterizing bias to get at better models of biodiversity. These are not perfect records of nature by any means. This is a paper we worked on to characterize the colonial legacy of herbaria. Last 15 years, I've worked on macroecology using herbaria, so doing large-scale phenological investigations across huge areas and large numbers of taxa. I already mentioned kind of some of the genomic investigations to build large trees to infer biogeography. And one recent thing that's been just a totally awesome and fun shot in the arm, I was a lead scientific curator for this Dutch retrospective on a woman named Rachel Reich. She painted incredible botanical still lifes in the 16 and 1700s. And basically, we did detailed floristic inventories of these. They'd never been done. We put them on a map, and they retrace the Dutch colonial empire, the kind of human ecologies underlying these. And we put them in proximity with herbarium collections and other collections in the Boston MFA and the Toledo Museum of Art, and also in Munich. It was just really a fun and wonderful experience for me, and also a way to highlight that art is an unexplored source of biodiversity data. So I didn't really know how to finish, but I woke up this morning and I thought, I'll continue with the ripples. I teach on alternate years Ed Voss's old flora course at the field station. I still have those Michigan roots. I still have family and friends here, obviously. But I just want to say that the thing that most moves me during a semester is they start with no knowledge of the green world around us. And if we do things right and the student is focused, they really, by midstream, they start to really make sense of that green world around them. And as an educator, it is one of the most astonishing and wonderful things to experience in a biodiversity-oriented class. And that's the foundation, the springboard, for so many other questions that can be addressed and answered. So thank you, Michigan, and thank you all.
Oh, yeah. All right. So this has been a wonderful day. We've heard a lot about the history of ecology and evolution at the University of Michigan. The next two talks are really going to highlight the future of ecology and evolution at the University of Michigan. So the last two talks of the day sort of launch us forward into celebrating the next 25 years of what's to come. So we're going to hear from a soon-to-be graduate student and a very recent grad student who's now a postdoc. First is Rosie Gloss. Rosie is a student in the Weber Lab and is rapidly becoming a world's expert in plant-insect interactions and has an incredible eye for detail and art as well as science. So Rosie, take it away.
Okay. Wonderful. Thank you so much, Nate, and thank you, everyone. It's a huge honor to be speaking to you all today after such an impressive lineup. Like Nate said, I am relatively early in my career, so I don't have the capacity for the grand retrospectives that we've seen today. So instead, I'm going to zoom in, and I'm going to share with you just a small vignette from my work here at the University of Michigan. And while I'm at it, I'm going to tie it with a little bit of EEB history. So Thais helped me out here today. She introduced you all to the women that you see in this slide. And I'm going to tell you just a little bit more about their research and their trip because it has quite a bit of bearing on my own research and experience here at the University of Michigan as well. So here is Elzada Clover, whom you already met, and her student Lois Jotter. And about 88 years ago, in 1938, they were the first non-Indigenous women to take a full trip down the Grand Canyon along the Colorado River. At the time, Elzada was 42 and a recent PhD graduate of the then botany department here. And Lois was her 24-year-old mentee. And despite being expected to do all the cooking and cleaning and logistics of this trip, and despite the international and national press trivializing or really ignoring the scientific mission of their expedition, And despite the fact that the University of Michigan itself did very little to sponsor the trip, and despite the fact that Elzada, though a recent PhD graduate of the department, had recently been denied a position at botany because of her gender, they nonetheless succeeded in performing an incredible floristic survey of the Colorado River. And Elzada did eventually get that professorship, becoming the first female professor in botany at the university. But this also has some personal relevance to me because unbeknownst to me at the time, 83 years after they did it, I got to do it. I wish I had known about their trip, but I did not at the time. But just like Elzada and Lois, I became absolutely fascinated by the geology, the natural history, and especially the botany of the Colorado River and of the Grand Canyon. And in particular, I fell in love with this plant right here. This is the desert stingbush, also called Euchnidiourens, a member of the plant family Loasaceae. And I had a background in field botany, in plant anatomy and systematics, and so whenever I was outside, I just wanted to look at plants as closely and as much as I possibly could. And one of the wonderful things you see when you look more closely at this particular plant are its hairs, or its trichomes. They are these fabulous, elaborate little barbs that look like medieval weapons. And they led me to love this plant. They led my trip mates to hate this plant. Because I'll spare you what they called it and still call it to this day. Because all those little barbs make it sticky like Velcro. And so if you're trying to take a hike, it sticks to your pants. If you're trying to climb, it sticks to your ropes. At one point, one of my friends threw a pile of climbing ropes down a gully and just straight onto one of these plants. And it was covered in trichomes for the rest of the trip. But I love and loved this plant and was even more fascinated when I found that it was covered in all manner of insects that had landed on the plant. And something I forgot to tell you about this trip, actually, is that it ended up being really important for my graduate career in two reasons. The first reason is that it almost prevented me from getting into grad school. Because unlike Elzada and Lois, who took their trip in the summer, I took my trip in the winter. I went in February. And as some of you might know, February is also usually when grad schools do their interviews for admission. And so before I left for this trip, I applied to 3 grad schools and then proceeded to miss the interview weekends for every single one of them because I was out of cell service in the Grand Canyon. And, but lucky for me, there was one institution that agreed to reschedule my graduate interviews. And that was not the University of Michigan. It was Michigan State. Don't boo, please. But when I finished this trip, I still didn't know if I'd gotten into grad school, but after 32 days on the river, I got in a truck, got out of the canyon, got cell service again, checked. I'd gotten into grad school. And while I was on the river, I had decided that if I got in, I was going to try to understand more about these incredible structures and the plants that bore them. And so I have spent the last 5 or so years basically trying to understand the interplay between species interactions and microscopic or otherwise hidden phenotypes, with an emphasis on interactions between plants and arthropods. And so I've spent quite a bit of time trying to understand those trichomes, and looking at functional and diversity in trichome phenotypes and their macroevolution across, in particular, the Loasaceae, but also more recently in domatium-bearing woody plants. This has led me to try to understand how trichomes interact with other plant defense traits, such as chemistry. That's what I'll talk a bit about today. And along the way, I've also been using the Loasaceae as a model to understand variability in species interactions, mediated not just by trichomes, but also some other traits, including pericarpial nectar. But I only have less than 10 minutes, so I'm just going to talk a little bit about trait-trait interactions. Normally, this is where I continue barreling forward talking about plants, but I understand that I'm in mixed company. There are some plant biologists here. There are also some people who don't study plants. So I'm going to start with something a little closer to home and a little, fuzzier. Hopefully everyone here recognizes this animal. Yes, this is a wolverine. if you decided to attack a wolverine, which I don't recommend, you would have to contend with two things. First, bone-crushing teeth. Second, sharp, curved claws. And if you wanted to understand what would happen to you when you attacked this wolverine, you would definitely want to take both of those things into consideration. Because for all you know, it's going to hold you down with the claws and bite you with the teeth. And with plants, it's really kind of the same thing. A plant, when defending itself against a potential herbivore, typically wields a wide array of different defenses simultaneously. So if we go back to that sting bush, for example, it kind of has some teeth. It has these incredible pokey needle-like hairs. It has some claws. It has these barbed trichomes that made it so sticky and made my friends hate it so much. And then it has also kind of a third, not so secret weapon, which is the defensive chemistry in its leaves. And so as people who study plant defense, we spend a lot of time characterizing these traits and studying their effects on herbivores. But a lot of people, long ago, far smarter than me, started to think about how traits might not just act in isolation, but also how they interact against an herbivore. Because an herbivore is never really encountering just one of these traits at once. Just as you, when encountering that wolverine, are not going to just think about its claws or just think about its teeth. You're going to think about the interaction between those two traits when it bites you and claws you. Same thing with this plant, and spoiler alert, it doesn't go so well for the herbivores that try to eat it. And so what has led me to wonder is, can I use this sting bush as a model to test what is now a pretty long-standing question in plant defense, which is, can two anti-herbivore strategies, in this case defensive chemistry and those leaf hairs, interact against herbivores? And I will admit, the desert sting bush is kind of an obscure plant. But these two defensive strategies, trichomes and chemistry, are some of the most widely used defensive traits across plants. And so we can use this plant to understand what might be going on in a lot more species. And so I have a couple minutes to tell you about one of the experiments I did to try to get at this. This is a pretty basic, straightforward factorial diet assay, where we fed, in this case, a generalist herbivore, the beet armyworm, diets containing either one of those defense categories or both, or neither. And we tracked their growth and their survival over the course of their lifetimes to try to understand whether we see a separate additive or synergistic effect of these defenses. It's worth peeking behind the hood real quick to just understand how I isolated these defenses, because I think it's a nice example of the absurd things that we do in grad school sometimes. So I dried a whole bunch of sting bush leaves on silica, and then I used a very high-tech piece of scientific equipment called an electric toothbrush to remove trichomes from the dried leaves and then collect them. This is my precious falcon tube of trichomes. And then I ground those same leaves finely and extracted the secondary metabolites from those leaves to use as my chemical defense in the experiment. Here's an example of the experiment in progress with a whole bunch of tiny little first instar beet armyworm caterpillars starting to munch away. We learned a lot about how this plant defends itself, but today I could pretty much show you one plot. And this is a plot of the growth of each of those cohorts of caterpillars over the course of the experiment up until day 12 when they reached their max mass, along with some pictures of those caterpillars at each stage. So first I'll show the data. You can start thinking about who might be who here. At the top, we've got the controls. These caterpillars looked pretty good, as would be expected since they were on kind of like the Cadillac of diets with no plant defenses in them. The group that got trichomes in their diet did grow a little more slowly and were a little smaller, but not significantly so. And then there's a big jump down for the group that got the leaf chemistry. So clearly whatever's in there, not good for a beet armyworm. But then the most extraordinary effect was those caterpillars that got both trichomes and leaf extract in their diet. And so this was tantalizing evidence that there may be a synergism between these two common categories of plant defense. Sure enough, when you model it we find that the caterpillars that experienced both of the plant defenses at once, as they would when they were actually eating a leaf, were about 75% smaller than expected at their peak mass, had those two defenses been acting only additively. And so this was exciting empirical evidence for something that has been hypothesized for some time. I have all of 3 minutes to tell you a little bit about some of the mechanisms that may be contributing to this synergy. And so first off, I'd like to talk a bit about what's actually in there. And for this, I have a shout out to my lab mate, Abby Soul, who helped move the chemistry. Because naturally, if we're throwing a whole bunch of leaf extracts at a bunch of caterpillars, we want to know what might actually be in there. And so consistent with previous work on this family, about 1/4 of the compounds present in those extracts were iridoids or iridoglycosides, which are a well-known category of plant defense chemicals. In the case of this particular herbivore, they are known to deter feeding, and they're also known in other herbivores to reduce digestive efficiency. So 2 things that you would expect could contribute to those reduced masses that we saw. And then at the same time, they're contending with those trichomes, which are kind of not your ordinary trichome in some ways, because they are fortified with a whole array of biominerals, including calcium carbonate, silica, and calcium phosphate. And these are the things you find in like mollusk shells and your teeth and sand. So all things you can imagine would not be very fun to chew on as a caterpillar, particularly when you consider the size of these trichomes relative to the size of like a tiny first instar caterpillar. And so curious about whether or not those trichomes might actually be causing physical damage to the caterpillars, we looked at their mandibles. So that's like the part of the caterpillar that is first chomping down on the trichomes and might have to experience them. Here's some images of control mandibles. And then if we compare that to mandibles that were eating trichomes, I apologize for the quality of that image, we can see that not only is there wear to the mandibles, there's just like straight up breakage. And so these poor caterpillars were trying to chomp down. I kind of imagine it like if someone just handed you a wine glass and told you to try to eat it. It doesn't go well. And they had to do that in order to get through the diet. And when you quantify that, there was a pretty noticeable difference in damage between the control and the treatment mandibles. There's also some tantalizing literature looking at how mechanical defenses in plants can potentially make chemical defenses more effective, leading to synergisms. Kano et al. Termed this the needle effect when they looked at it with raphides and cysteine proteases. I wasn't able to examine the guts or interior of the caterpillars in my study, but I was able to look at their frass, at their poop. And if you compare the frass of a control caterpillar to frass of a caterpillar who was eating trichomes, you can see that although they are chewing on them, they are also passing whole trichomes through their digestive systems. And so you can imagine that any herbivore encountering this plant is having those things go through them, which cannot be a pleasant experience and may well be contributing to the synergy that we observed. So in conclusion, it looks like these 2 common categories of plant defense can act synergistically. It's important to note that I used a naive generalist in my experiments, and so it was particularly susceptible to the defenses that I was studying. And so it's hypothesized that these synergisms I saw might be particularly important in the case of specialists, which may have evolved some tolerance to the defenses. So I'm interested in understanding how common these defensive synergisms are, and in particular, how specialists differ from generalists in their response. That's all for me. Thank you to everyone here and to Nate for having me, and the wonderful mentors on this slide. Thank you so much.
There you are. Great.
Hello.
Our final speaker today is Silian Song. He received his PhD from the University of Michigan last year, and his dissertation won Dissertation of the Year across all campus, not just in biology. Pause for applause. Yeah. But we all know no one reads dissertations, except for the judges in this panel. So look for his papers in journals like Nature, Ecology and Evolution, Evolution, PNAS, among many other outlets. So let's hear about some of that work now.
Thank you very much. Okay, thank you very much, Nate, for your introduction. It's a great honor for me to be here to introduce my work in this very special symposium. So the title of my talk is Adaptive Tracking with Antagonistic Pleiotropy Generates Seemingly Neutral Molecular Evolution. So the question is, what drives molecular evolution? This is probably the most important question in the field of molecular evolution and population genetics. And people divided on two camps on this question. Selectionists who believe that most of the substitutions and polymorphisms are due to selection, while neutralists believe that most of substitutions and polymorphisms are due to genetic drift. Well, Motokimura firstly proposed the neutral theory of molecular evolution in 1960s. It immediately triggers a very heated but long-lasting debate on between neutralists and selectionists. And although it's been very controversial in the very beginning, the neutral theory has withstood the test of time, and it's now being widely accepted, although not universally accepted. And today, the neutralist selectionist debate is still going on, and now we're trying to use some of the most recent empirical evidence to re-examine the debate. So one of the key premises for the neutral theory is that the beneficial mutation should be extremely rare. But we can do a simple calculation to see how rare that should be. So for example, if we assume the effective population size is 10 to the 7, and the selection coefficient is 0.0025, then we will see that the beneficial mutation rate should be very rare, it needs to be 10 to the 6 times smaller than the regular mutation rate. So that means beneficial mutation rate need to be extremely rare for the neutral theory to be true. And we wonder whether the beneficial mutation is indeed that rare in reality. So we surveyed many papers that investigated the distribution of the fitness effect of mutations. They covered many species: yeast, E. coli, green algae, Arabidopsis, and they used different experimental methods, mainly by deep mutation scanning, but also by other methods like lineage tracking, mutation accumulation, transposon insertion. And all those studies consistently reported a small but significant amount of beneficial mutation, ranging from 1% to 5%, which obviously way exceed the requirement of the neutral theory. And for example, Shen et al. 2022, they investigated more than 6,000 non-synonymous mutations, and they found that 1.6% of them are significantly beneficial, and the distribution of fitness effect of mutations are shown by this blue distribution here. And if we assume that effective population size is 10 to the 7th, then we can theoretically calculate the expected distribution of a fitness effect among substitutions, and it's shown by the orange distribution here. And we can see that even though that there's only a very small fraction of mutations being beneficial, almost all substitutions are beneficial. This is because the beneficial mutation have much higher fixation probability than neutral or deleterious ones. And if we want to be more quantitative, then we can calculate the relative non-synonymous substitution rate, omega, which is defined by dn over d neutral, the non-synonymous substitution rate to neutral substitution rate. And it turns out to be more than 1,000. And it is obviously ridiculous, because it means that functional gene would evolve 1,000 times faster than a pseudogene, which is inconsistent with the empirical observation, which the ratio is typically at 0.1. And we can also calculate the fraction of non-synonymous substitution that are beneficial, which is alpha, and it turns out to be very close to 1, meaning that almost all substitutions are beneficial, which again violates the neutral theory. So does that mean the neutral theory is wrong? So there is actually one explanation that can save neutral theory, which is that maybe the organisms are not well adapted to the lab environment where the fitness is measured. That's why we see so many beneficial mutations. And those organisms should be well adapted to their natural environment. So maybe beneficial mutations is indeed very rare in natural environment. So if this statement is true, we want to verify this. So to do that, we want to firstly show it takes a long time to become fully adapted to a constant environment. And one example is Richard Lansky's long-term evolution experiment. And they found that even after more than 40,000 generations, the population fitness seems to be still increasing. And the omega, which is the DND neutral ratio, is still significantly above 1, which indicates a strong signal of positive selection, meaning that the population is still actively adapting to the constant environment, even after evolution of more than 16 years. And another example is East long-term evolution experiment by Johnson in 2021, where they find that even after evolution of more than 5,000 generations, the fitness was still increasing significantly. So this means that it indeed takes a long time to become fully adapted to a constant environment. So actually the time it takes is too long that an environmental change is very likely to happen before it in the natural setting. So the organisms are typically far from fully adapted to the natural environment before it changes, just like lab strains are far away from adapted to the lab environment. So in principle, we can say that the DFE, the distributional fitness effect we measured in the lab, should be representative for the DFE we measured in nature. So the previous explanation is not valid. So we still need to face this contradiction, which is that there are too many beneficial mutations according to the empirical DFE, which predicts a very high omega. And also Richard